Good morning and welcome everyone to the 2021 American Brain Tumor Association's National Conference. I'm Ralph DeVito, President and CEO of the American Brain Tumor Association. I am so delighted to have many of you join us for both days of the program, and some of you only able to join us for a segment or two. A special welcome to those who are joining us for the first time. The ABTA National Conference is the largest educational meeting designed specifically for patients, survivors, and caregivers who've been impacted by a brain tumor diagnosis. We hold this meeting annually, and we convene the nation's leading brain tumor experts to provide the latest updates on brain tumor treatment, care, and research. This year, we made the decision to hold the meeting virtually like last year during this life-threatening pandemic. We wish we were together in person. There is so much value in the networking. On the other hand, we have so many people joining us that may not otherwise have joined us. So we're going to continue to offer virtual opportunities for people real time and after to watch the recordings. We are hoping to have the 2022 National Conference in person in Chicago. Stay tuned for the big announcement. We had about 1,000 people pre-register, so thank you for doing that. And they registered from all over the country, north, south, east, and west, and throughout the globe. So we welcome everyone, no matter where you are, whether you are joining us from your phone, your iPad, your home office. We welcome each and every one of you. It is just great to have you along for this two-day program that our co-chairs have put together. That is absolutely wonderful. This is an exciting opportunity to share information together as a community. We are all united for the same purpose: to improve, to extend, and ultimately to save the lives of those diagnosed with a brain tumor. It is now my pleasure to welcome Carla Varner. Carla is the chair of the American Brain Tumor Association's Board of Directors, and she has a few words to share. Carla?
Thank you, Ralph. I don't know about all of you, but I couldn't be more excited to be here today. This is a super unique opportunity to become better informed about the ABTA, about brain tumors, and the journey that lies in front of many of you. As Ralph said, my name is Carla Varner. I'm the current chair of the ABTA Board of Directors. I joined the board in 2014 after two close friends of mine lost parents to a brain tumor. And since that time, not through my volunteer efforts with the ABTA, but just in my personal life, I have encountered numerous individuals who have been diagnosed with, or someone that they know that they're close to has been diagnosed with a brain tumor. And from my perspective, if even one family's journey is made easier as a result of the ABTA research funding and the caregiver support services and patient support services that we offer, then my time as a volunteer is well spent. For those of you who don't know the history of the ABTA, I just want to talk to you a little bit about it. The ABTA emerged 47 years ago by Susan Kramer and Linda Goldstein, both mothers whose daughters, very young daughters, were diagnosed with a brain tumor. Back then, there were limited educational or support resources available, and brain tumor research funding was abysmal. Susan and Linda were determined to build an organization to serve the patient through information, support services, and research. There's truly nothing like the power of two mothers trying to help their children. I share this with you simply because I want you to know that this conference is uniquely designed for you. By bringing together medical leaders from all over the world, truly all over the world, we hope to provide you with the most accurate and relevant information to inform you, but most importantly, to empower you. We hope you find the information presented today and tomorrow helpful and meaningful to your brain tumor journey. Enjoy the conference, everyone. Thank you very much.
Thank you so much, Carla. And thank you so much for sharing how you are connected to the brain tumor community. I want to take a few minutes to acknowledge those who have helped create this wonderful conference. Thank you to all of our speakers whom you will hear from throughout the weekend for sharing the thoughtful insight and expertise tailored to the patient and caregiver experience. Thank you to our sponsors and funders for their support of this conference. I want to especially thank our titanium sponsor, Northwest Biotherapeutics, and our platinum sponsors, SDP Oncology and Novocure, for their support. Thank you to our researchers whom you will hear from during our breaks throughout the weekend. And last, but certainly not least, I would like to thank our conference planning co-chairs, Dr. Sean Hervey-Jumper and Dr. Avanthia Galanis, who have put together an incredible program bringing together national experts from top medical institutions to share with you today. Dr. Sean Hervey-Jumper is an associate professor of the Department of Neurological Surgery, director of the Glial Tumor Neuroplasticity Research Laboratory, and co-director of the Sherry Sobrato Brisson Brain Cancer Survivorship Program at the University of California, San Francisco. Dr. Avanthia Galanis is a professor of oncology and the Sandra J. Schultz Professor of Novel Therapeutics at Mayo Clinic. She is also the chair of the Neuro-Oncology Committee at the Alliance for Clinical Trials in Oncology. It truly has been a pleasure to work with them throughout the past few months to putting together this national conference. Please welcome Sean and Eva to share a few words. Sean, please go first.
Good morning. Thank you so much for this opportunity to be part of the ABTA community. I have to say that this meeting is deeply personal for me, as I'm sure it is for many of you. One of my earliest memories was taking care of my grandfather, Perry, during his medical journey. So I was introduced to medicine as a little guy, as a child, taking care of and being a caregiver with my grandmother, Charlotte, for my grandfather who was receiving treatment for a tumor within his abdomen. So attendees, this is all about you. We want to arm you with information, facts, and encouragement. And I echo Carla, Ralph, and everyone else's comments. Please send us your thoughts and questions over the next couple of days, and thank you all for tuning in.
Maybe I should start then. Good morning, and thank you so much for joining us today. My name is Eva Galanis, and I'm a medical neuro-oncologist at the Mayo Clinic. In addition, as Emily said, I'm leading a large cooperative group effort in neuro-oncology through Alliance. I have to say it has been a true pleasure and honor to work with Sean, Emily, Nicole, the ABTA staff in putting this program together. Thinking about brain tumors, both primary, starting from the brain, and metastatic, spreading to the brain. For a long time, we thought of them as a big black box, which we knew they were horrible, but we did not really know what was causing them. Now we have started demystifying this black box. And this is the focus of our conference, how understanding biology will allow us to develop better treatments, matching the right patient with the right treatment, and also understanding why a treatment does not work or why a treatment has stopped working and try to reverse that. And while we are doing all that, the focus always is our patients, our caregivers. And it's not about quantity. We all know that way too well. It's equally important about quality. So we all very much hope that you're going to find the program today enjoyable and informative. And on behalf of the organizing committee, we thank you very much for being our inspiration. Thank you.
Thank you so much, Sean and Eva. We would not have been able to bring you this quality educational programming without our co-chairs' guidance and support. You will hear more from our co-chairs throughout the two-day conference. I'm now happy to introduce our first session about the American Brain Tumor Association, who we are, what we do, and how you can get involved. Please welcome ABTA Chief Mission Officer, Nicole Wilmer.
I'm really excited to talk to you all today and share more about our mission. As Emily mentioned, my name is Nicole Wilmarth. I'm the Chief Mission Officer for the American Brain Tumor Association. And today, sorry, went forward one too many. Today, I'll provide some perspective on how far we've come since our organization was founded 48 years ago. And then I'll give an overview of the ABTA's mission and how we work every day to fulfill it. And then we'll go over our research program and services for patients and caregivers. And then I'll talk about some ways that you can further get involved with the ABTA. So the ABTA and the brain tumor field in general has come a long way in the past 48 years. Back then, there was no coordinated funding of brain tumor research. So our organization was founded on the need to support brain tumor research, and it's now our legacy. There also was no medical specialty that was dedicated to brain tumor treatment. So the specialty of neuro-oncology didn't exist at that time. So brain tumor patients were treated by a neurosurgeon and a medical oncologist. And now having neuro-oncologists with specialized training has really helped improve brain tumor patient care. There also was no national tracking of the incidence and the prevalence of brain tumor diagnoses, which means that we didn't really know how many people were impacted by a brain tumor. But now we have the Central Brain Tumor Registry of the United States, or CBTRUS, which tracks cancerous and non-cancerous brain tumors. And there also wasn't any national education, awareness, or funding programs that were focused on brain tumors. However, I will soon go into detail about the programs that we offer. And so today, all of these exist, and ABTA is proud to have played an important role in helping to close these gaps. But we also know that there's still much more to do. So that brings me to our mission. And for those of you who aren't familiar with the American Brain Tumor Association, our mission is to advance the understanding and treatment of brain tumors with the goals of improving, extending, and ultimately saving the lives of those impacted by a brain tumor diagnosis. And the ABTA takes a multifaceted approach toward our mission. We focus on 4 key areas: providing public education and awareness of the impact of brain tumors, delivering best-in-class patient and caregiver support, collaborating with other organizations and medical institutions through strategic partnerships in order to advance our research and our patient support efforts. And then also investing in research to discover better ways to diagnose, treat, and hopefully cure brain tumors. So now that I've given you a little background on our organization, I wanted to jump into our mission and action. So I'll start with research. And we have 2 main priorities, I would say, through our research program. The first one is to foster a new generation of scientists who will make important discoveries involving the causes, the effects, diagnosis, and treatment of brain tumors. And another is to fund high-risk, high-impact research that will lead to breakthroughs. Some examples of the areas that we fund are listed here on the slide: drug development, targeted therapies, imaging, biomarkers, immunotherapy. Really, we have a very diverse portfolio of research projects that we fund in many different topic areas. And for our research program, we primarily fund through different research grant opportunities. And each one is launched as an open call for applications, as we call it. Our applications go through a very rigorous peer review process by experienced scientific experts in order to select the best projects to fund. Each grant opportunity is different based on the level of investigator and on the term limits and the amount of grant funding. So I'll just briefly walk through the 4 different grant opportunities that we offer. The first is our basic research fellowships. The basic research fellowship is a 2-year, $100,000 grant for postdoc fellows who conduct laboratory or field-based research under the guidance of a mentor. The next is our research collaboration grant. And the research collaboration grant is the ABTA's newest grant mechanism. It was designed to foster collaboration. And this 2-year, $200,000 grant is awarded to scientists from different institutions to bring together different disciplines and viewpoints. And these are our largest grants and are typically offered every other year. The Discovery Grant is a 1-year, $50,000 grant for high-risk, high-impact research with the potential to change current diagnostic or treatment models. And a lot of times these grants act as seed funding for investigators to get a federal grant. And then lastly, we have the Jack and Faye Netchin Medical Student Summer Fellowships. And this is a $3,000 grant given to medical students who are looking to conduct brain tumor research during the summer break under the guidance of an esteemed mentor. And since our organization started, we've awarded over $33 million in brain tumor research. So we're really proud of that. And this includes over 760 research projects that we funded throughout the US and globally, and 678 different researchers that we've supported. We also fund research across all tumor types, and that means primary to metastatic and all ages, from pediatric to adult. And we believe it's imperative to not only support research, but also to continue to support early career investigators. They are entering the brain tumor research field and are the future of brain tumor research. And they bring many of the innovative, outside the box ideas that will hopefully lead to the next discovery. And so one way that we continue to support them is through our ABTA Alumni Research Network, or we call it the AARN. And we started this network back in 2012, and we typically hold a in-person 2.5-day meeting every year where we bring together our research alumni. But given the pandemic for 2020 and 2021, our AARN meetings have been virtual, but we were still able to bring together our members where they learned about brain tumor science, they network, they share ideas and resources, and then they gain knowledge around career development skills as well. And over the years, what we've seen is that several collaborations grow organically out of this network. And as you can see here, we've noted 71 collaborative groups have formed out of our AARN members. And these collaborations have led to an increasing number of joint research projects, as well as publications. In addition, we continue to engage our research network through professional growth opportunities, such as having them serve as reviewers for our grant applications or speaking at ABTA events. We also think it's important to collaborate with other groups outside of the ABTA to support research. For example, we're part of the Brain Tumor Funders Collaborative, and this is a partnership between private philanthropic and advocacy organizations to pool our funding to invest in studies that are larger than we can fund individually. And currently, the BTFC is collaborating on a study of long-term survivors of GBM to learn more about what makes them unique. In addition, we awarded $30 million to 4 different teams of researchers who are studying immunotherapy in primary brain tumors. We've also provided initial funding for a low-grade glioma patient registry to look at the response to treatment and clinical outcomes within this patient population. So stay tuned, because you'll be hearing an update on that project in a video later during one of our breaks. All right, now I'd like to move on to our patient services and support. And I will start with our website. So the ABTA website provides over 200 pages of content that has information on diagnosis, treatments, side effects. The content is vetted by experts in the field so that we can deliver accurate and trusted information to patients and caregivers. We also have resource centers for caregivers and listings of brain tumor treatment centers and listings of support groups. So please go online if you haven't already and take a look. Another resource that the ABTA offers is our vast array of educational materials. We have 18 printed educational brochures, which we provide and ship to patients and caregivers free of charge. The brochures cover a variety of topics, such as specific treatments like chemotherapy and radiation, or individual tumor types such as glioblastoma. There's even a brochure on clinical trials. And if you aren't sure which brochure to get, the primer, which is also known as the About Brain Tumors brochure, has a lot of helpful general information. And this was our first brochure back in the early 80s, and it remains a popular educational pamphlet today. All of our brochures are available for download as PDFs on our website, and some are even available in Spanish. You can also order your copy of whatever topic interests you through our online order form. And if you have questions, our Careline staff can help you to complete your order. So feel free to contact us. Speaking of our Careline is the ABTA phone and email information service. The purpose of the Careline is to really provide support, information, and resources to brain tumor patients, caregivers, and healthcare professionals. Our calls are answered by our trained and caring staff, and they have one-on-one discussions with patients and their family members. If you have a question about where to find information, this is a great place to start. The ABTA gets calls that range from those who need immediate emotional support to those who are seeking financial aid resources. We also get calls from healthcare providers and allied healthcare professionals who are asking about resources for their patients. And although this is a service that, this is not a service to offer medical advice, it is a service that can guide you on what questions to ask your doctor. Our Careline staff can also point you to some evidence-based articles on a topic of interest. Our hours are 8:30 to 5 p.m. Central Time, so please reach out to us. We're here to help. The next service that I'd like to highlight is Connections, which is ABTA's online support community hosted on Inspire. Connections is a private space where brain tumor patients, survivors, and caregivers can come together to share their experiences and support each other. For those who wish to have 24/7 access to support and who want to find others who can relate to your situation, this is a great way to connect. One benefit of Connections over other social media platforms is that it is confidential. You create a username and you don't have to disclose your personal information. Additionally, you can search by topic instead of scrolling through many posts to find what you need. Connections actually started back in the 1990s as a pen pal program, and it was initiated for patients and caregivers to connect with others in similar situations, but it has since grown online to over 25,000 members. So very popular. Next, we have our webinars. If you haven't joined us for one of our webinars, I'd highly recommend tuning in. We host webinars almost every month of the year, and they are live presentations from medical experts in the field about topics requested by patients and caregivers. These are typically 1 hour long. There's a live Q&A portion where attendees can ask questions of the experts. In addition, the webinars are recorded and saved so that you can go back and watch anytime at your own convenience. Topics are wide-ranging, from diet and nutrition to managing symptoms to advances in treatment. And if you want to watch any of our past webinars, you can just search American Brain Tumor Association on YouTube, and you can locate our Anytime Learning channel there. And we do have a webinar coming up on October 28th, so I want to point that out. It's called Survivorship Clinic, and we invite you to register and tune in to learn more. I also wanted to mention our patient and family meetings. These meetings are designed for patients, caregivers, and healthcare providers to network and learn from one another through physician presentations, through patient and caregiver panels, physician tumor boards, and other topics of interest. They're free to all patients and caregivers. And typically these meetings are held in person around the country. However, given the COVID pandemic, we have offered 3 virtual patient and family meetings this year. These meetings are held online so that patients and their families can tune in remotely from all over the US or globally. And our next virtual patient meeting is focused on the topic of survivorship, where you can learn to maximize your quality of life as a brain tumor survivor. Sessions will include topics such as adolescents and young adults and integrative approaches to your care. So we invite you all to register for the event, and it's taking place virtually on November 6th. And then our national conference, which you are all attending today, is our largest conference dedicated to patient and caregiver education. In fact, for this conference, we had over 1,000 registrants from all over the globe. It's typically a 2-day meeting held in person in Chicago, but for 2020 and 2021, we have been virtual due to the pandemic. However, please save the date for our national conference next year, which will be held September 9th and 10th. And we're hoping to be able to offer this as a hybrid of both in-person and virtual content. Our goal for the national conference is really to provide patients and caregivers with access to nationally recognized brain tumor experts to learn more about treatments, disease management, and support services. And we really bring together the whole brain tumor community, not only patients and caregivers, but also researchers, healthcare providers. And after the conference, presentations will be made available through our YouTube learning channel. And I do also want to point out for this year's national conference that our keynote speaker will be on later this afternoon. Her name is Courtney Burnett, and she has a unique perspective given that she's a physician that essentially diagnosed herself with a brain tumor. Her story is fascinating and motivating, so I encourage you all to tune in. And then if you're interested in one-on-one support, our peer mentoring service might be a good fit for you. Through our peer support program, we match mentors and mentees based on their diagnosis and encourage support through email or phone communication. And participants in this program say that it can be very helpful to know that they're not alone and can speak with someone who has walked a similar path to theirs. It can also be very rewarding from their perspective to share experiences with someone in need. We have mentors that have experience with a variety of tumor types, including metastatic brain tumors. And as you can see, to date, we've trained 215 patient and caregiver mentors and have matched 800 mentees. So this is a very popular program, and it's also unique. I encourage you to check it out. I also wanted to briefly mention our metastatic brain tumor initiative. Metastatic brain tumors are the most common type of brain tumors in adults, with estimates around 100,000 to 200,000 diagnosed annually. And metastatic brain tumors form when cancer from another part of the body spreads to the brain. And we realize that a lot more is needed to be done in order to address this large underserved patient population. And therefore, we created the Metastatic Brain Tumor Initiative to help inform, support, and empower patients who are diagnosed with a metastatic brain tumor. And since initiating this program, the ABTA has created several new educational resources. Those include a dedicated patient family meeting, a free brochure, and helpful questions to ask your doctor. In addition, we continue to support brain metastases research projects that will help accelerate treatments for this patient population. Our efforts in this area are growing through partnerships that we formed with 5 other organizations in the areas of lung, breast, kidney, colon, and melanoma cancers. Together, we collaborated on the first survey of patients, caregivers, and physicians, which allowed us to shed light on the disparities in care for these patients. So how can you get involved? If you're interested in giving back to the brain tumor community and would like to get more involved with ABTA, there are several ways to do so. First, you can participate through our ABTA events. For example, Team ABTA is our endurance running and cycling program. You can get involved as a participant or you can volunteer your time. You can also form your own fundraising event through our Fundraise Your Way opportunity. People across the country, they host their own fun events like wine tasting or bowling or golf tournaments. The possibilities are really endless. And our BT 5K run walks are another option. These are considered one of our signature fundraising programs. These are typically in person, but due to the pandemic are now conducted virtually. And the nice thing about being virtual is that anyone, anywhere can participate. You can walk in your apartment, you can walk up your stairs, you can walk through your neighborhood. And we have 3 walks remaining this year. You can see they're September 25th, October 2nd, and November 7th. So you can sign up as an individual or as a team and help us to raise funds for research and patient support services. And if you're looking to give back in other ways besides fundraising, the ABTA has many different volunteer opportunities. First, you could volunteer to help promote and plan the Breakthrough for Brain Tumors 5K run walks. Each BT 5K has a committee of volunteers that you can participate in. Another opportunity is through the ABTA's mentor program that I mentioned earlier. If you're interested in becoming a mentor, you can reach out to us. You could also be an event-based volunteer. These volunteers support ABTA hosted events, including the BT 5Ks, but also our patient meetings and our national conference. Our outreach volunteers, they work in their own communities to raise awareness about ABTA's programs, services, and resources. And they do this through distributing materials and representing the ABTA at conferences and local events. And then also there are skill-based volunteers. So these volunteers have specific skills that are often needed for special projects with the ABTA. So for example, graphic designers, photographers, videographers, etc. So if you're interested in volunteering through any of these roles, we're happy to get you involved. Just reach out to us. And finally, I wanted to encourage you all to stay connected to the ABTA so that you're aware of upcoming news, events, and programs. You can sign up for our Mind Matters, which is our electronic newsletter, using the link that's on this slide. I also encourage you to follow us on social media. We're on Facebook, LinkedIn, and Twitter. And in closing, the ABTA is here to support you every step of the way. Thank you for attending the meeting and for listening today as I shared a bit about the ABTA mission. And I hope you enjoy the rest of the national conference. So thank you.
Thank you so much, Nicole, for sharing with us about the mission of the American Brain Tumor Association. For those of you who need a break, please take this opportunity to stand up and stretch your legs. We invite the rest of you to stay on to hear a research update. We will return in just a few minutes for the next session on Know Your Tumor. Thank you so much.
My name is Javier Gans. I'm a postdoctoral fellow at Boston Children's Hospital and Harvard Medical School in the lab of Christopher Walsh. In our lab, we study human brain development, aging, and the effect of mutations, which are alterations in the DNA. I want to take a moment to thank the American Brain Tumor Association for the 2019 Basic Research Fellowship Grant in honor of Joel A. Gingras, Jr. The research I will present today is about trying to understand what happens in the cells before we are diagnosed with a brain tumor. Oncogenesis is the process through which healthy cells become transformed into cancer cells. It is characterized by genetic alterations, leading the cell to divide in an uncontrolled manner. As can be seen in the illustration, a healthy cell may acquire a mutation in the DNA, product of environmental factors, but also product of DNA errors that arise because of normal cellular processes, such as cell divisions during development. These mutations may affect important genes regulating cell division, and then cells may start to proliferate more. With more proliferation, also more mutations will be acquired, and more genes will be affected until cells can transition into a malignant state. Finally, when these cells achieve a considerable size to be detected, an individual will be diagnosed with cancer. We decided that in order to understand early steps in brain cancer initiation, we need to study what is the status of non-diseased individuals and not the brains of subjects already diagnosed with cancer, since oncogenesis started long ago, and those first steps or first mutations might be difficult to identify. With this research, we wanted to answer about the existence and behavior of oncogenic mutations in the non-diseased brain. We designed a two-arm experimental approach to answer those questions. First, we evaluated mutations in cancer genes in different brain regions from 110 individuals. Second, we evaluated 1,800 brain samples from databases containing fetal, newborn, young, and adult brain samples. And after integrating all our results, we learned that mutations with cancer-causing potential exist in the non-diseased human brain, that cancer mutations are mostly present within the white matter and are enriched in glial cells, that brain cancer mutations are found in young individuals, including fetal, suggesting that these mutations are acquired during development, and that the pattern of mutations observed in brain tumors can also be observed in normal tissue, indicative that normal mutational process of the brain drive brain oncogenesis. Our findings were published recently in Cancer Discovery, which I invite you to read if interested in the topic. So our work is important because we provide information on early processes in the acquisition of oncogenic variants in the aging brain, which are essential to understand brain tumor origins, improve diagnostic baselines, and tailor therapeutic interventions targeting early mutations. I also want to thank my colleagues, mentors, and institutions, and supporters such as the American Brain Tumor Association for making this work possible. Thank you very much.
Participants in the American Brain Tumor Association. That's why these tumors were chosen. So I think everybody in the audience is familiar with this flow of either yourself or your family members that have had a brain tumor. The patients develop symptoms, whether that's headaches or seizures or personality or vision changes. They go into the primary care or oncologist and get an image, and the image shows a lesion such as this one, a T2 bright lesion in the frontal lobe. It's then decided that that either needs to be biopsied or resected. And here is a neurosurgeon taking a specimen from the brain, and then eventually a diagnosis is made. Often that is a black box, and there's not often the understanding of how that process occurs. So I thought I would take you through that a second and show you what actually did happen to your tissue to provide a diagnosis that could then guide therapy. So once the tissue is biopsied or resected, it's put in a sterile cup by the surgeon. It's then transferred to a laboratory within pathology. We call this the gross room because these specimens are analyzed in their gross state. That means non-microscopic state. So they're evaluated, described, they're sampled, and then the sampled tissue is submitted into this cassette. This cassette then gets turned into a tissue block. You may have heard about tissue blocks. Once the tissue is fixed in formalin, it gets embedded into wax. It's paraffin wax. And then thin sections of that tissue are sectioned, 5-micron sections, and put on glass slides. And then we apply these purple and pink stains to them, hematoxylin and eosin. And then once the slides are made, then they go to the pathologist who looks at the slides under the microscope and determines what histologic class the tumor is in. Now, in 2021, this also involves ordering of additional tests, almost without fail. And I'm going to go over what some of those tests are because they're absolutely critical in establishing a definitive diagnosis. So for many, many decades and over a century, actually, we classified tumors under the microscope based on what they look like, essentially, the histologic classification under the microscope. So most physicians and many patients know that this is an MRI and know that this is a T2 bright lesion in the left mesial temporal lobe. However, looking at the tissue is not common knowledge to most physicians or patients, and this is our expertise in neuropathology. So what we're seeing here are individual tumor cells under the microscope that have the purple oval shape to them. There are some normal neurons in here of the native brain that are being infiltrative. So this is an infiltrative glioma. And based on what it appears like under the microscope, we would call this an astrocytoma based on histologic classification because the tumor cells look like astrocytes, a normal cell of the brain. However, this tumor could just as well be represented by this tumor type. Now, this tumor type looks different. It has these round, smaller nuclei surrounded by clear cytoplasm. They're also infiltrating the brain, but in this case, the tumor cells look like oligodendrocytes, and this would get called an oligodendroglioma by histologic classification. We also, in pathology, have this strange habit of naming cells by what they look like. So can anybody guess what food item these have been called? They've been called fried egg cells in the past because they look like fried eggs, and that's a helpful teaching point to our trainees. So that's histologic classification. And again, we've been doing this for over 100 years. So a neuropathologist, Percival Bailey, and Harvey Cushing, a neurosurgeon, put together a classification back in 1926 that uses the same histologic class and the same terms that we use today. And in fact, we used this type of diagnosis until 2016. You can see the term glioblastoma, astrocytoma, oligodendroglioma, and we still use those terms. One of the problems with this classification system is that it's very subjective. And there was variability in what pathologists would call an astrocytoma, or variability in the grade that would be applied. And it led to some challenges for clinicians like Martin van de Bent from the Netherlands, who wrote an article on some of the issues with inter-observer variation with histopathologic diagnosis. And his point was, we really can't tolerate or accept such lack of reproducibility, because we treat patients with these diseases much differently. If they have a grade 2 oligodendroglioma versus a grade 3 astrocytoma. So we need some consistency in diagnosis. And really the data support the concerns of the neuro-oncology community. This shows clinical outcomes based on histologic assessment of the tumors. And you can see that these survival curves are all intermixed. That means that we can't stratify very well the patient's risk based on histologic assessment. If we did, these curves would be much more separated than they are, but in fact, they're intermingled. So we're not doing a good job of stratifying patients based on histologic assessment. Fortunately, the last decade has brought us a far better understanding of the molecular underpinnings of the diffuse gliomas in particular, and other forms of brain tumors. So very high-ranking journals like the New England Journal of Medicine and Cell and Nature have published very impactful articles on the molecular characteristics of specific tumor types, such that now we are able to integrate some of these molecular genetic findings into our diagnosis. We incorporate both molecular and histologic characteristics into a final diagnosis. And I'd also like to take you through what that looks like. So, for example, in the upcoming World Health Organization classification of diffuse gliomas of 2021, you can see the names of the tumors, very much unlike the tumors of the Bailey and Cushing classification. These all include a gene that kind of defines the disease. Glioblastoma, IDH wild type. IDH is the gene. Astrocytoma, IDH mutant, and we grade 2 through 4. Oligodendroglioma, IDH mutant 1p19q co-deleted. So, and then there's others as well, and you can see how we integrate the molecular findings with the histologic class, and that provides a much more reproducible diagnosis and does stratify risk much better, which I'll show. Now, how do we obtain this type of information? Well, you remember that I talked about cutting tissue from the blocks in order to create slides. Well, we cut additional sections from that tissue. We use the same tissue and send the tissue for molecular analysis. And that analysis can be done within the same institution, or it can be sent out to a large reference lab. And really, it has become standard of care for diagnosing diffuse gliomas in the current era. We do that by gene sequencing. You may have heard of next generation sequencing, or NGS. We look at chromosomal level changes like amplifications, deletions of genes. And then a newer type of assay is DNA methylation of the entire genome to look at a methylome-based signature that I'll just touch on. But it is, I think, going to be the future of cancer diagnostics, certainly for brain tumors and sarcomas, and most likely for others. So again, here is what a diffuse glioma looks like by MRI. It's a flare or T2 bright expansive lesion involving the brain. And under the microscope, normal brain looks like this, very orderly. These oligodendrocytes are lining up here and here. The axons that belong to the nerves and are the wires of the brain are here in the pink, as well as other processes from the glial cells, the glue that holds the brain tissue together. In the disease state, the diffuse glioma, these tumor cells are infiltrating through the brain parenchyma, the brain tissue, and causing this edema. The white bubbly material in the background is water-like substance that is a reaction to injuries of all sorts, and that's what causes this T2 or flare brightness and causes the expansion. So this is clearly an infiltrative glioma, and then how do we determine what type it is using molecular data? Well, first we do gene sequencing, and this colorful and somewhat complicated image shows about 160 different types of diffuse gliomas, and each column is a different tumor. And you can see that when we look at the mutations, which are listed here, the mutations segregate these diffuse gliomas into three families. One shown here is IDH mutant and 1p19q co-deleted. We've redefined the term oligodendroglioma to require that these tumors are IDH mutant and 1p19q co-deleted. And now you can see that their mutational spectrum is very definitive of that diagnosis. Those tumors that have IDH mutations but don't have the 1p19q co-deletion, they're characterized almost 100% by p53 mutations and ATRX mutations. Now these fit into the category of IDH mutant astrocytomas. And then we grade those after having placed them in their category based on their molecular profile. Those tumors that don't have IDH mutations or 1p19q deletions are called IDH wild type gliomas or astrocytomas, and they have the genetic alterations that are typical of glioblastoma. So the mutation that I'm talking about most often is the IDH mutation. And what we have found is that IDH mutations are found in a specific subset of diffuse gliomas, and that subset are mostly from younger adults. They mostly involve lower grade gliomas and secondary GBMs that evolve from those lower grade gliomas, and they're characterized by improved prognosis, longer clinical outcomes. IDH wild type gliomas, on the other hand, occur mostly in adults with a median age of 62, they're mostly primary GBMs, and their prognosis is not as good as the IDH mutant gliomas. And this graph just shows the tall bars that the lower grade gliomas tend to have the IDH mutations, whereas glioblastomas do not. Now, that's all interesting, but what really turned our field on its head was the finding that not only do IDH mutated tumors have a better prognosis when they were glioblastomas, but the grade 3 IDH wild type astrocytomas actually had a worse prognosis than the IDH mutant glioblastoma. So this began a long journey in now understanding that these two diseases, which looked the same under the microscope, were very much different, and that we really needed to incorporate molecular findings into our diagnosis. The same is true for looking at amplifications and deletions of chromosomes. So again, these are the three categories of tumors that fall under the diffuse gliomas. You see the oligodendrogliomas shown here in light purple. They all have 1p loss, which is shown in blue, and 19q loss at the chromosome level. That again has become the definition of that disease, unlike before where it was the appearance under the microscope. Similarly, IDH wild type gliomas have losses of 10 and gains of 7. Gains of 7 are shown in red here. So these, again, they segregate these tumor types, not by what they look like under the microscope, but by their molecular and chromosomal alterations that define them. We also showed that not only do these tumor types have specific genetic characterizations, but on the right here, we showed that we could stratify risk much better than by looking under the microscope. You remember that tangled web of survival curves that I showed earlier? Well, if we define tumors by their molecular characteristics, we see much more cleanly delineated survival curves, with oligodendroglioma 1p19q co-deleted having the best overall survival, and IDH mutant astrocytomas of grade 2 and 3 having the next best survival, and IDH wild type tumors, whether they're grade 4 histologically or grade 3, having the worst long-term survival. So we've gotten to the point where we can, by molecular classification, do a much better job of stratifying the risk for an individual patient and hopefully pointing them to the most appropriate therapy based on that. The next era of diagnosis, I believe, that we'll have in the next 10 years will involve not necessarily sequencing for mutations and chromosomal losses and gains, but determining what tumors look like by their DNA methylation fingerprint. And that's what this interesting classification is showing. Now, we're not talking about mutations segregating these. We're not talking about histology. We're talking about the methylation of genes. And methylation turns on and turns off genes. And that is probably why their fingerprints are so different depending on the tumor type. So here you can see that even the disease glioblastoma IDH wild type has a number of different methylation fingerprints shown in green, whereas the IDH mutant tumors are very segregated for that from them. And all of these other tumors that you may have heard of, like ependymoma, meningioma, medulloblastoma, they're all segregated in space. And this type of separation has allowed this type of assay to become one of the gold standards in establishing a diagnosis. This has been really advanced by the group in Heidelberg, led by Andreas von Deimling and Stefan Pfister. And this article in Nature was led by David Kapper. And they've really, I think, done the neuro-oncology field a tremendous service, because if we have a discrepancy among our group of neuropathologists on a challenging case, we can send it or do our own methylation profiling to get a much, I think, more specific classification of tumors. I'd like to go over a couple of examples of how we make a diagnosis on a daily basis, so that you have an understanding of the flow of the tissue and the workflow that we did in diagnosing in order to establish first a preliminary diagnosis and then a definitive diagnosis. So this patient is a 34-year-old male who became tired over the past 6 months. An MRI revealed a left frontal lobe lesion that was T2 bright. Again, I showed the same image before, but without contrast enhancement. That typically signals that it's going to be low grade. We had the slides delivered and looked under the microscope, and we see the brain parenchyma in the background here with axons, the wires of the brain, with these tumor cells infiltrating through. These are not normal cells. You can see how dark and angulated they are. So we know that there's an infiltrative glioma present, but we also know that rather than provide a microscopic diagnosis, we need to do some additional testing. So many of the genetic alterations that are known to occur create a protein that is mutant that can be recognized by an antibody, by immunohistochemistry. And for IDH mutations, we are able to actually identify the mutant protein by doing an analysis on the tissue sections themselves, immunohistochemistry. And the dark brown here indicates a mutant IDH1 of a specific type, the R132H mutation, which is the most common and accounts for about 90% of all IDH mutations in diffuse gliomas. So now we know that this is an IDH mutant tumor, which we may have suspected because it looked low grade by the MRI, but this is definitive. You'll remember from that image I showed of the mutational spectrum of the diffuse gliomas that ATRX is mutated or lost in almost every IDH mutant astrocytoma. And in this case, the brown stain is recognizing normal cells. It's a stain for the normal protein, but in the tumor cells, the stain is lost. So this to us means it's an IDH mutant astrocytoma with a loss of ATRX, which suggests the diagnosis of IDH mutant astrocytoma. And then we also get a P53 stain. And again, you remember these tumor types were almost all P53 mutant, which we can identify by the overexpression of P53 in the tumor cells. So now we know that this tumor is characterized by IDH mutations, has loss of ATRX, and has a P53 mutation. And our criteria in 2021 allow us to call this an astrocytoma, IDH mutant, WHO grade 2. And then this information will be passed on to the patient and their families and the caregivers for the most appropriate therapy, which would involve a long discussion with the neuro-oncologist. A second case is shown here, a 41-year-old woman who developed new onset focal seizures and difficulty speaking. MRI revealed a non-enhancing right parietal mass lesion. Now, again, this MRI appearance could be consistent with a number of diffuse gliomas. When we looked at it under the microscope, we saw these rounder nuclei that were really packed together within the brain, still infiltrating, but it's really almost entirely neoplastic cells packed back to back. And here's some thickened blood vessels in the background. So I would think this looks like a high-grade oligodendroglioma, but again, we want to take this through our molecular genetic analysis, again, using immunohistochemistry as much as we can, because we get a one-day turnaround using immunohistochemistry for this testing, whereas next generation sequencing typically takes 2 weeks. And we do follow up with next generation sequencing, but we almost always start with immunohistochemistry. So this, again, the brown is positive. That shows that this tumor is IDH mutant. The ATRX in this case is not lost. It's retained in every single cell. That would be more consistent with an oligodendroglioma. And there's no P53 staining, so most likely not an IDH mutant astrocytoma. Now, in this case, we'd still be favoring oligodendroglioma, but in order to make that diagnosis, we need to look at the chromosomes. And in particular, in this case, we saw a loss of 1p and 19q. And it's now well recognized by work by many, including Bob Jenkins, that this specific alteration is due to a translocation that leads to these chromosome arms being lost, very specific to this disease. So with this specific finding, we can call this tumor oligodendroglioma, IDH mutant, 1p19q co-deleted, WHO grade 3. And again, this diagnosis is then passed on to the clinicians, the patients, and their families for a discussion of the best therapeutic option afterwards. And finally, the last case that I'll show for the diffuse gliomas is a 50-year-old female noted tingling in her right arm. MRI revealed a large left parietal lobe lesion that was T2 bright and showed patchy contrast enhancement shown here. Now, this patchy contrast enhancement is suggestive that this might be of a higher grade, but we will look at the tissue and see that on the smear preparations, you can see these elongated nuclei and fibrillary processes that suggest a diffuse glioma. Similarly, neoplastic cells are infiltrating through the brain and have astrocytic morphology. When we do the IDH staining, however, you can see that there is no positivity. So based on immunohistochemistry for the mutant IDH1 protein, this one's negative, or we call it IDH wild type. Wild type actually means the normal type. I don't know the origins of wild type, but it stands for normal type. We would then send this for additional sequencing because there's other types of IDH mutations aside from this R132H that might lead it to be an IDH mutant tumor. In this case, those results were negative. So this is truly IDH wild type. I'm sorry. It advanced a little bit more quickly than I wanted it to. So we think this is probably a high-grade IDH wild type glioma, but in order to make that diagnosis, since it's such a significant diagnosis, we want other molecular findings. In this case, the tumor was sent for next-generation sequencing and chromosomal array analysis. And the chromosomal array analysis showed that there was EGFR amplification. Well, that's a new molecular signature for glioblastoma. If an IDH wild type diffuse glioma has an EGFR amplification, we now consider that to be a glioblastoma IDH wild type WHO grade 4 tumor. And that's a relatively new advance. Historically, a tumor like this would be diagnosed under the microscope by the presence of necrosis and microvascular hyperplasia as grade 4. Now we've added 3 other criteria: EGFR amplification, gain of chromosome 7, loss of 10, or TERT promoter mutation, which is sluggish to show up, but there it is. Either one of these genetic alterations in a diffusely infiltrative wild type glioma would lead to the diagnosis of glioblastoma IDH wild type WHO grade 4. So that's a major advance that not only classifies but grades tumors by the absence or presence of genetic alterations. A big step forward. Now, many of you have probably also heard of the test MGMT, methylguanine methyltransferase, which is quite a mouthful, so we tend to say MGMT. And this is a gene that, when it's methylated, leads to its lower expression. It's silenced, if you will, by promoter methylation. And this enzyme usually undoes the effects of chemotherapy. So the chemotherapy cross-links genes and proteins. This enzyme undoes it. So the lower levels that are present in the tumor cells, the better the chemotherapy can do its job in being cytotoxic to the tumor cells. So we often get this genetic test as well, mostly for IDH wild type gliomas, but some physicians will also order MGMT for IDH mutant tumors, because this marker, when the gene is methylated, predicts a better response to chemotherapy, temozolomide chemotherapy in particular, for IDH wild type glioblastoma. So I'm going to switch gears a second away from the diffuse gliomas to actually a more frequent type of tumor, the meningioma. Now, meningiomas aren't occurring within the brain substance itself. They arise from the dural covering, shown here. So this is the dura, which is the protective covering of the brain. It's called dura mater, tough mother, because it forms a barrier that's watertight around the brain. These tumors arise from the dura mater and push brain aside. They tend to be contrast enhancing by imaging, this very white appearance. They occur most often in women that are over 50 years old. They're often slow growing, and 80% of them, or 75 to 80%, are grade 1. And when there can be a complete resection, that's associated with improved clinical outcomes. So you can imagine that the surgeon could go in here and create a plane between the brain that's being pushed aside and the meningioma and entirely remove it. Sometimes that's more challenging than others, but here is an example of a meningioma with a very smooth surface that appears to have been completely resected. And this is the dura mater that it's attached to over the surface. Now, there's a lot of prognostic factors for meningioma. One is the extent of resection. Tumor that's left behind tends to recur. That's a truism for all types of brain tumors. The histologic grade determines outcome or prognosis. The patient age does as well, maybe not like you may think. Actually, although this is a tumor of older individuals, young children actually have more aggressive tumors as a whole. And patient gender is also important, because the meningiomas that men get tend to be more aggressive than those in women. Tumor location can be related to extent of surgical resection. And then, as we'll touch on, we now know, again, based on a lot of research that's been done in the last decade, that molecular genetic alterations are a prognostic factor as well. So meningiomas look very different under the microscope than diffuse gliomas. The one thing that you should recognize right away is there's no brain in this tissue. It's just pure low-grade tumor cells. And again, that's because these tumors push aside brain rather than infiltrate. They have this spindled appearance, and they form whorls, these round spherical-like structures, and these whorls tend to calcify and turn into what's called somoma bodies. So the meningiomas are diagnosed by the presence of whorls and somoma bodies in a solid low-grade infiltrating tumor. This is what the majority of meningiomas looks like, but our history in pathology has separated tumors into what they look like under the microscope, and we still use some of these terms to describe meningiomas. Most of them shown up here are associated with a grade 1 diagnosis, but others like the choroid meningioma and clear cell meningioma, just based on their histology, are known to have a higher risk of recurrence and are associated with a grade 2, whereas others like the papillary meningioma and rhabdoid meningioma have a more rapid growth and are associated with a grade 3 behavior. So many of these histologic types do have genetic associations. And some of the histologies are associated with a higher grade. We don't do wide scale genetic analysis like next generation sequencing on the majority of meningiomas because in many cases it's not that prognostic. There are some specific genetic alterations that are though, and I'll touch on that. So a diagnosis for the future for meningioma would probably look something like this. And we're getting to this on a routine basis, not quite there, but it would include a diagnosis of meningioma. It would have a grade 2, and that could be based on the methylation class, the presence of a TERT promoter mutation, or NF2 mutation. So we are incorporating the genetics into the diagnosis of meningiomas more and more, in particular in those cases that seem to be at the borderline. So we may look for chromosome 1p loss, which is associated with an increased risk of recurrence, similar to that of a grade 2 lesion, or we may, if the tumor is a borderline between grade 2 and grade 3, look for TERT promoter mutations or CDKN2A/B deletion, which is associated with a more aggressive clinical behavior. And then the whole genome DNA methylation profiling, as we discussed, can pretty well segregate tumors into benign, intermediate, and malignant classes that predict clinical outcomes as well. So we've evolved with meningioma grading in the new WHO. Like I said, the vast majority of these tumors are WHO grade 1, probably don't need extensive molecular testing. Grade 2 includes those tumors that are shown to invade the brain microscopically or be more proliferative and have a lot of mitotic figures or have other atypical histologic features. The real malignant meningiomas that are grade 3 and would require more aggressive therapies either have a sarcomatous growth pattern, a very high degree of proliferation, or loss of CDKN2A/B or TERT promoter mutation. Both of those are now criteria for grade 3 in the new WHO classification. So I hope this has been helpful to you in understanding the flow of tissue and the work of pathologists in making a diagnosis. Pathologists in the lab do play central roles in establishing a brain tumor diagnosis, which then goes on to guide therapy. Molecular classification of brain tumors is here to stay, but is still a work in progress, and new platforms for classification are being developed. We do incorporate molecular results into our diagnosis based on the WHO classification and grading, and new platforms like DNA methylation profiling will probably change the field of tomorrow. So with that, I would like to provide these disclaimers and to say thank you. It really is an honor and a privilege to be able to speak to patients, their families, and loved ones. We don't get this opportunity, and I think there may be an opportunity to ask questions if we have enough time. I don't know if I was too long-winded or not, but I'll let the organizers determine that. Thank you.
Thank you so much, Dr. Bratt, for your wonderful presentation. We do have time for questions. As a reminder, if you have a question that you'd like to ask, please type and submit it using the Q&A button at the bottom of your screen, and we'll answer questions as time allows. All right, so we've already gotten quite a few questions here. One of the questions we have from one of our attendees is, you talked about some genetic markers for some brain tumor types. Are you aware of any genetic markers for schwannomas?
There are genetic underpinnings of schwannomas, like loss of neurofibromatosis genes, but nothing that's prognostic to my understanding. The vast majority of schwannomas are low grade. They tend not to go under malignant transformation over time. That doesn't mean they can't recur, but they most often don't undergo malignant degeneration. So the answer to your question is we don't have genetic markers for prognosis in schwannomas.
Thank you for providing that clarification. Another question we have is, when you are performing histological and molecular analysis, is there a minimum size of the tumor in order to undergo molecular testing? Does the tumor have to be of a particular size to have enough tissue to undergo that type of analysis?
Yes, it's a very good question. I would say that, and the answer is yes. You'd be surprised at the small amount of tissue that we can do molecular analysis on these days. It has to be probably 2 millimeters by a centimeter, a nice needle core biopsy to make sure that there's enough cells in there. But the technology is advancing further and further, and now with some technologies, with some sequencing capabilities, we can actually detect altered genes in the plasma, which are much lower in concentration than in the tumor. We don't do that so much for brain tumors, but we have the capability. But yes, we like to have sufficient tissue because some of it goes to immunohistochemistry, some of it goes to next generation sequencing, some of it goes to cytogenomic arrays for chromosomal analysis, and some of it goes to MGMT analysis for promoter methylation. So, we probably need 30 unstained slides for that in general from a small biopsy. So tissue can be limiting, but often is not.
On the topic of tissues, can you talk a little bit about getting a second opinion? How does that work?
Yes, we see that quite a bit.
Nice.
And
okay, how does that work for patients who are wanting to get a second opinion? Is the tissue sent over to the other institution where they're getting the second opinion, or how does that work? Can you talk a little bit about that?
Sure. So second opinions can either occur when the patient seeks care at another institution. And in general, the radiology and the pathology follow the patient. And we receive the slides and the blocks and do our analysis when the patient arrives to be seen by the neuro-oncologist. Or if it's a challenging case and the family is concerned about the diagnosis, or the neuropathologist wants assistance, they can send a tissue, and we receive the block and the slides, the report. We accession it to our institution, review it. We may need to do additional molecular studies. Typically, the molecular studies have been done already, maybe not as extensively as we would do. So yes, that happens on a daily basis. We get as many consult cases into our department as primary diagnosis through our neurosurgical service.
Thank you so much for that answer. Another question we have, you talked about some of the genetic markers for different types of brain tumors like glioblastoma. Are these genetic components hereditary? Is there any testing that patients' family members should be undergoing to see if they may be at risk?
Another wonderful question. We know from previous studies that only about 5% or less of primary brain tumors are familial. When we do our molecular analysis, there are suggestions that might be the case. Usually there's a family history, though, that is more telling. If it's a sporadic case that's germline, which means it's hereditary. We'll often see that when we do the next generation sequencing. In those cases that are hereditary or germline, we see twice the number of mutations in the tissue because every cell has 2 mutations, both the germline that the patient was born with and the second one. So there is information in the next generation sequencing that might suggest familial patterns of inheritance, but there's also usually clinical clues to that as well.
Thank you. Another question we have here is going back again to the tumor sample and the tissue. Are these tissue samples saved for future testing? What happens to the sample after you've already undergone molecular testing?
That's a great question. It's a very informed audience. And the answer is yes. By our standards in pathology, our certification standards, we have to keep the tissue for 10 years. Most institutions keep it for longer, exactly for this reason. So if I was a patient or family member that had a diffuse glioma diagnosis prior to 2010, for example, I would want to know its IDH status, its 1p19q status, if it potentially is an oligo, because those tests weren't done back then. We're 12 years out from 2010. I mean, IDH was really only, I think, discovered in 2006, 2007. So but the testing has been around since about 2010, 2012. So, and other genetic alterations might influence the diagnosis as well. So, yes, contemporary testing can be done on old tissues that are in archives within pathology departments. Sometimes it's challenging because there may be some genetic degradation over time, but it can usually be accomplished.
Thank you for sharing that insight. Another question we have here is asking about recurrences. If a brain tumor has reoccurred, does the molecular profiling change? Can it change?
It can evolve modestly in most cases. There may be a genetic alteration that's associated with progression. The fundamental tumor type stays the same, but some genes may be deleted that confer more malignant behavior. Occasionally, there may be a gene that is mutated that could be a target of therapy. And then perhaps more importantly, the tumor can become what's called hypermutated occasionally. And in other types of cancer throughout the body, that hypermutation, the tumor mutational burden being high, is a predictor of response to immunotherapies. That's not entirely clear to be the case with brain tumors, but I'm not the expert on that. Ava and Sean may be, but those are the type of alterations that can occur. And when the clinicians request repeat molecular analysis, we certainly do it. We don't necessarily do it routinely, though.
Thank you, Dr. Bratt. Another question we have here is about where molecular testing and genetic testing can be done. Is this something that's available at smaller community hospitals as well, or do patients and families need to go to larger institutions to have this type of analysis done?
Certainly the larger institutions do have this type of molecular analysis, most of them. But there are large reference labs also that will provide that. I think the biggest ones are Foundation Medicine, and I don't have stock in these, by the way, no disclosures, and Tempus and Caris, and obviously the Mayo Clinic. So there are large reference labs that these can be sent to, but if the patient is seen as in a large academic center, which most tend to be, most of those hospital labs do have the capabilities. And as these diagnoses really require this type of molecular analysis, more and more institutions will be acquiring those capabilities.
All right, so it looks like those are all the questions all the time that we have for questions for this session. Thank you so much, Dr. Bratt, for taking the time to answer the questions. Our presenter will stay online and try to type answers to any of the questions that we might not have had time for. I'm going to go ahead and turn it back to Emily to move forward to the rest of the program. Thank you again, Dr. Bratt.
Thank you.
Everyone. In this next session, you will learn about the role of surgery to improve patient outcomes and how surgeons maximize safety and efficacy. Please welcome back our co-chair, Dr. Sean Hervey-Jumper, who will be presenting on this session, along with his dear friend, who he will introduce.
Thanks so much, Emily. What a morning. This has been great so far. So in this next session, we're going to learn about the role of surgery to improve overall survival and where surgery is and is not beneficial. So I'm not going to do this alone. I really have the distinct pleasure of working alongside my friend and colleague, Dr. Keynote, who's a neurosurgeon and chair at the Mayo Clinic in Florida. So Professor Q, or Q as I call him, is a neurosurgeon, but he's really what we call a triple threat. And for those of you who don't know what that means, he not only is an outstanding neurosurgeon and doctor, but he's a celebrated author. He's an NIH-funded researcher in cancer biology. And I won't spend time this morning going through or recounting all of his accolades. They're online, you can read them anywhere. But please know that his life and his work from his beginnings as a farmhand to being one of the leaders, if not the leader in the field of neurosurgery, including neurosurgical oncology, please know that his work is an inspiration to us all. So we're going to go about this in a couple of ways. So Emily, if you could share the slides with me, I will present first for the first part and then hand over to Dr. Q, who will take it from there. Again, thank you ABTA organizers and the ABTA community. I specifically am a neurosurgeon and a cancer neuroscience researcher at University of California, San Francisco. And as we walk through this program, hopefully everybody is getting the feel here. This program was designed to mirror the patient journey. So Dr. Q and I have been charged with talking about neurosurgical advances, and there's so much more that you're going to anticipate and encounter throughout the next couple of days. In setting the stage, I'll review the how we think about surgery in the context of benign and malignant brain tumors within different areas of the brain. I will discuss when we operate and what are our considerations for operating. And Dr. Q will talk us through specifically how you get that done and what are the goals for each and every patient and how those goals may differ from one patient to another. I have no disclosures. Ooh, the slides are skipping. Okay. I have no relevant disclosures here. So as a surgeon, when we begin to think about patients with tumors, the patient comes into our office, we first ask ourselves a couple of questions. First and foremost, is this tumor within the brain or outside of the brain? And second, where in the brain might it be? Is this in a specific area or a focal area, or is it more diffuse or spread out? And so let's tackle the first question first. So if the brain tumor is within the brain, we think about approaching it from working within the brain. If it's outside of the brain, we take what we call extraaxial approaches, working outside the brain. And the screen left, you'll see two different images of MRIs for patients with meningiomas. This is a type of brain tumor that originates from the tissues that line the brain, or the meninges, also called the dura, just like Dr. Bratt spoke about. And as you can see here in this example, the tumor itself is spread along the falx. It's spread along with the part of the brain that separates the left and right hemispheres. So this is really centered along outside of the brain within the skull. And so that's how we approach it, or we think about approaching it from along that dural surface. And you can see on the screen right, you think a bit differently, where this is a tumor where it's in the left frontal lobe, and you can appreciate how it's within the brain tissue itself. It's within the brain parenchyma itself. And so as we think about these different approaches, let's dig a little deeper into how we can address them when we're thinking about and talking about each individual patient. So let's talk about our surgical framework for tumors within the brain first. Oops, it's a bit of a delay. Okay. So as we walk through intrinsic brain tumors, let's start by thinking about and talking about gliomas. So these are the types of tumor that originate within the brain. Like neurons, glia are important cells in the nervous system. Scientists used to think that glial cells were specifically brain glue, meaning they held the brain together. And most of the focus was on neurons, but and that's really where the term glia comes from. It's Latin for the word glue. However, we now know that glial cells are not just bystander cells sitting in the brain. They're really important cells for control of neurons and brain function. And if these cells become abnormal, based on the WHO classification system that Dr. Pratt just walked us through, we can get diffuse gliomas, grade 1 and 2, for low-grade gliomas, grade 3 and 4, for high-grade gliomas. And going further, we can then distinguish these cells based on how they look under the microscope, as oligodendrogliomas or astrocytomas, depending upon their appearance under the microscope, as well as their genetic signature, which we've already heard about. One particular marker is particularly important, that's the isocitrate dehydrogenase gene. As surgeons, we often don't know that information up front. We get clues from the imaging about in the patient characteristics about what those molecular markers may be, but we know that surgical resection is a benefit to patients regardless of which IDH status you have. And then we finish off with grade 4s. Those are the malignant gliomas, the glioblastomas, that can be IDH wild type or IDH mutant as well. Now the other type, looks like there's a delay here. Now the other type of tumor that's within the brain itself is really called a brain metastasis. In fact, of all the patients with brain tumors that show up, tumors that spread to the brain from other places, called brain metastasis, are really the most common. Emily, I might need you guys, oh, there we go. There's just a bit of a delay. If the slides don't advance, I'll just ask you guys to advance them for me, okay, if you don't mind. So you can see that brain metastases can be the most common types of tumor in the brain. And there are particular types of brain metastases that are more adept or more efficient at lodging into the brain tissue. And those are brain metastases that originate from lung cancer, breast cancer, and melanoma particularly. So taking this back to the surgery perspective, as surgeons, we think about who would benefit from surgery and who wouldn't by focusing our thoughts initially on whether the brain metastasis or this tumor intrinsic to the brain is confined to one particular area, or in the example you can see here, spread out to multiple areas in the brain. So you can see on the screen right that this is a patient with brain metastasis that contains at least 6 tumors, one of which has some blood and bleeding in it, that's the little black spot. Therefore, applying an approach in which we remove a tumor in a single focus or multiple focus wouldn't really necessarily be the most effective, because you're really talking about a biological process that's fairly spread out in the brain. So we need to think about systems or treatments that are more systemic or spread out as well. And that's where chemotherapy and radiation come in, and reserve surgery here in this setting more for tissue diagnosis or tissue confirmation. Let's see if this will advance. Here we go. So if we then shift our focus to extraaxial tumors, meaning tumors that are from outside of the brain, meaning they're in the skull itself, but outside of the brain tissue, these are most commonly called meningiomas. And meningiomas really are named or based on where they attach to, where their dural origin is. So if they're centered along the dura, or the coverings of the brain over the frontal lobe, we call it a convexity tumor. If you can see that it's centered along just the area around the nose, like what we call the olfactory area, it's called an olfactory groove meningioma. And we name them that way. So there are grades. These are graded, the tumors, based on the pathology that is sent to the neuropathologist. And sometimes the growth really is determined based on the tissue pathology that we find. But as surgeons, we operate or think about surgery in a couple of settings. One, if the tumor is growing, or two, if the patient has symptoms that relate to the location of the tumor. So again, the more tumors removed, the better for the patient if surgery makes sense based on the tumor's location, based on whether it's growing or not, and based on the patient has symptoms that we can attribute to the tumor, meaning focus on whether the tumor is impacting your life or an incidental finding. There's some studies suggest that meningiomas are really common and can be found in many, many, many patients. So we only operate in certain distinct settings when we think it's causing a problem for the patient. So let's try to crystallize these concepts a little bit better by walking through decision-making and when we consider surgery versus no surgery. So here's a case example of a 50-year-old woman with no significant past medical history. She's right-handed and has a 2-week history of headaches. So first, you can see we ask ourselves, is this a lesion? Do we think this is a tumor? Do we think it's something else? Vasculitis, inflammation, infection? Well, there's certain imaging characteristics as a surgeon that make you think that this is a tumor. One being the fact that it's expansile. You can appreciate that on the right side of the brain, that the area of brain affected is thicker or wider or swollen in comparison to the left side. We also ask our question of whether it's multiple or single, and it looks to be in one specific area. We call that area the right frontal lobe and the insular cortex. And then we ask ourselves, what might the risk be to the patient if we consider surgery? And that's based predominantly on tumor location. So you can, as we walk through the two approaches, for this case, which is a patient who we believe has a glioma, and particularly a low-grade glioma, we have a couple of options. So one option is to watch and wait and see if the tumor grows over time. We call that a watchful waiting approach by getting intervening imaging. That would mean within several months, you get another MRI to see whether the tumor has grown. And there's a lot versus the other intervention, which is early surgical intervention or surgical resection. And there's really a lot of data out there now to drive this decision-making in this day and age. So published studies have shown, for the most part, that if the tumor is within an area that can be safely approached, then early intervention, meaning before watching and waiting for growth, leads to greater amount of tumor removed. It leads to longer survival and better outcomes for the patient. And when we watch and wait, the outcomes in general are not as good, as survival is often shorter, and the risks of removing the tumor can be large, and the rate at which a brain tumor, a low-grade tumor, would turn into a high-grade tumor, something called malignant transformation, is a bit higher when you use the watchful waiting approach. And this is, again, this question has been looked at in a number of different contexts in different areas of the world, and one of the most compelling examples comes from a study in Norway. And Norway is an interesting place because in this setting, patients based on geography are treated at one particular hospital versus another. And so it turned out that we've learned that in one hospital, historically, patients were treated with the watchful waiting approach. And in another hospital, they had the early resection approach, early intervention approach. In the hospital, and the patients in the hospital that had the early intervention had a markedly improved overall survival, and of course, follow-up studies suggest better quality of life with early intervention, better seizure control, etc. And so in that setting, in the context of Norway, the treatment paradigm changed, and it helped us in many centers around the world understand what the relative benefit of early intervention might be for patients based on this example. So you can see here, again, that biopsy and watchful waiting can be considered, and there are times when that is important. And we can go over that in more detail. But often, for many patients, it's inferior to early resection when it is feasible and safe to do so, and in line with the patient's goals of care. Let's look at another example here. So another scenario here, this is an example of a 75-year-old right-handed man with high blood pressure, diabetes, and trouble with speaking, also called aphasia. You can see here a roughly 3 centimeter left frontal contrast enhancing mass. So what do we do surgically in this situation and why? That's the key. Well, the goals of surgery. Here we go. Thanks. So if we list our possibilities and think about the goals of surgery in this setting, in which we will find out is a glioblastoma or a high-grade glioma, we obviously don't know this at the time of surgery, so we go based on our differential diagnosis or list of possibilities, what we think it might be. And as surgeons, we walk the patient through the surgical plan based on the value, put another way, the surgical advantage or survival advantage that may be associated with resection. So having as little of the mass in your brain as possible offers a benefit for patients. And we know that surgery isn't a cure for patients with malignant glioma. However, removal of as much of the contrast enhancing disease as possible is essential and offers a therapeutic benefit. And it's also true that in a subset of patients, or at least a subset of patients, we believe, removal of the tissue adjacent to the contrast enhancing tumor. This is something called a supramarginal resection or super complete resection, offers an even greater benefit. And that's a very nuanced discussion that we have with the patient and the family based on where the tumor is located and the patient's goals of care. So with our surgical goal, we proceed to the operating room. We use a number of different techniques to maximize safety that Dr. Q will go over with you. We apply oral chemotherapy and oftentimes it's temozolomide followed with radiation. And the goal, of course, again, is driven by patient wishes, but the point is to remove as much as can be done safely. And the key is how we do that safely. And this makes a good transition point to turn over to my colleague, Professor Keynote, who will walk us through some additional scenarios, as well as how we maximize safety.
Thank you,
so much.
Professor.
Thank you so much, Sean. We will hand it over to Dr. Q now.
Great. Great. And maybe we can just keep Sean with the control of the slide, so it makes it easier, because I think that he figured out already, you know, and if I can ask him for the next slide. So I am honored to be here with all of you, and we'll talk, as Sean just mentioned, and Dr. Hervey-Jumper just mentioned, about some of the nuances. And my goal is to go back and forth here and maybe shed a little bit of light into this complexity. The topics can be complex. We just, sometimes in the middle of that complexity, there's a simplicity. And what I can say is, anytime we propose surgery, number one priority is that the patient is the priority, making sure that we can get in, do whatever we need to do, whether it's a biopsy, a resection, a maximal resection, whatever we do, our goal is to always keep the patient as intact as one can. And I think that I go back to some of those cases that Dr. Hervey-Jumper just showed right now. We're going to go over some of those. So if I can go to the next slide, I don't have anything to disclose, you know, and we go to this case. And let's say you saw a little bit of this picture. The question that Dr. Hervey-Jumper asked is, do we need to do surgery all the time? When is it? You know, what is the logic that we use in deciding whether or not a patient needs to have surgery? And if the patient needs to have surgery, what kind of surgery needs to be done? And can we do it, yes or no? Can the patient get into the operating room and come out safe? Those are the questions. So this particular patient, 70-year-old patient, a history of non-small cell lung cancer. And that is relevant to this. And we'll get into the discussion right now. And is known to have pleural metastases that were diagnosed a number of years back. That patient has been already treated with some medications, including an ALK inhibitor, also relevant. And right now, the patient is on treatment with a controlled systemic disease, very well controlled with brigatinib, and he's got a 1-month history of headaches suddenly present. And of course, the oncologists are amazing in taking care of these patients. they immediately say, well, you know what, we got to make sure that we take care of you. They immediately say headaches, not good. Let's get an imaging study. They probably start with a CT scan. They move to this MRI, and they find multiple brain lesions just right there. What you see is 6 of them that we are, and potentially a 7th somewhere hiding, in one of the sides of the brain. That is very, very, very subtle finding. So the question is, should we do surgery for this lesion? Dr. Hervey-Jumper, what are your thoughts about this? Even before we go to this right here, because we got the answer right here. But it's got multiple lesions. Because you get patients like your colleagues, they call us about this. Okay, so should we do surgery?
Yeah, so great question. In my hands, again, after discussion with the patient, I would not be eager to offer surgery. Surgery is a focal ablative tool, and what I'm seeing is a more diffuse process. So the goal would be to get this patient a treatment that can be applied to multiple areas of the brain, something more like radiation or the systemic chemotherapy option that might penetrate into the brain to touch each of the tumors at the same time.
Well, I'm gonna ask you a different question now. Let's say that this patient has headaches, the disease is well controlled in the body, and they only have 3 of these little lesions, and one of them is very large with a lot of mass effect and a lot of edema and swelling.
Yeah, Q, that's such a common scenario that we get each and every week in working with patients. So first question, to bounce back at you, is what does the oncologist feel about the patient's overall disease control in their body?
I think it's amazing. He's doing great. Body is responding so well to the therapy. And they said, we were surprised to find these 3 lesions. 2 of them are tiny. One of them is the size of a golf ball.
Yeah, so that's a different story here. So with a dominant symptomatic tumor, with a patient who is in good performance status, meaning rigorous, robust, willing to go through the recovery from surgery, combined with an oncologist that thinks they have more systemic chemotherapy options or radiation options for the patient, removal of the dominant tumor can get them to the next stage in treatment and help the symptoms, so improve quality of life as well.
And this is important, of course, for our audience, because sometimes they get confused. They said, oh my God, but my friend had multiple lesions. He or she did have surgery. That multiple lesions is not the same on every patient. We look at the systemic disease, and we go to the next slide because that's where the answer in this particular patient, multiple brain lesions. We know that surgery, as Dr. Hervey-Jumper just said, is not the right thing. We're going to hurt that patient with surgery. We have better therapies. If the patient has got 1 lesion at new diagnosis, yes, we will offer surgery. But in this case, we're talking about 6, potentially 7. Those are the ones in one cut. There may be even more all around the brain, multiple lesions. We still have other alternatives like gamma knife, cyber knife, stereotactic radiosurgery. You'll hear this terminology, or WBRT, whole brain radiation therapy, and we have very good data, but the role of surgery in treating multiple brain metastases may be beneficial in patients with accessible, as we said, symptomatic lesions and controlled systemic disease. And those are 2 very different scenarios. One of those, in this case, unfortunately, the patient doesn't have a very good control of the disease, multiple brain lesions. I think we have still tools, and radiation presents a very, very viable option. Anything else to add right here, Dr. Hervey-Jumper, in this case?
No, that's a great description. I love the case.
Beautiful. Well, let's go to the next case. So just for our audience, so they understand a little bit more, we're going to go the same deal. So this is a patient, 35, young patient, non-pertinent past medical history. Sometimes patients are going to get very, very scared, and they will call Dr. Hervey-Jumper and myself immediately. They want to talk to a brain surgeon. I said, let's relax. Let's try to figure out what this is. First of all, they get an MRI, as it is right here, and then it indicated for head trauma. They probably sustained a fall in this case. They got this. It was an incidental finding, and they see this lesion. And it's left-sided. And we know, Dr. Hervey-Jumper and I, we do a lot of surgery right around this area. You're looking at an MRI sideways like this, all righty? So small little cuts. what we call a sagittal. So side was like this, you're looking like this in the left side. And we find this lesion. This lesion, how would you describe it, Dr. Hervey-Jumper, this particular lesion?
Oh, I would describe this as a, again, left temporal contrast enhancing lesion that's under 1 centimeter in size. And importantly, I don't see a lot of swelling or shift or any concern that the brain itself is upset by the lesion. The brain itself seems very happy and not compressed around the mass itself.
Absolutely. I would describe that exactly that. I think that this lesion has probably been there for a while, and sometimes we even get some other images. You'll hear us talk about, oh, let's get this little CT scan because they may tell us a little bit more about calcium. And we did, and we know to the best of our knowledge that this is very likely a meningioma. To the best of our abilities, we go to the next slide right now. And in this particular case, in this particular situation, the best thing that we can offer, remember, not because we're brain surgeons means that we need to, or we want to, or we have to do surgery in every patient. This particular patient will be better served with observation. She's 35, his lesion is small. I normally tell them, we'll follow you. It varies. Some surgeons may want to do MRIs a little bit closer, some other surgeons a little bit more. But what I do tell them is we are going to have a long-term relationship. We are going to follow you. We're going to follow each other, not because we're not proposing surgery. That doesn't mean that we're not doing anything. We're doing a lot. We're going to go ahead and monitor the situation. If I will present this case to Dr. Hervey-Jumper, and I would say, Dr. Hervey-Jumper, that image below, we see in one of the images that there is a fair amount. The lesion is only this big, but the size of a marble, but the swelling is the size of a golf ball. What would be your thinking now, Dr. Hervey-Jumper?
Yeah, I'm concerned about two different processes. One, that this is a more aggressive meningioma, meaning many meningiomas are classic. Roughly 88% are the kind of slow growing or not growing benign meningiomas, but 12% are meningiomas that are growing faster. The other concern I have is that it could represent spread of a cancer somewhere else in the body to the brain, either what we call a dural-based metastasis or some other type of tumor that's growing faster in the brain. Again, the brain's reactive. When the brain is reactive and swollen, that's not a good sign. That's when we start to worry or think there could be a more aggressive process.
Beautiful. In those cases, of course, what Dr. Hervey-Jumper and myself would recommend is let's get it up. We need to get a diagnosis. If it's accessible, if it's right on the surface, as it appears to be in this case, we are going to recommend surgery and then find out what is the histology, what is, were we able to get it all out? Did we clean the dura, that little part of the layer that's right under the bone that is covering the brain, is very thick, leathery. Did we take that out? So we maximize the chances that there's no need. But if the pathology shows us that it's a little bit more, it's that the grade 2 or the grade 3 that Dr. Hervey-Jumper was talking about, if it is a meningioma, then we may recommend to follow up with around the radio. This is where we have our boards. In both Dr. Hervey-Jumper and myself, we work in places where we have a brain tumor board and we have multidisciplinary discussions about what we do in these cases. So these are the scenarios. Anything else right here, Dr. Hervey-Jumper, that we didn't include?
It's beautiful. Nope.
Beautiful. Let's go to the next case. Let's see what we have. The next one, all right? So the next one is the histology, the degree of resection, the tumors, we talked about it. Let's go over some of these techniques for these gliomas or meningiomas. Some of the times we're going to have to use techniques that will give us a sense, as we said, if it is a low grade, a grade 1, meningioma, we're going to observe. If it is a grade 1 meningioma, we left a little bit of tumor, maybe we'll recommend gamma knife, cyber knife, stereotactic radiosurgery. If it is a grade 2 meningioma, we may recommend observation with some fractionated radiotherapy. And then we go all the way down to the grade 3. It's very rare, as Dr. Hervey-Jumper mentioned, but we have to be thinking about it. And now we're beginning to understand more that those cases will benefit from the resection, that we'll try to do maximal safe resection, or at safety of the patient is always a priority, followed by radiotherapy and some chemotherapy, and we are now beginning to do peptide receptor radionuclide therapy, and hopefully in the next 5 to 10 years, we'll continue to do more of those things. So these are the ways that we think about the meningiomas. Let's see if we move to the gliomas, because I want to get and ask Dr. Hervey-Jumper some questions. So some of the techniques. The first technique, which is the next slide, is a technique that we both use very much, awake craniotomy. Dr. Hervey-Jumper, you published extensively in this topic. Maybe it's a good time, to tell people what does it mean to be awake.
Yeah, it's actually not the perfect name for this technique. It's really, Dr. Q, as you know, probably better described as asleep, awake, asleep. You're not really awake the whole time. The key in the operation is that when the surgeon believes and working with the patient that they need to reserve the right to stop, meaning that the tumor is within an area that's functional, at some point may need to test that function to make sure it's preserved. That's really the key to an awake procedure.
Absolutely, I love that. And you can see a lot of the little numbers. What we do is we stimulate parts of the brain, and our goal is to find what parts of the brain are so important that we should not touch them, and find different corridors to attack a brain tumor and try to take it out. That's really the principle. And for that, we use a state-of-the-art technology, stimulation. We have an amazing team inside with us, all the way from neuropsychology, neurologists, neuroanesthesiologists. It's amazing. And I can tell you, most patients don't realize how strong they are. And no matter what we create in life, at the end of the day, there's nothing more beautiful and more precise than your own brain telling us what can or what we should not do when we're doing this. Let's go to the next slide. This was a two picture. If we go to the next slide, it'll tell us some of the techniques. This is also from one of Dr. Hervey-Jumper's papers. We use everything from asking them. We use electrodes to monitor the function of the arms and the legs and the face, and we stimulate the brain. And we just, once again, sometimes they can be under general anesthesia when we do that. Alrighty, especially in this case, we can also achieve things under general anesthesia that are very precise. We just have to know how to titrate this anesthesia. We follow with electrodes in the muscles. We can stimulate, especially when we're taking tumors in parts of the brain that are more important for motor function. Of course, speech function, being asleep is not as, there are some documented things out there in life, but the reality is, for us, speech will have to be completely, as we said, part of the surgery awake, not all of it, but part of it. But if it's motor, we can do it with the patient under sleep condition, but very light sedation and stimulation of the brain. All righty. And our goal is to resect as much as safely possible. Notice right there at the bottom, we're talking about extent of resection as high as 90% of recent studies. We can achieve those things. Having an experienced group of surgeons will allow you to be able to do that. Anything else to add right here, Dr. Hervey-Jumper?
Oh, beautiful. Good.
Let's go to the next slide, because we have about a minute or so, and I wanted to open it for questions. The LIT, laser institutional thermal therapy. These are new techniques that we're beginning to use, where we put a small little electrode and we can hit these tumors, and it's actually working in some really, really wonderful cases. I think it's selective ablation. We can go with it. Not all patients are candidate for this. Is very, very important for some gliomas, some metastases, some radiation necrosis, and sometimes, especially patients that have those tumors deep down in the brain, the thalamus, the brain stem, we can use this technology. If we go to the next slide, it's something that has been pioneered in places like UCSF, the label dye-based intraoperative imaging, all the way from 5-ALA. I know that there was a clinical trial going at UCSF, sodium fluorescent. These are techniques that are being utilized. I think that they're wonderful techniques that many of our colleagues around the world, but if you hear about them, that's what it is. It's basically labeling in the operating room. You're looking at it under the microscope, and I think the next picture will tell us the actual microscopic utilization. We can use the microscope with this sort of fluorescent label dye techniques. Some of them are given oral, some of them are given intravenous, as in this case. I think they're wonderful techniques that we're using, and we use the microscope. Microscope have special filters that allow us to see this right here. So I think the technology has evolved tremendously over the last 10 years. But at the end of the day, I go back to the same principle. The knowledge of the anatomy of the surgeon, The knowledge of the function of all the team members will always allow us to do the best job. These techniques are adjunct, nothing compares to that judgment that a team of physicians will use in the operating room to be able to take these tumors out. Anything else to add right here, Dr. Hervey-Jumper, that we should be considering?
No, I echo your comments perfectly, completely. And actually, we do have another 5 minutes. So if we can finish up the last 2 slides, we're good. Take your time.
You're very, very good. So this one right here is actually very, very important right here. The simulated Rathman histology, it turns out that we have a challenge in the operating room when we are doing surgery. Dr. Hervey-Jumper and myself are doing surgery. We get to the point that we're at, especially sometimes at the edges of the tumor, and we can tell what's tumor and what may actually be brain that is already beginning to get more normal, but we don't know if it's functional, we don't know if it's cancer, we don't know if it's tumor. So the technology nowadays is allowing us to be more precise. This is one of those technologies, simulated Rathman histology, that allows us to be there, have a pathologist with us in the operating room. Right now, we have to send the tissue outside, and it takes, even in the best centers, 10, 15, 20 minutes. Imagine if you want to do it over and over and over, you want to go millimeter by millimeter, you can make a surgery last for hours and potentially even a day. So this technology is allowing us to be able to get a sense of what is going on in the operating room right there. We have rapid resolution of images and of unprocessed tissue. We have histological data readily available during surgery, and we have diagnostic precision within 2.5 minutes. So that allows us to move more expeditiously, but we do still do this in combination with the pathologist, but now allows us to have the pathologist right there in the operating room being part of the team. So the team continues to expand and be more precise in what we're doing. So that way we do maximal safe resection and able to get the diagnosis. And if you combine that with the patient being awake, it's even more precision, all right, that we can do. So that's the histology. We have one more, I think, but there was one more slide right here, Sean, if I remember correctly.
Yes, yes.
This one, I think this is where the technology, I remember I finished UCSF with Dr. Hervey-Jumper before he got there in 2005, and we were beginning already to do these amazing tumors. Not only the pituitaries, nowadays we're beginning to do the meningiomas also through the nose, some of these large meningiomas. That particular tumor that you see right there is a pituitary tumor. We can go through the nose. Back then we were going through the lips, we using tube. Nowadays we just small little cameras, smaller than the tip of my finger, that we can introduce. And it is amazing the amount of work that we can do to take out these tumors, especially the meningioma. So the technology continues to evolve, and we're very grateful that our patients, trust us with their lives, of course, with their loved ones. And we're very grateful that technology is allowing us to move some of the state-of-the-art techniques into the operating room. As you can see in this particular case, going through the nose to take a meningioma into the brain. So I think that the future is bright, and we're excited, and we're looking forward to answer any questions that you may have.
While the questions come up, Emily, stop me if we're over time. But Dr. Kinases, one important question that came up in the question and answer session really struck me. The idea of awake craniotomy is scary. And the specific question is, do you experience or notice with your patients any sort of post-traumatic stress or any sort of negative experience that patients feel when they partner with you in the operating room to maximize how much tumor can be removed? Is this traumatic to the patient?
Beautiful question. I actually find it the other way around. I found that the patients, when they felt part of their own journey, they felt part of surgery. They didn't feel like they were more than just another case. But I think it has to do, Dr. Hervey-Jumper, that it all depends on the team and how it is approached. Of course, at our centers, our patients get to meet everybody, all the way from the neuropsychologist to the neuroanesthesiologist, the neurologist, before they go. So they feel part of a family. And of course, nowadays, what we're doing, remember, there are pain sensors on the skin and some of the dura that covers the brain. And sometimes you can have that psychological pressure. But I think patients receive, as you said, Sometimes they're asleep at the beginning. You wake them up slowly, comfortably. They know already, they expect it, they train before the surgery. They are not foreign to the concept, and they have amazing experiences. I yet have to hear a patient that has had a bad experience in the operating room in general. They said it was an amazing experience. I mean, some of them even look forward when they have more than one craniotomy awake. And I have already patients with more than one, 2 or 3 times actually.
Yeah, no, I echo that comment. You know, I think one of the parts of all of the therapies that we offer for patients that gets overlooked is the fact that you didn't deserve this, right? It came out of nowhere, you had no control, and you didn't deserve this. And there's only a few places in the treatment paradigm where you as the patient can intervene, and kind of your participation directly correlates with the outcome received at the end. And so I agree with you. I would say by and large, the patients are enthusiastic to gain back some of that control and partner with their team to do a really good job and show that perseverance that gets them better on the other side. I couldn't agree with you more. You mind if I ask another couple of questions that have come through? I think this is an important one. If a patient has a subtotal resection, a lot of the tumor removed, but not all of it, is it possible to come back and have an additional surgery at another time? Or if you talk to the doctor and they say, before we start radiation, maybe we should consider more surgery, is that safe to do? And in which setting do you consider that?
Beautiful. I just had it happen to me maybe about 3 or 4 weeks ago. One of my colleagues went into the operating room at a different institution. They did what they felt it was safe at the time. It was a large tumor with a lot of mass effect, you know, in the dominant hemisphere, left side, near speech areas. So what they did is they did a partial resection. They got the patient out of trouble. The speech wasn't good. The speech improved. And then they said, you know, you should probably go and see our team at the Mayo Clinic. The patient came, we saw and realized, yeah, we should get a functional MRI. Likely now your speech is better, so we can get a functional MRI, find out what the landmines are. And then we plan a second stage, literally 3 weeks after the first one. But the patient understood the first surgery was good to get the patient out of trouble, get the functional of the speech improved, allows us to get a functional MRI and allow us to plan a second stage. The first time they had taken about 30% of the tumor out, enough to get the patient out of trouble. The second time we were able to get about 97 to 98%, which is, of course, based on our studies, a wonderful thing to do. So there are specific times when that will be the case.
Yeah, I think that's a great comment. You know, there's a question that came through, and I will honestly say this is a question that keeps me up at night a little bit. And you know, as you know, Q, we think about building systems and offering treatments that are accessible and available to all patients. But the truth of the matter is that you and I are at University of California, San Francisco, Mayo Clinic. We're at resource-heavy centers where not every patient is there, right? In a resource-heavy region. They could be either remote or urban, but not resource-heavy. How do you talk through with a patient who is seeking care in their local community? If it is not resource rich, how can they have access to treatments or know about them or access these treatments or kind of interface with folks that may be able to help them? And if you don't have the answer, that's okay too. I know that's a difficult question, but what are your thoughts there?
My thoughts are the following. Medicine is evolving, no question about it, that you and I have our own families. That not everybody's going to have access to UCSF, to the Mayo Clinic, to some of these centers because geographical location. But I think that what we're beginning to see is that patients are becoming more savvy. Not everything, not all the surgeries are going to need a major center. Obviously, the tumor is near the speech area that we know is tough. If the tumor is near the motor area that we know is tricky. We understand also that if the patients do have deficits, they don't do as well. So maybe those patients should come, at least for the surgical portion, for the beginning of the portion, to these places. Luckily, for instance, for some of the tumors that are protocolized already, whether it's the chemotherapy or the radiation therapy, so then we can coordinate our efforts. We do that. Your center, my centers, is our responsibility as big centers to coordinate with the local physicians, the local oncologists, the local radiation oncologists to be able to do so. And if they feel uncomfortable, then we got to figure out a way that those patients come in. But suddenly you go from 100 patients that are trying to get to the major tertiary care center to maybe a fraction of that, and maybe even a smaller fraction that needs to have the entire length of the therapy at these major tertiary care centers. I think the key right here is going to be to open the channels of communication to make sure that the physicians in our less heavy, resource centers know that they have access, to our tumor conferences, they have access to our tumor boards, and the patients have the right to ask for a second or third or as many opinions as they want to.
Excellent. You and I could chat all day about this. Aubrey and Emily, how are we looking on time?
Thank you so much, Dr. Hervey-Jumper and Dr. Q. This was such a wonderful discussion. We will take a couple of more questions. We have a few coming in here. We have one question here from one of our attendees who's asking about impairment, functional impairment after resection. Is this something that you find improves over time? Does it resolve? Is it something that becomes permanent? Can you talk a little bit about that?
Beautiful question. Short answer from my side is that not all these deficits have to be permanent. And this is where we go. That's why we keep our patients awake. We know we have created a certain knowledge as to how much we can push the surgery. And knowing, that deficit is going to get better. Maybe that deficit won't be 100%, but it's rare that we're going to say that deficit is going to be there forever. If it's a surgery, what we cannot anticipate sometimes is the disease progression and the effects that the other therapies may have on top of that deficit. This is why obviously having some level of experience and having a good access to rehab centers that can work with the patient is going to be crucial, because I know that we like to think that what we do is important, but the reality is what everybody else is doing is equally important for the benefit of that patient. So, but what do you think? Sean, what are your thoughts?
Yeah, Aubrey, I really love this question. I wish we could see the people who are asking these questions, because it's an important one, and I'll answer a couple of points here. So first and foremost, the short answer is yes, there is a certain amount of organic recovery that most patients will receive for language, motor, cognition, etc. People get better after surgery, even after a resection in a functional area. That is because in this modern, and that's driven our surgical goals and consultation with the patient, where oftentimes we push the resection to the point of inducing an impairment because we understand a bit better what plasticity looks like and the timing of plasticity with relationship to the disease status or the type of disease that we think we're working with for patients. I will, however, say in counseling with patients, I've stopped or started to pause in talking about impairment rates. And I say that because, particularly for young patients, if you talk about a 1% complication rate, a 2% complication rate, et cetera, there are many patients that after a glioma surgery or after surgery for anything in their brain, they just feel different, right? And they sometimes can't put their finger on it. I feel different. Multitasking at work is different, or my interactions with my family are different, et cetera. And I stopped using, or I take a pause when I give these numbers, even when they're single digits, because I think many patients come out on the other end feeling other than or different if they are that 1-2 single digit percentage person. And I'll say that these number of folks who do feel different or may have subtle neurocognitive behavioral changes that impacts their life long term, the point is to get them into cognitive therapy and rehab so that they can lead a full life, even in the setting of those slight existing differences. So I try to use the term, you may feel different. I may not be able to quantify what that difference is, but you may feel different after this treatment. That's really common and I think an important point.
Thank you so much. Unfortunately, that is all the time that we have for questions for this session. Thank you again for your insight. I will turn it back to Emily and we'll move on with the rest of the program.
Thank you so much, Dr. Hervey-Jumper, and thank you so much, Dr. Q. That was such a great interactive session, and we really appreciate the dialogue between the two of you. For any unanswered questions we were not able to get to live, we are asking Dr. Hervey-Jumper and Dr. Q to reply to the Q&A box by typing in some responses. If neither of you are able to get to them, ABTA is documenting such questions and we will try to return them back to you. We will now have our lunch session. During the lunch session, you will hear from our sponsors who will share what resources they offer to patients and our researchers with important scientific updates. We encourage you to listen in during the lunch session to gain useful information. For those of you who need a break, please take this time and opportunity to do so, stretch your legs, use the restroom, grab some lunch, and we will meet each of you back here for our afternoon session. Our first session of the afternoon will be on personalized medicine and individualized treatment, and we will begin at 1:15 p.m. Central Time. Thank you so much, and please enjoy your break.
My name is Ini E, postdoc at Jackson Laboratory for Genomic Medicine. This is a great honor to share my research on tracing extrachromosomal DNA inheritance pattern in glioblastoma using CRISPR. This work was supported by the Basic Research Fellowship in honor of Bruce and Brian Jackson. Why is cancer so hard to treat? If tumor must consist of identical cells, then the cure strategy for this tumor wouldn't be complicated. However, unfortunately, brain cancer is the most complex tumor type, and there are substantial differences among the cells in abundance and types of genetic material. We call these differences tumor heterogeneity, and it makes treatment strategy more complicated, cause treatment failure, development of resistance, and ultimately cancer recurrence. So understanding tumor heterogeneity mechanism is important for managing complex tumor more effectively. In a normal cells, we have chromosome, the well-organized package of genes. And the centromere on the chromosome is essential to inherit the mother cell's genetic material equally to daughter cells. However, scientists found circular DNA outside the chromosome in half of the human cancers, and we call this extrachromosomal DNA, ecDNA. It contains major cancer-causing genes. Brain cancer has the highest ecDNA frequency. Since the ecDNA doesn't have centromere, it has been assumed to be unevenly inherited. However, there was no such tool to explore its behavior in living cancer cells. My project was focused on developing an ecDNA tracking tool. This tool was designed to tag multiple fluorescent molecules on ecDNA specifically. Then we can see ecDNA molecules labeled with fluorescent color in living cells under the microscope. Using this tool, I observed cancer cell division. I marked the ecDNA molecules with red arrows. A mother cell with multiple ecDNAs is divided into two daughter cells. But at the end, each daughter cell has a different number of ecDNA molecules. Here are the schematic cancer cell division. Regular chromosomes are equally inherited, but ecDNA are unequally inherited. If we look at the cells at fourth generation, of course, all cells have same number of chromosomes, but the amount of ecDNA in each cell extremely varies. So my results indicate that ecDNA can repeatedly generate tumor heterogeneity within only a few cell divisions. Unexpectedly, during live cell ecDNA tracking, I observed multiple ecDNA gathering together in more than 50% of cancer cells harboring ecDNA. But what is the benefit of ecDNA clustering? To be functional, all the cancer-causing genes should be transformed into a functional RNA and protein. And it requires some tool called transcriptional machinery. What I saw is that the major transcriptional machinery is found in the ecDNA cluster. Thus, the multiple ecDNAs get chance to make more functional RNA simultaneously by sharing the transcriptional machinery in its cluster. The cell with large green spots reflecting ecDNA cluster showed more functional genes labeled with red color than the left cell without ecDNA cluster. In summary, ecDNA are unevenly segregated during cell division. ecDNA generate clusters to make more functional RNA. ecDNA is a key factor of tumor heterogeneity. Based on this ecDNA tracking tool, we will be able to explore more unknown ecDNA biology, and I hope we can re-engineer it to be useful for diagnostic and therapeutic application. Thank you.
This is my mother's sister. Her name is Gila. She was really the life of the room.
Oh my gosh, this makes me sad. But yeah, so this is my father. They had a great sense of humor. He was always joking. They were just corny as can be. But it was dad jokes, you know.
My brother in the middle, his daughter on one side, and his wife, Clarine, on the other side. Clarine, she had that sparkle that made everybody love her.
In this picture, I'm with my Uncle Dale. I've always asked myself, well, what if someone I love gets this disease? But it seems impossible, right? And it ended up being a GBM tumor. And he's in the midst of that battle right now.
Glioblastoma is one of those incredible challenges for our specialty, but in many fronts, enormous progress has been made.
The Ivy Center right now, today, is the largest single program focusing on brain cancer research in the world. We're really operating at the cutting edge of science. Our brains are all different, so it really does have to be individualized. That's what we focus on.
With the phase 02 approach, we are trying to capitalize on weaknesses that the tumor might have, and we learn about what those weaknesses might be through molecular profiling. It's a very targeted, very specific, very scientific method of attacking the tumor and documenting a response.
Instead of going through a trial and then finding that you had absolutely no benefit, it really goes through the trial with various steps of checking to see whether there's potentially a benefit or no benefit. So it's a stop and go sequence.
By providing them with different options that are more suited to them, and we can give them something beyond what's been typical in the field for the past several decades and give them a new kind of hope that we believe is really leading the field forward.
Although I knew I did everything I could, the very best I could for my own father, it has become my passion to try to do things differently than we've always done.
The level of urgency that we have in the center for some people on the outside can seem a little hurried. But we just don't have the luxury of waiting. Every moment that passes, there's another Gila who is just engaging this for the first time. And there's another family like mine that's starting on that pathway. There's no time to waste. Today is our first patient for sonodynamic therapy in our phase 0 clinical trial. This is the first in human study. So sonodynamic therapy is a combination of a drug and a device. It's non-invasive, doesn't require an operation. A patient with a malignant brain tumor gets this drug the morning of the procedure, and then there is a frame placed on the patient's head. It's fixed to the patient's skull. It's done with local anesthetic, so it's painless. The patient doesn't feel any discomfort. And then following that, the patient goes into the MRI scanner and gets attached to the sonodynamic therapy device. Neuroradiologists, radiation oncologists, and neurosurgeons at the Ivy Center work together to identify the target and then deploy the MR-guided ultrasound to actually target those areas with the focused ultrasound energy. So for us, this is exciting new territory that's been unexplored to date, and this is the first time for any cancer type where sonodynamic therapy has really been investigated in a patient.
While recurrent high-grade glioma is a challenging diagnosis, scientists are always striving to develop new therapeutic options. One innovative approach under clinical study at the Ivy Brain Tumor Center is the use of sonodynamic therapy, a technology being developed by the company Sonalisence. In this approach, aminolevulinic acid, or ALA, is administered to the patient intravenously, crosses the blood-brain barrier, and enters the brain. Because glioma cells are more metabolically active than the neighboring tissue, ALA preferentially accumulates in the tumor, making it sensitive to light. Delivery of non-invasive MRI-guided focused ultrasound to the tumor produces a burst of light, killing the glioma cells without damaging the normal brain tissue. Learn more about the company behind this innovative therapeutic approach by visiting sonalisance.com.
We are, I think, uniquely blessed that we have teams that do nothing but focus on whatever illness it is that needs to be treated. Patients who come here, we review their situation with all of the disciplines. So it's a very comprehensive opinion. We have the luxury to take as much time as is needed to see a patient, to understand their specific individual situation, and to come up with the best possible options. We do have a lot of clinical trials to offer, and so I think those things are value added for a patient coming here.
My name is Christian Bader. I'm an assistant professor at the Massachusetts General Hospital and Harvard Medical School. I received a 2019 Discovery Grant supported by the Uncle Corey Foundation. The title of my talk is Therapeutic Targeting of Endoplasmic Reticulum Stress in GBM. Previous work from our group has shown that brain tumor environment exerts a form of stress known as the endoplasmic reticulum stress on brain tumors. And this stress, in fact, causes an elevated synthesis of fatty acid or lipid synthesis in glioblastoma. And this increased synthesis of fatty acid actually does promote protection for the cancer stem cells and the GBM in general, and it helps support brain tumor growth. So we asked ourselves a question: can we leverage these metabolic dependencies on fatty acid synthesis in order to develop therapeutic for GBM? And if you want an effective GBM therapy, you need three major components. First, you need an effective therapeutic target. Second, you need a drug that actually gets to the brain and hit this target. And third, you need a model that more or less accurately predicts the clinical outcome in patients. And these are the three major points that we focused on in our project. So first of all, the target. Our target is an enzyme called SCD. It's an enzyme involved in lipid metabolism. And essentially what this SCD does is that it converts a certain form of lipids known as saturated fatty acids, shown here in blue, into another form of lipids known as the unsaturated fatty acid, and shown here in red. Now, these red lipids are actually important for the cancer stem cells. They help them proliferate and they help the tumor grow. So what happens if we block this SCD? And here you can think of it as a kitchen sink model where SCD is actually the outflow. So if you block this outflow, first thing that's going to happen is you're going to deprive the tumors from these red lipids that they are really addicted to. But also what's going to happen here is you're going to flood this sink, and you're going to cause this major accumulation of these blue lipids. And when these blue lipids accumulate, they actually become toxic and they kill the tumor cells. So essentially here, our therapeutic strategy is to poison GBM tumors using their own metabolites. So our drug is actually a clinical grade inhibitor of SCD that was initially developed to treat Parkinson's disease. And this drug is currently in a phase 1 clinical trials in Parkinson's patients. And our model consists of patient-derived GBM tumor cells. These were collected from patients during surgery. They are brought to our lab. We grow them in the lab. We can at any given moment inject them using surgery into the brain of mice. They will form brain tumors. We can image these brain tumors, and we can also monitor the survival of the mice. And this is one way for us to look at therapeutic efficacy. So our project achieved two major outcomes. First of all, we were able to see on-target effect of this SCD inhibitor in GBM mouse models. So essentially, we confirmed that this drug gets to the brain and inhibits SCD in brain tumors in mice. But most importantly, we showed that there is a therapeutic efficacy of this SCD inhibitor in GBM mouse model. Essentially, mice that were treated with this SCD inhibitor lived longer. We also compared this therapy to temozolomide or Timodar, which is the chemotherapy given to patients. And both TMZ and SCD actually extend survival. But the best therapeutic outcome happens when you combine both these two therapies together. One important statistics here, if we look at long-term survival after 6 months of implanting those tumors, none of the control mice were alive. 25% of the temozolomide mice were alive, whereas 37% that were treated with the SCD inhibitor were alive. So these data overall is really encouraging, but there's still more work to do. Now, in terms of impact, from a research perspective, we were actually able to convert the ABTA support into roughly $1.8 million in funding through NIH and the Department of Defense, and that is to validate SCD therapeutics and to study the role of stress signaling in glioblastoma. Most importantly, from a clinical perspective, we now have a partnership between MGH and the biotech company that developed this SCD inhibitor. There are ongoing discussions to run a clinical trial to test this therapy in GBM patients. Finally, these are my acknowledgements. I want to thank the member of my lab who worked on this project, our funding, especially the ABTA, who has supported me for many years now. This is my contact information, and I thank you so much for your time.
Hello, everyone. My name is Odessa Yabud, and I am an assistant professor in the neurology department at UCSF, where I have been researching new therapeutics for pediatric and adult brain tumors. I'm honored to be here today and share some of our recent findings on targeting gliomas by exploiting DNA damage repair pathways. This research was funded by the ABTA Research Collaboration Grant in honor of Humor to Fight the Tumor. As the name suggests, the work I'm going to present to you today was done in collaboration with Dr. Julie Miller, who is a neuro-oncologist at Massachusetts General Hospital and Harvard Medical School. Julie and I embarked on investigating whether beta-lactone may be a candidate drug for a subset of gliomas, those that are marked with isocitrate dehydrogenase or IDH mutations. Briefly, let me just give you a background on beta-lactone. It is a naturally occurring compound extracted from the bark of this beautiful lapaco tree. And for generations, this bark has been used for medicinal purposes. In the last decade or so, studies have revealed potential anti-tumor effects of beta-lactone in several cancers, such as the ones listed here. I will present today some of our findings as to whether beta-lactone may prevent tumor growth in gliomas. This work was initiated when a preliminary drug screen indicated efficient induction of cell death in IDH mutant cells, which are the ones shown here in red, but not in IDH wild type cells, which the ones shown here in green. It became our goal subsequently to validate the efficacy of beta-lactone in inducing cell death in additional patient-derived glioma cells in culture and in glioma animal models. Our hypothesis was that beta-lactone induced cell death in the presence of high levels of this NQO1 enzyme. In the interest of time, let me just summarize that in the presence of NQO1, a series of chemical reaction increasing these ROS compounds leads to metabolic disruption resulting in cell death. We confirmed that beta-lactone resulted in higher ROS levels in IDH mutant cells, which you see here after beta-lactone treatment. We further confirmed dose-dependent increases in ROS levels after beta-lactone treatment in additional patient-derived glioma cells. But also surprisingly, we find dose-dependent increase in ROS levels in IDH wild-type glioma cells. Furthermore, the dose-dependent increase in ROS levels translated to increase in cell death, which is what you're seeing here, the decrease in cell numbers in IDH mutant cells, but also in IDH wild-type glioma cells after beta-lactone treatment. These findings were quite surprising given the variability in levels of NQO1, which are present in some sample glioma cells, but not in others. Thus, we still have to figure out the exact mechanism of beta-lactone's action in these cells. We also examined the efficacy of beta-lactone treatment in a glioma mouse model generated by xenografting or injecting patient-derived IDH mutant cells into the flank of the mouse. And then we treated them with beta-lactone for 5 days or for 20 days after the tumors have formed. These experiments showed a significant latency in the formation of, or the growth of tumor in mice treated with beta-lactone for 20 days, which is here in orange. These data are preliminary given the variations in responses in the control treated mice here in green. And so further experiments involving more mice will need to be completed to determine the significance of these observations. But what's promising is that analysis of these tumors showed high rates of cell death, where the beta-lactone treated mice have these white areas, and much of these areas have high levels of cleaved caspase staining, and the white areas are less obvious in the control mice. In summary, I've shown you that beta-lactone induced cell death is not uniquely occurring in IDH mutant gliomas, but also in IDH wild-type glioma cells. Thus, it appears that it's not dependent on high levels of NQO1, indicating a need to understand beta-lactone's mechanism of action in glioma cells. But nevertheless, we have a promising preliminary in vivo data where we observed the potential slowing of tumor growth and increase in cell death after treatment. Future studies are needed to determine if this effect will drastically increase in combination with other treatments available for gliomas. And with that, I'd like to acknowledge everyone who has worked on this project. I've mentioned Julie Miller, who has been such a great collaborator throughout this grant period, and our talented and very dedicated senior research associate, Thomas Noel, who's helped with performing the in vivo studies, as well as others who contributed to the project early on. Of course, we thank ABTA as well as the National Cancer Institute for their support, and feel free to contact me should you have any questions at this email. Thank you so much for your time.
My name is Aintron, and I'm a postdoctoral scholar at Northwestern University. I was awarded the 2019 Basic Research Fellowship in honor of Ned Smith and Tim Smith-Strong. Today, I would like to talk about my project, Tissue Factor as a Regulator of Receptor Tyrosine Kinases in Glioblastoma. Glioblastoma, GBM, is the most common primary malignant brain tumor in adults, and is one of the most aggressive cancers overall, with very high rates of recurrence and resistance to treatment. Patients diagnosed with GBM usually have a rapid onset of disease and progression, with a median survival of only 12 to 15 months from the initial discovery of the tumor. The treatment options for GBM tumors are limited, and there has been very little progress towards effective new therapies. Inside GBM brain tumor initiating cells, or BTICs, they express stem cell markers, so sometimes they are called cancer stem cells. BTICs are resistant to therapy, which is a problem because they can self-renew and initiate tumor. After treatment, non-BTICs can be killed. The BTIC will survive and cause the tumor to grow back. For both BTICs and GBM, receptor tyrosine kinases, RTKs, are very important. They are cell surface marker. You can picture an RTK as an ignition switch in your car. With the right key, a chain of reaction will happen to have your car start and running. Similarly, RTKs are the regulator of the normal cellular processes and play a critical role in the development and progression of cancer, including glioblastoma. Overactivation of RTK promotes malignant behaviors, including migration, invasion, proliferation, and tumor-initiating activity. The question is, how can we regulate RTK and tumor-initiating cells? We believe tissue factor could be the answer. Tissue factor is a protein originally known for its role in blood clotting process. When you have an injury, tissue factor will start the process to have the blood clot and prevent you from bleeding out too much. However, tissue factor can also activate protease-activated receptor 2, or PAR2, leading to effects unrelated to clotting. PAR2 is known to promote cell proliferation, Oncogenesis, and cell immigration. If it sounds familiar to you, they are the same effects that RTKs have. Our research hypothesis is that, similar to when a car is hardwired, tissue factor can activate PAR2, lead to activation of multiple RTKs and BTIC maintenance, leading to the promotion of malignant behavior in glioblastoma. We found that when we added tissue factor to GBM cells, many types of RTK was activated. On the other hand, when we removed tissue factor, we had opposite effects. We found that tissue factor could force RTK to activate even when we tried to inhibit this with RTK therapies. However, when we took PAR2 out of the equation, tissue factor became ineffective. Furthermore, we found that when we removed tissue factor from GBM cells, we decreased BTICs. You can see here that BTIC marker CD133 was much lower in cells without tissue factor, suggesting that the cells was less capable of initiating new tumors. And strikingly, GBM cells without tissue factor could not grow into tumors in mice, allow the animals to survive. Our project revealed a completely new way for the disease to evade treatment, high activation of RTK by tissue factor. This mechanism can support BTICs, which play an important role in therapy resistance and recurrence. PAR2 is an especially promising target for cancer therapies with minimal adverse effects. Overall, our results offer an exciting prospect to improve outcome for GBM patients by identifying a reason why current therapy fail. And with that, I would like to thank our team at Northwestern University, especially my mentor, Dr. Craig Kabinski, as well as our collaborators. But most of all, I would like to thank you for the support. this progress would not be possible without the help from the American Brain Tumor Association.
Well, I'm sitting in this chair because I'm still alive.
The day that we were told Chad had a glioblastoma, we started reading and we knew how serious it was.
I was notified that I had a mass, and my first question was, do I have a chance? Doc said, I don't know.
Dr. Wanabo told me that I had two options: surgery to remove the tumor alone, which he did not recommend, or surgery with gametile therapy, which would keep the tumor from growing back.
It really just targets the areas that need to be targeted and leaves the rest of normal brain tissue alone. He said that there'd been a lot of good success with it and not really have any hair loss, etc. I was very interested.
After I had the gametile procedure done in October of 18, I felt fine, as if it didn't happen.
I felt great. This is probably the best recovery I've ever had.
And I feel really good knowing that there's no ill effects that's going to come 5-10 years down the road. I expect to be here, believe that.
Gametile offers hope where there wasn't any.
This experience has been very gratifying. Since the gametiles were put in there, the surgeon has been very pleased with the results. So I've made him promise that I could be the poster boy.
Hello, my name is Marty Emerson. I'm the CEO of Monteris Medical. Monteris is the maker of the NeuroBlate system, a truly minimally invasive, MR-guided laser surgical tool used to ablate primary and metastatic brain tumors. The NeuroBlate procedure is performed under MRI visualization, which allows the surgeon to be very precise in targeting the tumor. Because the NeuroBlate procedure is so minimally invasive, a patient is typically discharged from home in 2 days or less. The NeuroBlate procedure has been used with over 3,000 patients and is supported by very robust clinical data. Monteris Medical understands just how important time is to those patients with brain tumors and to their families. Our objective is to deliver hope and healing for those patients with brain tumors. The following video is a brief overview of the NeuroBlate procedure. If you have additional questions, please contact the ABTA or visit our website, monteris.com. Thank you.
The NeuroBlate system is a minimally invasive, robotically controlled, MRI-guided surgical tool used to selectively destroy or ablate unwanted tissue in the brain using a laser. Laser ablation is possible when a focused area of brain tissue needs to be targeted. Your medical team will perform testing to confirm if you are an appropriate candidate and can discuss any questions you may have related to the procedure. Generally, because it is minimally invasive, the NeuroBlate procedure is well tolerated, has a short recovery time, and requires 1 to 2 stitches to close the incision. Your doctor will advise you of any pre-procedure requirements. You must be able to undergo MRI exposure. Shaving your hair is generally not required. A minimal area of hair may need to be removed. You will receive anesthesia, and you will be asleep during the procedure. The surgeon will create a small opening in the skull about the width of a pencil to allow access for the laser device, which is called a laser probe. The procedure is performed in an MRI machine. While you are in the MRI, the doctor will guide the laser probe through the opening. Because the procedure is MRI guided, the surgeon can see the specific area of the brain to be ablated. The surgeon controls the delivery of laser light energy to the targeted tissue. This laser light heats and destroys the affected tissue. The precise nature of the procedure helps to lessen the likelihood of harm to nearby healthy brain tissue. When you wake up after the procedure, you will have a stitch or two in the skin where the probe was inserted. In general, patients experience a short recovery time with minimally invasive procedures. You will schedule follow-up appointments with your doctor as instructed. Each person's situation is unique. Your doctors are the best qualified to let you know how a procedure may affect your outlook.
Where should you start when you're told you have cancer? Start with getting a second opinion at Cancer Treatment Centers of America, where nearly 7 out of 10 patients choose to treat with us after a second opinion. Get advanced diagnostics and precision treatment options from our team of experts who treat only cancer, and learn about treatment options you may not know exist. Because a second opinion might be the right one. Call now.
Welcome, and thank you for joining me in learning about Optune, an FDA-approved treatment for glioblastoma. I would like to thank the American Brain Tumor Association for allowing me to present to you. My name is Renee Adams, and I am the Thought Leader Liaison for Novocure. Today we'll learn about Optune, which is an FDA-approved wearable portable device indicated to treat a type of brain cancer called glioblastoma multiforme in adult patients 22 years of age or older. In newly diagnosed glioblastoma, Optune is used together with chemotherapy called temozolomide, or TMZ for short, if your cancer has been confirmed by a healthcare professional and you have had surgery to remove as much of the tumor as possible. In recurrent glioblastoma, Optune can be used alone as an alternative to standard medical therapy if you have tried surgery and radiation and either they did not work or they are no longer working, and you have tried chemotherapy and your GBM has been confirmed by your healthcare professional. Optune is not for everyone. Talk to your doctor if you have an implanted medical device, such as a programmable shunt, a skull defect, such as missing bone with no replacement, or a bullet fragment. Optune has not been tested in people with implanted medical devices, which may cause the devices not to work properly. Optune is not tested in patients with skull defects or bullet fragments, which may cause Optune not to work properly. A known sensitivity to conductive hydrogels, the gel on the arrays placed on the scalp, like the ones used on EKGs. When Optune comes into contact with skin, it may cause more redness and itching, or may rarely cause life-threatening allergic reactions. Do not use Optune if you are pregnant or planning to become pregnant, as it is not known if Optune is safe or effective during pregnancy. Optune should only be used after receiving training from qualified personnel, such as your doctor, a nurse, or other medical staff who have completed a training course given by Novocure, the maker of Optune. Do not use any parts that did not come with the Optune treatment kit sent to you by Novocure or given to you by your doctor. Do not get the device or transducer arrays wet. If you have an underlying serious skin condition on the scalp, discuss with your doctor whether this may prevent or temporarily interfere with Optune treatment. Some of the most common side effects of Optune when used together with chemotherapy, temozolomide or TMZ, were low blood platelet count, nausea, constipation, vomiting, tiredness, scalp irritation from the device, headache, seizure, and depression. The most common side effects when using Optune alone were scalp irritation, such as redness or itchiness, and headache. Other side effects were malaise, muscle twitching, fall, and skin ulcers. Please visit optune.com/safety for Optune patient information and operation manual, as well as complete information regarding the device's indication, contraindications, warnings, and precautions. Talk to your doctor if you have any of these side effects or questions. Please note that an Optune user or caregiver cannot provide you with medical advice because each person is different. Consult with your doctor for medically related questions. Today we will provide you with a general information about glioblastoma multiforme, Optune, and matters related to using Optune. For questions about your personal health or your treatment plan, the best person to ask is your doctor. Your healthcare provider can review how Optune works against your tumor and how it fits into the daily lifestyle to determine your best path forward. What is glioblastoma? Glioblastoma is also called glioblastoma multiforme, or GBM. GBM is a type of primary brain cancer. This means that GBM tumors begin in the brain rather than starting in another part of the body and then traveling to the brain. GBM is the most common type of primary brain cancer diagnosed in adults. Optune is a wearable and portable FDA-approved treatment for glioblastoma. Optune uses 4 adhesive patches called transducer arrays to deliver tumor-treating fields, or TT fields, to the location of the GBM tumor. It has been shown in clinical trials to safely deliver continuous therapy to the area of the brain where the GBM tumor is located. These transducer arrays are applied to the scalp and are connected to the device and battery. Optune treats where the tumor is. Glioblastoma tumor cells divide quickly, creating more and more cells that form a mass or tumor. As you can see in this illustration, Optune affects where the tumor cells begin to divide. When Optune is turned on, it creates low-intensity, wave-like electric field called tumor-treating fields. Using those 4 adhesive patches called transducer arrays, tumor-treating fields are delivered to the location of the GBM tumor. In this illustration, you can see that tumor-treating fields interfere with the dividing tumor cell. Tumor cell division may then slow down or stop, and some of the cells may even be destroyed. If you have newly diagnosed glioblastoma, Optune is used together with chemotherapy after surgery and radiation with chemotherapy. If you have recurrent glioblastoma, Optune is used alone once treatment options like surgery and radiation have been exhausted. In people with newly diagnosed glioblastoma, Optune is proven to extend survival. A large clinical study showed that by adding Optune to chemotherapy provided the greatest opportunity to live longer compared to chemotherapy alone at 5 years. In a 5-year follow-up, people on Optune plus chemotherapy lived longer across all groups analyzed, regardless of well-being, age, gender, and how much of the tumor was removed. Additionally, nearly half of people using Optune plus chemotherapy were alive at 2 years, compared with 31% of people on chemotherapy alone. Continuous treatment with Optune can be received almost anywhere. If your doctor says it's okay, you can use Optune while you go about your daily routine at work, while traveling, and at social functions. Transducer arrays need to be applied directly to your scalp to work properly. You will need to shave your head and change the arrays at least 2 times a week, or every 4 days at the most. To prevent transducer arrays from getting wet, cover them with a shower cap when bathing or showering. Optune should be worn and turned on for at least 75% of the time, which equates to about 18 hours per day, to get the best response from treatment. As you can see in the photos of these actual patients, head coverings are optional. It is important to select a head covering that is breathable and lightweight. For a complete list of these head coverings, please visit optunedailylife.com. Novacure offers webinars throughout the year to help you stay informed and inspired. Optune users and caregivers will discuss their experiences living with GBM and integrating Optune into their lives. You'll also have a chance to ask questions and discuss other topics about Optune. To find out more or to register over the phone, call 1-844-247-1636 or visit optuneopenhouse.com to register for a webinar today. Our Encompass Complete Support team supports you every step of the way, and we provide customized support based on your need. We offer 24/7 technical support via phone. They're able to reorder supplies, answer ongoing questions about Optune, and offer travel tips and resources. Additionally, the device support specialist, another member of the Encompass team, delivers Optune, provides training to help you get started, and shares tips and resources on how to incorporate Optune into your daily life. Additional benefits of the Encompass team are that they can help provide information on Optune and how it works, answer questions about the potential benefits and side effects of Optune. Thank you so much for joining, and I hope you found this education beneficial.
I have never been more hopeful than I am today about the future of brain tumor therapy. There are 3 pillars for care of brain tumors. One is the surgical pillar, the second is radiation oncology, and the third is medical therapies and neuro-oncology. When it comes to surgery, our approaches are getting less invasive. Our ability to individualize our brain maps is getting better every day. New therapies are coming online, including more advanced intraoperative MR techniques, robotics, augmented reality, and fluorescence.
Hello, everybody, and welcome back. I hope each of you enjoyed your lunch session, were able to take a little bit of a break and grab a snack. We will now resume our afternoon session. Our co-chair, Dr. Eva Galanis, will introduce our next presenters.
Good afternoon, everyone, and thank you for joining us in this session. I hope you found the morning sessions interesting. Thank you so much for your attendance, your engagement, your insightful questions. So we heard this morning about molecular classification of brain tumors and advances made in that. In this next session, we will learn how genetic alterations in tumors can be used to create new personalized patient-specific treatment options. We're really honored to have with us today two pioneers in this field. One is Dr. Ingo Mellinghoff, who is professor and chair in the Department of Neurology at Memorial Sloan Kettering Cancer Center. And the other is Dr. Priscilla Brastianos, an associate professor at Harvard and director of the Central Nervous System Metastasis Center at the MGH. So in addition to them being brilliant researchers and clinicians, they also have compelling stories as to why they're drawn in neuro-oncology. So in order to get to know them a little bit better, I asked them not only to discuss their scientific part, but also tell us a bit more about themselves and what is that inspires them in their work. So without any further ado, please welcome Dr. Mellinghoff and Dr. Brastianos. And Dr. Mellinghoff will present first. So Igor, welcome.
Thank you very much. Thank you very much, Eva, for this very kind introduction, and to you and Sean at the ABTA for putting such a great program together. It's a real pleasure to be here. Thank you for the invitation. And I've greatly enjoyed what I've heard so far in this morning session, and I'm trying to build on what you have all learned about neurosurgery, pathology, and we're taking it more into the direction of targeted therapy now with Priscilla in my presentation. I am, in terms of, by the way of background, I came up through the oncology side of things. And I was studying, after finishing all my medical training, studying in a research laboratory at UCLA at the time, how a lot of these signaling within cancer cells are connected and how that could be exploited for cancer therapy. And that was in the late 90s, I'm dating myself. And that kind of inspired me because glioblastoma seemed perfect and prime to exploit some of that knowledge about signal transduction. And so I took my research into squarely the direction of glioblastoma and glioma signaling. And that's kind of where I still am, trying to develop a targeted therapy for glioblastoma. I'm at Sloan Kettering now since 2007, and I'm again really honored and to be here and invited. Thank you.
You should now have control of the slides.
Yep, these are my disclosures. Got it. Okay, so again, let's start with this slightly dense cartoon. This shows you really the inner wiring of a cancer cell. And there's a lot of detail on this slide, which usually means you don't need to pay attention to the detail. I just want to make a few very general points. What this slide shows you is that the cancer cell takes an information from its environment, and that usually happens through something on the surface of the cancer cell, like a growth factor receptor. And then that information is taken through a series of signaling nodes toward the sort of, center control, which is the nucleus of the cell, which tells the cancer cell to divide or take on a different morphology or even kill itself. So those are sort of the key steps, how signal transduction links the cancer cell to its environment, its tumor environment. And that also is sort of the foundation how we try to influence the fate of the cancer cell by switching around some of these signaling nodes, increasing the signaling or stopping the signaling. And the other piece of information that you see on this slide is there are a lot of 3-letter and 4-letter words in there. And these are all the names of sort of targets. These are generally proteins that are able to transmit a signal, receive a signal and transmit it further down. And all the 3 or 2-letter or 4-letter words that are in red on these slides are proteins that are altered in cancer. And what you can see here is that there are a lot of different ways that cancer cells can manipulate the signaling network. Now, this is sort of the reality, exactly what a broken cancer cell genome looks like. You've seen earlier a similar figure by Dr. Brett when he was introducing the pathology of brain tumors. And what this slide shows, each row on this slide represents one of these signaling nodes, and each column represents a tumor. So in this particular case, you're looking at the genome from 649 different GBM tumors from 600 different patients. And what you can see, there are 2 points here I'd like to make with that slide. One is that there are a lot of little differences. It looks a little bit like a mosaic, and every tumor really is unique in its genetic profile. Sometimes you see an alteration in NF1, just to go through some of these, or RB1, and other times you see an alteration in PTEN or EGFR. So it really is a unique sort of mosaic, is the cancer genome. But the other important point is that there are patterns. There are patterns of ways in which these tumors arise. So there are 2 or 3 alterations that tend to be associated with each other or mutually exclusive. And it is that type of pattern that we use to sort of come up with an educated guess what might be a suitable targeted therapy for you based on this information. And this is the type of information that you get in these molecular reports that you heard earlier about. The type of either, so we're having our own in-house tumor sequencing panel. It has about 500 genes. Some other centers also have an in-house panel. And there are the commercial partners, like Foundation Medicine is a commercial provider, Caris, or other centers. But that is the type of information we get from the molecular tumor profiling. And here is now the important, amazing thing. Even though each tumor clearly has more than one genetic alteration, when you block a certain one of these alterations, that can be enough for the tumor cell to kind of go away, either change its shape or growth potential completely, differentiate into almost like a normal cell, or also commit suicide and die, which of course we like either one of those 2 responses. And the key challenge, of course, is to know which of these alterations is the one to go after. In this case, would it be A, B, C, or D? In this particular case, it would be A. And this is this idea of molecular addiction now in practice. This is a cover of Time magazine from 20 years ago, as it turns out. It was on the cover on May 28th, 2001. And that was the first time that this idea of targeted cancer therapy really came through very strongly with a pill called Gleevec. Now, it was the very first example of a highly precise molecularly targeted therapy of cancer. And what was kind of revolutionary here is it is a targeted treatment that specifically hurts the tumor cell and not other cells in the human body. And also it is a pill, as opposed to many chemotherapies for other cancers, which tend to be chemotherapies that are given intravenously, at least at that time. And fast forward 20 years now, this idea of targeting the cancer with a very specific, precision medicine pill actually has stood the test of time. And there are now a large number of similar drugs against many of the signaling nodes I have shown you earlier in this cartoon. This sort of garbled series of 2- and 3-letter words. There are signals that we can now block with FDA-approved drugs in many of these cancer types. And this, of course, is the reason why we are sequencing a lot of the human cancers now as sort of standard operating procedure. So how about brain tumors? We have, we meaning our group and many other groups represented here today in this symposium, have tried to take these lessons with targeted therapy to an alteration that is particularly common in glioma, and that is IDH. You've already heard quite a few things about IDH today. It is a metabolic gene. And the reason why we're so interested in IDH is because most low-grade gliomas have a specific point mutation in either IDH1 or IDH2, usually IDH1. And people became very interested in IDH early on as soon as it was discovered, because it seems to happen very early during the development of these tumors. And it seems to stick around even though things change over time. And also it is associated with a specific prognosis and epigenetic alterations. So all of these observations suggested that this could be a drug target for low-grade glioma. And these sort of insights fueled clinical trials with first-in-class inhibitors of mutant IDH in low-grade glioma. And this is an example of a patient in one of these early phase clinical trials. In this particular case, I'm showing an example of a patient treated with vorasidenib, which is now in phase 3. And what you can see in the middle is the MRI scan prior to starting the study drug. And then on the right, you see the MRI scan after, many months on study drug. And to the left, you see the MRI prior to going on study. And of course, we can now measure these volumes much better than we used to. And you can then track sort of the trajectory of the tumor as it grows. And once you start the investigational treatment, if it changes. So that's very encouraging. One other major, I would say, stepping stone for us as we develop these targeted therapies is to develop an approach to monitor the evolution of the tumor in spinal fluid. So you've probably heard that so-called liquid biopsies are a very hot topic now in cancer, and many cancers where oncologists take a blood sample and try to get a little bit of tumor DNA out of the blood and then get an idea whether the tumor has changed or has become resistant to a targeted therapy. Blood samples have not been that informative so far for patients with brain tumors, I would say. But CSF, cerebrospinal fluid, is a unique fluid. As you all know, it sort of flows around and in the brain. So it's natural that that would probably be the place to find tumor DNA in brain tumors. And this was the example where we have really looked in spinal fluid in glioma patients for the presence of tumor DNA. And the long and the short of it is that in about half of our patients, the being able to detect tumor DNA in spinal fluid has, number one, been a very representative snapshot of the tumor genome at the time. And we know this because we had patients who we had collected spinal fluid and done surgery immediately or shortly thereafter, so we could compare it. So it does provide, we think, a good snapshot of what's going on in the tumor. And we think this could be a very useful tool. This is now increasingly done, and the technology is advancing rather quickly. So we're hopeful that this will be useful to monitor the genome. And you have heard earlier that from the pathology lecture that the genome can evolve modestly. It is true that not everything changes in the genome over time, but certain things do change. And oftentimes it is exactly what the drug targets that does change. And so for that reason, we believe it is a useful exercise to really develop this further. So in summary, I would like to pose the somewhat provocative hypothesis that we are actually just at the end of the beginning for targeted therapy for glioma. We really have come a very long way in the last 5 to 10 years. It is clear that we are much more accurate now in subgrouping glioma into prognostically distinct subtypes, and that's very important if you're trying to interpret clinical trial results. It is also clear that we have now access to technology that allows us to profile tumor and fluid in really unprecedented ways at a very high resolution reproducibly. So some of these exploratory technologies have now really graduated towards clinical grade technologies, and I think they will be used increasingly. And you already heard earlier about DNA methylation profiling. I didn't even put that on the slide yet, but that is another sort of frontier of molecular profiling. And having a better understanding of these disease subgroups and having access to these cutting edge technologies to really learn more about these tumors as they evolve is a critical ingredient as an entire set of new classes of drugs are entering cancer medicine. These bispecific T-cell engagers, antibody drug conjugates, there are lots of new drugs that are coming down the pike, and it's really wonderful to be able to build a framework around how to develop these drugs and take the relevant measurements, molecular measurements, to help us guide development of these drugs. And the last really great source of optimism for me is that we have also become much more efficient in the clinical trial development space, how we design clinical trials. We used to always have a control cohort in each trial, and that is important typically to develop drugs and understand if they're working. But there are now modern ways of designing and developing trials that have an adaptive design where the likelihood of ending up on the investigational drug is higher if the investigational drug starts looking ever more promising as the trial goes on. That's an adaptive randomization. And there are a whole series of other clinical trial design changes that you will hear about later today and also tomorrow. So I think these are all really exciting developments that I think make all of us optimistic that the landscape of treatment for primary brain tumors, metastatic brain tumors, will really fundamentally change in the next 5 to 10 years. So with that, I would like to close and thank you very much for the opportunity to present here.
Thank you so much. And thank you so much for the kind introduction, Eva, and for Sean and Eva for inviting us to speak today, and for ABTA for putting together this amazing conference every year. It's such an important conference, and I love being a part of it. So thank you again. So these are my disclosures. But now that Dr. Mallinghov has introduced precision medicine, I'm going to talk about 3 different examples where precision medicine is showing promise in brain tumors. So I'm going to start with precision medicine approaches for meningioma. So meningiomas are the most common primary brain tumors, representing about 36% of all primary brain tumors. And typically, meningiomas are managed or have been managed primarily surgically. However, they can occur even after surgery. So there are 3 grades of meningiomas: grade 1, grade 2, grade 3. And grade 1 is the lowest grade. And even among grade 1 meningiomas treated by surgical resection, 20% can recur. And resection of grade 1 meningiomas at difficult locations can be associated with higher recurrence rates. And recurrence rates are higher for the grade 2 and 3 meningiomas. And about a third of patients only have partial resections, and 5-year overall survivals are decreased, can be decreased in this patient population. Sorry, my pager just went off. Sorry. Radiation is frequently used as an adjunct to surgery. However, there aren't any effective chemotherapeutic options when surgery and radiation fail to offer long-term disease control. So here's a summary of studies, a busy slide, so I'm not going to go through this in too much detail, but here's a summary of studies of different drugs that have been investigated in recurrent meningiomas. So these are meningiomas that have progressed after surgery. Historically, they have not been very successful in general. And prior to a few years ago, what did we know about the genetics of meningiomas? So you heard about gene mutations earlier from Dr. Mellencamp's talk and from earlier talks. So we knew that this gene NF2 is disrupted in about half of meningiomas, and the loss of this chromosome occurs in 40 to 70% of meningiomas. However, drugs that target this gene NF2 have not historically been effective, and a poor understanding of genetic drivers of meningiomas really has limited the development of therapeutic agents to supplement surgery and radiation. But in this new era now of genetics and genomics, that's likely going to change. And what I hope to tell you about is that there is lots of promise in primary brain tumors. So the question we ask is, do meningiomas have clinically actionable mutations, meaning mutations for which there are drug targets, as you heard about? And I'm going to briefly go through some of the key findings of significance from the last few years. So meningiomas have what are called AKT mutations. So these are the same mutations that have been known to be targets in breast cancer, colorectal cancer, lung cancer. And this is just a map of the gene and where we see the mutation. We see the same mutation in the same area. And AKT is a member of a signaling pathway that increases cancer cell survival. But this is a known targetable mutation in multiple cancers, and there are inhibitors in clinical trials right now, so that makes it exciting that meningiomas have this. There are mutations in another gene called smoothened. And again, similarly, this is a mutation that has been described in other cancers, and mutations in this pathway has been linked to other cancers, and there are FDA-approved inhibitors in other cancers. So again, represents a potential target for meningiomas. So based on this, we've started a multi-center clinical trial sponsored by the NCI Cooperative Group, the Alliance, and we're focusing on meningiomas that have progressed after surgery and or radiation. And this is actually a trial that we're doing in collaboration also with Dr. Agonists. And we screen specific mutations, specific tumors for mutations. And then if they have that drug, if they have that mutation, the patients receive that inhibitor. So this type of trial design, where you're testing the tumor for mutations, then treating with specific drugs, is called an umbrella trial. And this trial is already showing some promise. So we've completed accrual to one arm of this study, this particular arm, so the NF2 arm. And an early analysis of this arm shows that the percent of patients who did not progress at 6 months was higher than what one would expect compared to patients who had been enrolled to other previous trials. So here are the actual numbers here. So this is quite promising, and the trial is continuing to enroll patients. So just to summarize, genomics uncovered potentially clinically significant alterations. A national multi-center umbrella trial is underway, and target therapies will likely play an increasingly important role in the management of meningiomas. So let's switch to a different type of brain tumor called craniopharyngioma. So I've shown some promise in meningiomas. We still have a long way to go, but there is promise. So let's talk about this other brain tumor called craniopharyngiomas. So craniopharyngiomas, brief overview, are rare tumors that occur in children and adults, and they can cause significant impairment through compression of critical structures and side effects of treatment. And they're usually treated with surgery and or radiation. And like meningiomas, medical treatment hasn't historically been that effective for this tumor type. Classically, there are 2 subtypes, adamantinomatous, so that rolls off the tongue, big word, and papillaries. Adamantinomatous tend to occur in children and adults, and papillaries tend to occur in adults. And to date, these two subtypes were managed in a similar way with surgery and or radiation. And historically, drug treatments have not worked. So in a study from a few years ago, we showed that the different subtypes are characterized by single mutations, meaning single genetic changes drive these tumors. So the adamantinomatous type had one type of mutation, beta-catenin, and the papillary tumors had BRAF V600E mutations. So 95% of those tumors had those mutations. And you heard a little bit about those mutations earlier. And because BRAF inhibitors have shown efficacy in melanoma, we started a multi-center phase 2 trial in craniopharyngiomas, looking at BRAF and MEK inhibitors in these patients. So cohort A, newly diagnosed, and cohort B, recurrent. So here are the results of the study so far. So we finished accrual to the newly diagnosed papillary craniopharyngioma cohort, and 15 out of the 16 patients who received those drugs responded to therapy. So the one non-responder, the patient who did not respond, unfortunately, to therapy, only received 2 days of treatment before having to stop therapy due to toxicity, unfortunately. But the responses were quite dramatic. And here is an example of a response. So here is an example of a craniopharyngioma, papillary craniopharyngioma. The patient had a BRAF mutation, so we started the BRAF MEK inhibitor, and you can see this dramatic response at 8 weeks. So we're still completing the trial, but to summarize, this particular type of brain tumor have BRAF V600E mutations. These inhibitors demonstrated a remarkable response in patients with papillary craniopharyngioma thus far, and we're doing this clinical trial now to further evaluate the role of BRAF MEK inhibitors in papillary craniopharyngiomas. And finally, the last 5 minutes of my talk, I wanted to talk briefly about precision medicine for another type of brain tumor, brain metastases. And brain metastases are when cancer, as you've heard, has spread to the brain from other parts of the body. So how are brain metastases managed? So first, let's say brain metastases are an unmet clinical need. So they're the most common malignancy of the brain. Most are from lung, breast, and melanoma. And this was actually an ABTA-funded cross-sectional survey that we finished, where we actually surveyed physicians, patients, and caregivers, and they all indicated the need for increased treatment and research funding for brain metastases and more clinical trial availability. So this is definitely an unmet clinical need, and we're really grateful to ABTA's support for helping us with this huge clinical issue. So how do we manage brain metastases in general? They're managed with, so surgery and radiation are used for local treatment of brain metastases. Many chemotherapies in general have not traditionally been able to cross the blood-brain barrier. Some of the newer therapies that you've heard about, immunotherapies, are revolutionizing the treatment of many cancers, including in brain metastases, specifically for melanoma. And targeted therapies are showing promise in patients with brain metastases, such as HER2-positive breast cancer, BRAF mutant melanoma. But there are still challenges of precision medicine and brain metastases, and there's lots of research in this area. One is, in the past, there have been limited number of prospective trials, and many people now are trying to fix that and run more trials in this area. Many of those trials were smaller, underpowered, meaning they weren't necessarily powered for us to be able to look at the effects of specific drugs. Most patients with brain metastases, unfortunately, have already progressed on several therapies, so often their cancer is more difficult to treat. And importantly, scientifically, the extent to which brain metastases share the genetic profile of the primary tumor is unknown, meaning how similar is the brain met to the primary tumor. And that takes me to some of the ABTA-funded research that we've done, where we've genomically characterized brain metastases. So ABTA provided us with the funds to initiate this massive collaborative effort to study tissue samples from brain metastases. So we created a tumor bank of brain metastase samples and initiated prospective collections of brain metastases and primary tumor samples to study brain metastases and to understand how genetic changes are different in the brain metastases compared to the primary tumors. So this is our ongoing collaboration. We have institutions from all over the world participating. We're hoping to get as many tissue samples as possible, particularly from patients of different ethnicities and race also. And what we're doing is we're really trying to understand, again, how brain metastases are different from their primary tumors to find those drug targets. So in this first study that was funded by the ABTA, we comprehensively characterized brain mets matched with primary normal tissue, 104 samples. And for each matched brain metastasis and primary tumor, we map out the genetic relationship between the brain metastasis and the primary tumor, meaning we're trying to understand where the different genetic alterations occur. Are they in the primary tumor? Are they in the brain metastasis, or are they shared? And what we find across all the cases, that there are new mutations in the brain metastasis and new mutations in the primary tumor. So there are different drug targets in the brain metastasis compared to the primary tumor. So how will this influence our clinical understanding of brain metastases? So the question we're asking is, do brain metastases have different targetable mutations that are not in their primary tumors? And here's just one example. So this is a patient who had a HER2 positive breast cancer with the brain metastasis that developed after she had gotten targeted therapy with trastuzumab, which is HER2 therapy. And we found that the brain metastasis, the primary tumor, there were some shared mutations, but all this red, these were new mutations in the brain metastasis. So this patient had an EGFR mutation in the brain metastasis, which is probably what made this brain metastasis resistant to her therapy. So I just told you about these differences. Were there common changes so we can start thinking about clinical trials for brain metastases? So we found more than half the cases had alterations in this, what's called the CDK pathway. So there are CDK inhibitors that target this pathway. And also PI3 kinase signaling pathway and alterations. So about 40% of cases had alterations in the PI3 kinase pathway. So these all represent potential targets for clinical trials. So based on this, we started a national biomarker-driven trial in brain metastases. So specifically, we're targeting again for the pathways that are altered specifically in the brain metastases of patients with these tumors. So here's the study design. So this trial is now activated at more than 300 hospitals throughout the US. So in conclusion, what I hope I conveyed to you today is that mapping the genetic profiles of primary metastatic brain tumors is critical for uncovering therapeutic targets, and precision medicine is a promising approach for primary and metastatic brain tumors. And I know Eva had asked us to talk about our personal stories. So my personal story and what drives me is, so my grandmother was diagnosed with metastatic breast cancer in her 20s and passed away at the age of 29 of metastatic breast cancer. And I grew up hearing her story. And later, my mother also developed breast cancer and died of metastatic breast cancer. And she was quite young, in her 50s. And the stories of those two women really drive me every day. Seeing how devastating metastatic cancer can be fuels my energy every day, and we need to find better treatment options for patients, and that's what drives me every day. So on that note, I'm looking forward to questions.
Thank you both so much for your wonderful presentation. As a reminder, if you have questions that you'd like to ask, please type and submit it using the Q&A button on the bottom of your control panel, and our presenters will answer questions as time allows. So one of the first questions that we have here is asking, we've been talking about molecular profiling and genetic markers. What do genetic markers tell us about survival, about chances of tumor progression, and how likely a patient is to go into remission or no evidence of disease after treatment?
Yeah, that's a great question, and I can start and then Ingo also. So yes, there are specific genetic changes that can be associated with prognosis and survival, and different tumor types will have different specific mutations. So it's certainly something that that is an active area of research, and for which there are specific genetic mutations and biomarkers that can that can tell us if there's a tumor that's more aggressive than others. And Ingo, I don't know if you wanted to add to that too.
Yeah, I think this is really the holy grail of targeted therapy, that question of so-called predictive biomarkers. So there, there's two types, just a little more general comment. There are two types of biomarkers that we distinguish carefully. One is prognostic, which means, does the presence of the mutation make the disease different, better or worse, in the absence of any particular treatment, just in general. We call that prognostic. A predictive biomarker is something different. It's specifically tailored to a certain therapy. So in the case Dr. Bastianos presented with the BRAF mutant craniopharyngioma, where she showed like 16 out of 17 patients responded, that seems like a pretty good, probably predictor for response, even though to formally prove it, you would really have to design larger studies and have biomarker positive and negative sort of groups and so on. So it's hard to sometimes tease it out. In other words, there are some genetic alterations that are prognostic. You heard earlier from Dr. Brett, like IDH mutation, for example, is an example of a genetic marker that has a strong influence on prognosis, and maybe also is predictive for response to IDH mutant inhibitors. That would be the current thinking, but that remains to be proven. So it's really important question. And by and large, I think, despite the enormous amount of genetic profiling we have done and continue to do, there's a short list of genetic alterations where we would really commit to calling that a reliable predictor of treatment response or outcome. So much is still to come out, but on the positive side, I think our field and the cancer at large is really focusing toward what people call data science, where we take in a lot of information that individually we find difficult to process, like, for example, entire genetic profiles from 500 different genes, and also MRI, tumor volume, growth rates, and so on. And we're hoping that we will be smarter in 5 years. And Priscilla has shown you some beautiful examples to really isolate and prove certain predictive and prognostic biomarkers through that sort of data science approach.
Thank you so much. This next question is actually specific for Dr. Brastianos, but Dr. Mellinghoff, please feel free to jump in. This question is asking, are you aware of any targeted therapies that are being studied or that have had success for GBM that is IDH wild type?
So that's another very active area of research, and I know Ingo talked about gliomas. So if you want to talk more about gliomas, Ingo. Otherwise, I'm happy to answer it too.
Yeah, not many, I guess is the short answer. A lot of work, but no success stories yet. For example, you heard from Dr. Brastianos about BRAF. For example, V600E is one specific common type of alteration in a gene called BRAF. So even in glioma, and even there's a subset of glioblastomas, for when they carry that alteration, they're more likely to respond. I would say, I would go that far. For many of the others, such as EGFR, we're still struggling to really commit any kind of reliable signal out of targeted therapies. So I would say short list right now. Priscilla, do you agree?
Yeah, I would say, I was going to say exactly that. It's, we're still working on it. I think as a field, we're still working on it. I think that BRAF is promising. But it's definitely an active area, and there's many clinical trials right now evaluating the biomarker-driven approach for gliomas. So still a work in progress.
And part of the problem is the frequency of some of these alterations is very, very low. For example, TRAC alterations, fusions in a gene called TRK, TRAC, less than 1% probably in GBM. So even proving, like finding the patients at the right stage of the disease to even ask whether that kind of therapy would be suitable is difficult to do. So I think there's a little bit of a logistical challenge even for these very rare sort of almost unique molecular flavors. FGF receptor TAC fusions are another one where there are kind of promising agents, but developing this in a very sort of efficient way, I think we struggle as a field. And that is one of the reasons why I think consistent molecular profiling and being plugged into a clinical trial environment would help everyone, I think, move this forward.
And there's certainly really promising sort of biomarker-driven glioma trials that are sort of national and international collaborative efforts that are, I think, going to hopefully pave the way for better treatments for IDH wild-type gliomas.
And different drugs, just one last comment is, the pharmacology kind of matters. Like for example, with the BRAF inhibitors in melanoma, there were a good 5 years where everybody thought this is not the right concept because the first generation of drugs failed. And there was, the sort of tide was swinging toward a negative view of all of this until the pharmacology was figured out and improved. And the drugs were able to really turn off that signaling node efficiently. And all of a sudden, there was a bit of a sea change that, oh yeah, mutant BRAF is in fact a target. So I would not be too discouraged by our current inability to target these signaling nodes. I think there are different drugs, these antibody drug conjugates and so on. So there's a lot going on there. And I think we have at least, Priscilla, a pretty good list of what we want to do, right?
Yeah, and also I think the other issue I want to bring up is, even if we have the right drug target, it's that blood-brain barrier and, making sure that drugs get into the brain too. And so that's always been an issue. But certainly now a lot of the companies are working towards developing more CNS penetrant or central nervous system penetrant drugs. So the issue with brain tumors in general is, you want the right target, but you also want the drug to get into the brain too. So all those are active research efforts in gliomas and also non-glioma tumors too.
Thank you so much. I think we have time for one more question. You had mentioned in your presentation the importance of liquid biopsies. Can you expand on that? Can you tell us a little bit more about what a liquid biopsy is and what's the purpose of that?
Yeah, a liquid biopsy is, any kind of biospecimen that is not an actual piece of tumor, so to speak. So it could be a blood sample, it could be a urine sample, it could be a spinal fluid sample. And the idea is that many tumors shed a little bit of that DNA that's in the tumor into other sort of body fluids. It kind of leaks into it. And depending on the type of tumor, the blood, you can find it in the blood, or you could find it in the spinal fluid, or you can find it in the urine. That's the general idea. For bladder cancer, you would say, let's try the urine. For brain cancer, maybe spinal fluid. Maybe for metastatic lung cancer, you check the blood. So there are some sort of obvious sort of first choices. What type of liquid biopsy might be helpful for brain tumors? There's a lot of excellent work from Hopkins early on. It depends a little bit, probably how you collect the fluid and where the tumor is relative to the spinal fluid within the brain. So there are like some things that need to be sorted out. But generally speaking, I would say it has been shown now by multiple groups that this is a productive way to try to get tumor DNA and not having to do surgery. That's the main idea. To obviate the need for a neurosurgical procedure by looking somewhere else in a sort of longitudinal regular way, sort of monitoring the tumor. I think that's the idea. And Priscilla, do you share that optimism on the liquid biopsies?
I do. I think liquid biopsies, both from spinal fluid and blood, ultimately will hopefully be able to minimize some of the more invasive surgical procedures. So absolutely, I agree with you, Ingo.
Thank you so much. That is all the time that we have for questions for this session. Thank you both for your help. I'll turn it back to Emily.
Yes, thank you so much, Dr. Mellinghoff, and thank you so much, Dr. Brastianos. And we appreciate you sharing why you have chosen this career and why you were passionate for it. So thank you so much for what each of you do. We will now take a quick break, and please feel free to do so. If you need to use the restroom, grab a snack. We invite the rest of you to stay on here to listen to a research update, and we will return in just a few minutes with our brain tumor board. Thank you so much.
Good afternoon. My name is Trang. I'm a postdoctoral research scientist at Columbia University. Today, I would like to introduce the project Activation of LXR beta inhibits tumor respiration and is synthetically lethal with the BCL-X long inhibition. These projects were awarded by American Brain Tumor Association Basic Research Fellowship in memory of Katie Monson in 2019. So in this project, I will combine two different drugs that target two different angles to reduce the tumor growth in the GBM cell. Cholesterol is a major component of membrane, and the brain contains nearly a quarter of the total cholesterol in the body. So first drug, I'm going to target about the cholesterol. So I'm going to use the LXR623. This will activate the liver X receptor. This is the regulator of the cholesterol, fatty acid, and glucose homeostasis. When LXR is activated, the ABCA1 will be produced, and this is a major exporter of cholesterol-containing lipoprotein. LXR623 treatment also limits cholesterol uptake which leads to a drop in the intracellular cholesterol supply and driving to the cell death. The BCL2 family members play an important role to induce cell death also. And then upon the stress stimulation, the BH3 only protein such as NOXA, it will be activated to induce the apoptosis by releasing PAX and PAX. One PAX and PAX is released, so the cytochrome C will be activated, and then the cascade will be active to induce cell death, or we call the apoptosis. The second drug I'm going to use is the ABT263. It will bind to the BCLX lung to facilitate PAX to induce apoptosis or cell death. The recombination treatment between the LXR623 and ABT enhance more cell death compared to the single treatment, and we can also observe in different cell lines. The LXR623 treatment will enhance the NOXA protein level, and then when we silencing of NOXA, we will observe the attenuation of the recombination treatment. Since NOXA bind to MCL1 to facilitate PAX, we also silencing of PAX. Silencing of PAX also protect the cell from death by the recombination treatment. So we conclude that NOXA and PAX are the key elements for the combination treatment to assert the cell death. Next, we also found that LXR623 treatment will reduce the ATP level. The ATP is the source of energy for use and storage in the cellular level. And we also observe the enhance of the ATF4. This is the marker to show the stress stimulation, and then this phenomena will be rescued in the presence of the ATP or energy and in the presence of the cholesterol. Silencing of the ATF4 will reduce the NOXA protein level upon of the LXR623 treatment. LXR623 treatment also reduce the oxygen consumption rate and also the ATP level by the extracellular flux analysis. And this phenomena will be rescued in the presence of the cholesterol. And finally, we apply the recombination treatment into the mouse model, and we observe that the recombination treatment will enhance the growth reduction compared to the single treatment. For the future direction, we plan to apply this drug to test in the human slide culture to see the effect of this drug in the human brain. And then if the efficacy is good, we plan to start for the clinical trial. And with this, I would like to acknowledge my mentor and all of the lab members to support me with this project. And thanks for this award. I have some collaboration with other labs at Columbia University and also in the Weill Cornell Medicine. And with this, I conclude my presentation today. Thank you for your attention.
Welcome back. In this next session, you will listen in as a multidisciplinary panel of expert clinicians simulate the review of brain tumor cases to determine the best course of treatment for patients. This session really shows you how complicated care decisions can be. It is my honor to introduce you to our panel facilitator, Dr. Mark Gilbert. Dr. Mark Gilbert is the Chief and Senior Investigator of the Neuro-Oncology Branch Center for Cancer Research at the National Cancer Institute, National Institutes of Health. Some of you may recognize Dr. Gilbert as he served as our national conference co-chair last year. We are so honored and excited to have him again returning this year as a speaker. Please welcome Dr. Gilbert, who will introduce all of our panelists.
Great, thank you so much. I hope you can hear me okay.
I can hear you just fine.
Great. So it's a great honor and privilege to return this year. And one of the highlights of our meeting last year was the Brain Tumor Board. And as Emily indicated, this is a very common occurrence at many of the medical centers that our patients get treated at. And we wanted to give you a glimpse of what it's like to actually be present at a tumor board. So we've put together this event, and we're very fortunate to have just some outstanding faculty. I think I should have control. Do I have control? I do. All right, so joining me today are some outstanding colleagues. So Dr. Nancy Bush, who's a neuro-oncologist at the University of California, San Francisco. She will be our neuro-oncologist. Dr. Alfredo Quinones Hinojosa, a very prominent neurosurgeon at the Mayo Clinic. Dr. Ranjit Bindra, a radiation oncologist at the Yale School of Medicine, and Dr. Daniel Bratt, a neuropathologist at Northwestern University. And what we're going to do is go through a couple of cases, present them, we'll stop at various points. And again, for the folks listening in on the tumor board, it's really less important to kind of focus in on what decisions we're making, but really how those decisions are made. And we want you to get the sense of the multidisciplinary interaction and how important it is to have this group of experts come together with their thoughts and really come up with a consensus for the treatment of patients. And okay, so before we go further with the cases, these are the disclosures for the various panelists. And here's our first patient. So the patient's a 42-year-old right-handed man who presented with headaches, nausea, and vomiting. He underwent an imaging procedure. The physical examination revealed some right-sided weakness. So the pronator drifts means that with his eyes closed and his arms outstretched, the right arm starts to go down without his knowing. And he has some vision problems in the right side of his visual field, and a little bit of problems with naming. And we're told that there's no significant past medical or surgical history. So here we are, and we do have the images. And since we do not have a radiologist on the panel, which is often present during a tumor board, but we will ask our neurosurgical colleague, Dr. Q, if you would like to describe what you see on the imaging studies.
Thank you, Dr. Gilbert. Well, young patient, symptomatic for what I read right here already with some deficits, eloquent part of the brain to the best of our knowledge, even though we don't have a functional MRI, but we don't have to have a functional MRI to realize that it is eloquent in the sense that speech, auditory and picture naming is already being compromised. Eloquent because the vision is already compromised. Some of the vision pathways are going through. And then looking at the images, we have T1, what is called T1 weighted image with contrast. So clearly this patient got something intravenously. We have the second image that tells you a little bit of the swelling around the brain. And then we have the next two images that give you a little more of a picture as to what may be going on, because we have to be thinking about a differential diagnosis, all right? I think everything seems to point out that this patient was healthy otherwise, and suddenly here we are with something that is giving them trouble. So the way that I would be thinking about this is, first of all, we got to make sure that we consult with our neuro-oncologist, that we have a good workup in this patient, that there's nothing else going on and on the body, i.e. any other disease that could potentially be going up to the brain. Doesn't seem like it, but we have to double check on that there's nothing else that would make us think that this is not something that came from the brain. In this case, I think this is a tumor that comes from the brain. I suspect, given the imaging characteristics in all four panels, this is going to be some type of glioma, potentially a high grade, what we call. I'm always reluctant about giving a name, but I'm thinking to myself, let's get an our tumor board as we are right now. But I suspect that we're going to have to do brain surgery on this patient. We're going to have to try to preserve the function. We're probably going to have to do an awake craniotomy, mapping all these different pathways that I mentioned right now, and make sure that we maximize the resection. So maximum safe resection. That's how I'm thinking about this, Dr. Gilbert.
Okay, so Dr. Q, let me ask you a question that even if you knew that this patient had a primary cancer, we just heard Dr. Brastianos talk about brain metastasis. So, just to sort of summarize, you said the differential diagnosis. So the diagnostic possibilities would be either a primary brain tumor, meaning it started in the brain, or a metastatic brain cancer, meaning it started somewhere in the body, like a lung cancer. In either circumstance, so let's say you knew the patient had lung cancer, would you still consider a surgical procedure in this case, given the size and the effect that it's having?
Very good question. If it is the only lesion, and Dr. Brastianos is telling me this patient has systemic disease control, no issues. One lesion in the brain, severe amount of edema, and especially if we know that some of these metastatic tumors to the brain are not very responsive to radiation, and given the mass effect, there is definitely good data to potentially also consider a surgical resection in that case.
Okay. Dr. Bush, anything to add to Dr. Quinones' differential and discussion?
Absolutely. So I completely agree. This patient doesn't have history of anything like fever. They don't have strong restricted diffusion to suggest that maybe it was an abscess. That would be another thing that we'd always worry about if we saw contrast enhancing a lesion, tuberculosis, those kinds of things. But if, as long as he has a good systemic exam, I think we can rule most of those things out. Doesn't look like CNS lymphoma, which would be another thing that maybe we wouldn't necessarily go after with surgery based on imaging. So I completely agree, based on how symptomatic he is and the imaging characteristics, that I would strongly recommend sending him to a phenomenal surgeon and getting it taken out.
That's a great point, and I'm going to turn it back to Dr. Q and say, this patient finds out they have a brain tumor. Is this an emergency, or can this patient and many other patients actually go and find an expert brain tumor neurosurgeon like yourself?
It sounds to me, Dr. Gilbert, that the patient has some opportunity to potentially seek another opinion if they feel that either they're not sure about the expertise of the center where they are, or whether or not they have someone else that is being recommended. The exam, as far as I can tell, is stable. Of course, we got to make sure the patient is not ceasing, is not at any risk of deteriorating rapidly. Sometimes physicians do a wonderful job putting these patients in a little bit of medication to decrease the swelling, which is good, and we use steroids for that. Sometimes they put them in a little bit of medication to prevent having a seizure, especially if they're having some potential concerns about that. And that can give the patient a few days to a few weeks to really go and visit another place. Some patients are concerned, can I fly with something like this? As I usually say, if you are on some medication already, if your examination is stable, if you've been seen by a physician, most of the time they can travel and they can look for a second opinion. So it's not emergent, but I wouldn't call it elective either. I think it's somewhere in the urgent category that this needs to be dealt with sooner rather than later. I wouldn't be waiting a month or anything like that, but there are days to potentially a couple of weeks to go and potentially seek another opinion.
Outstanding. Thank you so much. So we don't usually at this point consult a neuropathologist, but we're lucky enough to have one of the top neuropathologists in the country. So Dr. Bratt, what would you ask the neurosurgeon to help you out with the job you know you will need to do after you get the tumor material?
Well, these days, thank you, Mark. I appreciate the compliments and the involvement with this crew. The most important thing is that we get tissue and sufficient tissue to make a histologic diagnosis and to break up the rest of the tissue so that we can do next generation sequencing, so we can do immunohistochemistry, so that we can incorporate cytogenetics and MGMT promoter methylation studies. At least at our institution, that's 4 different studies that require a sufficient amount of tissue. So that would be my main concern. That's if this is a high-grade glioma. If it's not, and it happened to be an infectious agent, we would need cultures, obviously. If it was CNS lymphoma, we may choose to do flow cytometry. So much of the downstream testing depends on what the disease represents. We tell that to Dr. Q at the time of operation by doing a frozen section. We get a small piece of tissue and do a fairly rapid diagnostic assessment and try to put the disease at least in a category that guides either further resection or potentially less resection in some cases like lymphoma. And it also guides what kind of testing we know we may need more immediately than the next few weeks. So that would be my guidance at the preoperative and operative period.
Excellent. So let me just ask a follow-up question. And, I think everybody who's participating with us and watching this discussion would say, oh my goodness, that looks really big. But tell us the practical reality about how much of that mass will actually be usable based on what you see on the imaging.
Yeah, that's a good question. And I think our audience can see that there is in the center of the lesion, at least on the left image, a big dark zone followed by a rim of brightness. And that central dark zone is probably all necrosis and not very usable. So it's the viable tissue that's really a part of that enhancement, and outside of it, that's going to be the most useful to do these diagnostic studies. So we would want to make sure during the frozen section as well that we get viable tissue. With an experienced neurosurgeon like Dr. Q, that would not be an issue, but in some other instances, it may be.
Right. So again, one of the goals in a tumor board if this was one of the presentations, again, at the time of discovery of the mass, there would be potential dialogue between the neuropathology colleague and neurosurgical team saying, we really have to be careful. A lot of this is probably not going to be very useful for diagnosis. So I think a really important point that even before diagnosis is made, there can be communication that would be instrumental in optimizing care. Dr. Bush, anything to add before we move on to the next slide?
I think we can move on.
Okay. All right. So, whoops, I apologize. I must have hit it twice. There we go. All right, so they were successful in removing 98% of the disease, and we're very fortunate that somebody circled the remaining tumor, and likely that it was considered to be in an area that would be dangerous and causing dysfunction if they removed that area. So eloquent brain, as Dr. Q had mentioned before. So that's a very, very good resection. We're going to now ask Dr. Bratt to again describe what the pathology report says.
Okay, so the diagnosis is glioblastoma. That is a high-grade primary astrocytic neoplasm, and it's grade 4 based on the criteria either seen through the microscope of necrosis and microvascular hyperplasia, or some of the genetic alterations, as we talked about earlier today, that are seen in the molecular report: CDKN2A and B homozygous deletion, EGFR amplification, P10 mutation, TERT promoter mutation, gain of 7, loss of 10. Those are classic genetic alterations for a glioblastoma. So we incorporate that genetic information, especially the fact that it does not have an IDH mutation, into the final diagnosis of glioblastoma, WHO grade 4 IDH wild type. We also do MGMT promoter methylation studies to find out whether or not the MGMT enzyme is active. If it is methylated, the tumor would be more responsive to chemotherapies. In this case, it was unmethylated. So we do that histologic exam, we do the molecular studies, and we do MGMT promoter methylation studies whenever the tumor is a GBM, and incorporate all of that into a report.
Great, thank you. So Dr. Bindra, the patient, we now have a diagnosis, and we have a great resection, and radiation is obviously the cornerstone of treatment of glioblastoma. So can you describe the thought process as a radiation oncologist about what type of radiation, what the fields would be, and how would you counsel the patient, and what recommendations would you make to us, the rest of the tumor board panel, in the context of this case?
Oh, thank you so much. Can you hear me okay, Mark?
We can. Mm-hmm.
Great. Great. Well, the first thing we like to think about here is the timing of what we call the simulation for radiation. And that's really sort of a dress rehearsal for the treatment that we're going to give. And the timing is important because the treatment itself involves a team of dosimetrists and physicists beyond us. We outline the area that needs to be treated, but there's a lot of complex dosimetry to get all the beam angles right and make sure we treat the areas that we want and preserve the areas that we don't want exposed. And that often can take 10 to 14 days. We typically like to start radiation about 1 month after the surgery. A lot of folks have asked us in clinic, could we start earlier? How much later? And interestingly, there have been studies that have suggested that starting too early, surprisingly, can have a detrimental effect. And that's because just the way the microenvironment of that resection cavity evolves can actually impair the ability of radiation to have the effect that it's intended. Similarly, waiting too long can also have an adverse effect on the treatment outcomes, more like 6 or 8 weeks. And so with that sort of time gap in mind, we always like to sort of, even looking at the image here, you can see there's a little bit of swelling in the back side of the patient. And we'd like to see if they're going to lay on a table for a CAT scan and for a treatment planning scan, if that's going to be okay for us to begin our simulation. For the radiation, as many of you know, there are different ways to give radiation. In the old days, it used to be a parallel opposed field, so one field this way and one field that way. And then it evolved into 3D conformal, which just really means 3 or 4 beams kind of encircling that area. We've now moved to something that many of you have probably heard the term of IMRT, or intensity modulated radiation therapy. And that's really just a fancy phrase for sculpting the radiation dose to really precisely hit the areas that we want to treat. The final modality that some of you may be aware of are protons. And protons have been very, very active area of interest because the actual radiation itself sort of stops right wherever we direct it. There's no what we call exit dose, so minimal exposure to the rest of the tissue. We use that primarily in children when we're worried about what we call late effects during the child's development if we're treating the whole brain and the spine. In the case of an adult here in the location, relatively little benefit for the use of protons, still technically an ongoing question, and some would argue that you could consider that in the context of the clinical trial. And I'll stop there for now.
Okay, well, let me ask a follow-up question, and thank you for that very eloquent discussion of the types of radiation and sort of the molding of the radiation field. But somebody would potentially look at the scans here and the addition of the nice red circle and say, boy, that's a pretty small field. Is that all the radiation the only area that the patient's going to get? If it's so small, I've heard something about radiosurgery. I mean, what is the actual plan, and how does one go about the radiation? How much radiation should somebody get? How do you guys decide all of that?
Yeah, wonderful question, and thank you for that. What we now understand is that even if you get the best surgical resection, and Dr. Q obviously did a wonderful procedure here, we know from studies over the last 40 years that within about 1 to 2 centimeters of the resection cavity, even if that red circle wasn't there per se, we tend to have recurrences in that area, and we believe it's microscopic cells that you can't even see under the MRI, which is often hard to believe. And we know from studies over 40 years ago that you could resect a much greater area and still have a problem of tumor cells right at that margin. So for those reasons, when we look at the T1 post-contrast scan, which we're looking at here on the left, we tend to cover, or almost always will cover, the entire resection cavity. And we draw a little extra margin of about 1 centimeter, depends what institution you're at, and certainly also an active area of investigation. And we put that little centimeter extra to make sure that those areas where we think it could recur are covered adequately. On the right there, you can see as well, we consider both a multitude of different imaging studies, and sometimes we'll treat a larger area with a smaller dose and then do something we call a cone down, which is then shrink down just maybe to that red area, as well as just the actual resection cavity. We like to go to 60 gray. The standard dose is 46 gray to a larger field, and then we bring it down to a total dose of 60 gray, which has really been based on an elegant series of studies of the last 20 or 30 years, starting from 1970 up into the early 2000s, that have really guided us to believe that this is the optimal dose that we should use for patients. So that ends up being about 6 weeks. We treat Monday to Friday. We give our patients and the dosimetry team and a break on the weekends, and it can be a rather lengthy process, but with these new modern technologies, we do believe we've mitigated a lot of the toxicity from the older days by being able to focus really on that resection cavity and whatnot. And the last question, Dr. Gilbert, was a wonderful question of, do we boost or add additional dose to that enhancing area? And a number of studies have actually tested that, whether we should actually add additional dose above the 60 gray, either with radiosurgery pinpoint dose to that area. And even though it sounds like a good idea, what we found is it doesn't really change the stability or the persistence of the effect of the radiation, but it increases the toxicity and the damage that we do to the normal tissue. So we tend to not boost this maybe in the recurrent setting. We'll definitely come back to that.
Great. Thank you so much. Dr. Bush, you have this patient, your radiation oncologist has just told you that he wants to give 6 weeks of radiation. He's described to the tumor board what the field's going to be. What additional recommendations for treatment would you make at this point?
Sure. So when I look at this patient, like we just kind of talked about, this is a very classic looking glioblastoma. So typically the standard therapy, in addition to the 6 weeks of radiation, would be daily temozolomide given at a low dose. It's a pill, like many of you know. For Dr. Bindra, he gives them the weekends off, only 5 days a week. But for the chemotherapy pills, you got to take them through the weekend. So they get a full 6 weeks of chemotherapy. Then typically we give about a month break, and then we proceed with more chemotherapy at a higher dose, usually double the dose, given in a different way, 5 days in a row every 28 days, so in 28-day cycles, and constantly monitor with MRI. So a few other little things to think about for this patient. He has MGMT unmethylation, and Dr. Bratt touched on this for sure, but I like to describe it to patients as MGMT is a DNA repair gene. It fixes broken DNA. Our chemotherapy works by breaking DNA. So when MGMT is active, it comes along and fixes a lot of the damage that our chemotherapy makes. And MGMT is active in its unmethylated state. It's inactive in its methylated state. So patients who have MGMT methylation tend to have a better response to the chemotherapy with Temodar. That being said, there always is a proportion of patients that do have a response to the temozolomide, even if they're unmethylated. And I am not the expert, but I think the truth is that we truly don't quite understand MGMT methylation. If it's the extent of methylation, a pattern of methylation, certain methylation sites that really confer that benefit. So for the vast majority of patients, even if they're unmethylated, we'll still give them standard treatment with the temozolomide. And then this patient also was on a clinical trial. So I don't know if you want me to comment on that now or wait. Okay, clinical trial. So like everybody's been kind of talking about, so these tumors are really tricky to treat. And even when the surgeon cuts out everything we can see, like Dr. Bindra said, there are these microscopic cells left in the normal brain. So because of that, we cannot cure this. The goal is truly to put it into hibernation for as long as possible. And we know that right now our best treatments are radiation and chemotherapy. But that also means that we're constantly looking for better treatments. And so if there's a possibility for a patient to go on a clinical trial, it's something that I always try to discuss with my patients. And this particular patient did have the opportunity to do a clinical trial. It was a phase 2. And for him, this clinical trial was a 50-50 shot. Either you got standard of care therapy or you had this addition to radiation and temozolomide of a compound called BMX, which is injected into your leg twice a week. And the goal of this particular compound was to potentiate the effects of radiation and actually also protect the normal brain, because it worked on reactive oxygen species. And so this is a clinical trial that was done through radiation and was in addition to standard of care. So I like to tell patients, this is a 50-50 chance of you getting something extra on top of what everybody else gets. And it's usually an opportunity if there's not too much toxicity. So it's definitely something that we would recommend. And then just in general for clinical trials, I always tell them they're always voluntary. You can stop at any time for any reason. Even if you just don't want to come back and see us, you just got to let us know so it can be safe. We never keep someone on something that's not safe for them. So if their labs go strange or they're not tolerating it, we'd always come down off the medication or stop if we needed to. And we never keep people on experimental medications that aren't working. So I think in general, clinical trials are really great for all of our patients because it gives them an opportunity to have potentially more or different treatments that could work better. And we do try to do it in the best and safest way for our patients.
That's fantastic. Thank you so much. It's really such an important part. And you know, again, the discussion in the context of a tumor board are there available trials, and would a particular patient be a candidate for one or actually several of the trials? And again, for our participants today, recognize that the clinical trials are scientifically reviewed. They're reviewed by institutional review boards, and we don't know the answer. So we're often asked, which is the best? And if we knew that, then we'd know that treatment worked and it wouldn't be a clinical trial, it would be part of the standard of care. And conversely, if we knew something wasn't going to work, we certainly wouldn't subject people to that treatment. So really, really great discussion of the clinical trials and importance. So this patient was on a clinical trial. And let's see if we can get to the next slide. There we are. So the patient did complete 6 cycles of the post-radiation temozolomide and also used the Optune device, which we can talk about in a little detail. And there we can see the imaging studies. And Dr. Bush, do you want to talk about?
Sure. I'm sorry, I forgot to mention that. So with the adjuvant temozolomide, another FDA-approved option for treating brain tumors is called Tumor Treating Fields. In the United States, it's through a company called Optune from Novocure. And what it is a totally different way of treating brain tumors. It is actually a device. They're electrodes that you wear on your head. So it uses electromagnetic waves to basically disrupt how tumor cells divide. So it is a little bit of a tricky treatment. So it doesn't cause side effects like nausea or fatigue, you know, those kinds of things. It doesn't drop your blood counts like a chemo, but you have to shave your head. And then you wear 4 pads of electrodes on your head and you place them in a way to target directly to your tumor. And if you wear this at least 18 hours a day, studies have shown that it did also improve overall survival in combination with temozolomide for patients with newly diagnosed glioblastoma. So this is always an option that I bring up with all of my patients, usually after radiation is over, because it's a lot to think about in the beginning. And it's added to that second phase of treatment where you do the chemotherapy in the cycles. So this patient elected to do that in addition to his chemotherapy.
Great, thank you for that. And Dr. Bindra, we have all of these scans post-radiation, 2 months post-radiation, 5 months post-radiation. Tell us what you consider to be the optimal observation of the patients, and what would you say about these 5 scans?
Yeah, these scans. Oops, okay. Yeah, these scans look quite nice. We typically tell our patients that we really want to continuously monitor every eight weeks or so, up to sometimes stretching out to 12 weeks for really one of 2 things. One is a possible recurrence, obviously, but also something that you'll hear folks say, which is sort of, we'll start as radiation change, which can be radiation necrosis, which can be a little bit of toxicity to the normal tissue, but also something called pseudoprogression. And sometimes that's actually a good thing to some degree, because it's almost like a reactive response of the brain parenchyma, or that area of the brain tissue, sort of a blossoming on the MRI scan. And typically a patient like that will come in and look quite good and feel quite good in their scan 2 months after radiation. They show a paradoxical increase in any of the enhancement that was there before the radiation started. If the patient looks good, we'll often continue to treat with the temozolomide in this post-radiation period and get another scan, and often we'll see that that can actually resolve and was what we call a pseudo-progression, meaning not a real progression. But it's important to get these scans every 8 weeks or so, because we often find that radiation toxicity, either the pseudoprogression or the actual necrosis or radiation damage, can occur even as late as 12 months, if not more, in some cases. And so we have to be actively looking at this in our patients. And so looking at these scans, very nice. You can see that little rim of enhancement or post-surgical change seems to have resolved at the 5-month spot. And also we don't see any other spots that are outside of what we think the radiation field is. So for example, in the 5-month scan, if we saw something that was way, way up top, where we can really feel that this is not in the radiation field, we do get worried about possibly some distant recurrence, which as our team here has mentioned, that this is often a problem and impairs our ability to cure disease. And so what we'll often do is take that MRI at the 5 months and we'll fuse it with the original MRI that we use for the radiation planning scan, and we can overlay the dose painting, the areas that were treated, and we can say, well, this area didn't get any radiation, so it would be hard to call it radiation change. And that is very helpful for us to kind of assess what's going on at that time point.
Great, thank you so much. So we'll continue on. Okay, so 9 months after the radiation, we do see some changes. And I'm going to ask Dr. Q if you would take a look at this, because based on what you think it is, the question always is, do we just watch it? Do we take it out? Are there studies we can do to try to figure out what these changes are?
Well, thank you, Dr. Gilbert. I think that in these particular cases, 9 months later, as Dr. Binder was saying, we're getting the scans regularly, following the patients, and it's clear that at this point, there's something that is changed slightly, a little bit of a mass effect there. There's a little bit of what is called flare, that's a swell in the fluid, attenuation, inversion, recovery, signal normality. And there's also, as I mentioned, the enhancement that is been increased. And the perfusion studies also is showing some changes that could potentially be concerning for tumor progression. We, despite of our best images that we have, we don't know 100% of the time either way. At this point, we are all getting together, and I saw some of the questions. How can one brain tumor board say one thing and another one with equally qualified said another thing? Is the reality is this is the art of what we do, is our interactions with each other. In this particular case, I would say that I would be more concerned that this is indeed progression, not just pseudo progression, that it is something that we should potentially consider either biopsy, and that's another point that we can talk about, or resect. I think that it's going to depend on the experience of the team. It's not just the surgeon. The surgeon goes into the operating room with sometimes a team of 20 people, all the way from the neuroradiologist that gets special imaging to the neuropathologist being available to get the diagnosis in the OR, to the neuropsychologist, neuroanesthesiologist, everybody, epileptologist that can also sometimes be part of the team. This particular case, I think, is going to merit an exploration, Dr. Gilbert. That's my suspicion at this point.
So your recommendation would be, with these changes, given that everything looked so good before, to go in, take it out, and figure out what it is definitively.
In another awake craniotomy, the patients are amazing. I think that we'll probably have a good functional MRI this time. The neuroradiologists give us some beautiful images with all the cables that are connecting the brain, so we know where those cables are located. It's called diffusion tensor imaging, and we get the functional MRI. We keep the patient awake, safe, and we can give the patient a diagnosis. So that way, our team of neuro-oncologists, radiation oncologists can decide what we're going to do next.
Perfect. Okay. So the patient underwent surgery. That area was removed. So all of the abnormal area was safely removed, again, awake. language mapping, just as you said, Dr. Q. And now we're going to call on Dr. Bratt, because here is what the pathology looked like. And if you would describe for everybody what you see.
Sure, thank you. What we see is an H&E stained slide. That's our typical tissue stains. And we see a combination of viable tumor, which is the area with the purple dots that are easily recognized, and then areas of necrosis, which are areas where they're just kind of a mixture of pink and purple, and you can't see individual cells anymore. So this is a low magnification that shows indeed there's going to be a combination of tumor necrosis and viable tumor, and it would be grade 4. In this case, we would certainly review the previous diagnosis, but because in some cases there's such extensive dead tissue that we can't really make a diagnosis of what type of tumor. So we would review the previous pathology, determine what the mutational spectrum was, and then report out that this is recurrent high-grade glioma, which implies that there's viable tumor, and there's also extensive necrosis, which in a certain time period may imply that the therapy actually did the job it was supposed to do and killed the tumor, if you will. So I think there's a next slide. And this just shows I think is a better example of recurrent active tumor cells on the left here, right around that blood vessel that has the red blood cells in it in the center. So that's an important distinction, because at the time of recurrence, there's been surgical resection prior, there's been radiation therapy, there's been chemotherapy, and all of these things are toxic to tissue. And in some recurrences, it does not appear that the tumor cells are still proliferating, still active. In this case, it is. We can judge that by mitotic activity, the feature of cell division that we can see morphologically, or we can do a proliferation marker, or the type of necrosis that is seen seems to indicate that it's tumor cell growing that's associated with the death rather than the radiation. There is a lot of therapy effect in this case, though. There's inflammatory cells that you see on the right-hand side. There's fibrosis. There's thickening of the blood vessel walls. All of those are associated with the therapy and not with the tumor growth. So this is a good example of viable high-grade glioma and necrosis. Now, the question is, should we be doing additional molecular studies? And Mark, do you want to introduce this?
Sure. So I wanted to say that molecular studies were done. I wanted to point out, as you very eloquently described, the mixed appearance of the tissue, and that sometimes the molecular analysis of the recurrent tumor can look quite different. So if you would give us sort of a summary, and also Dr. Bratt, also discuss the conundrum that you face sometimes in trying to determine whether the tumor cells that you're seeing in a specimen from a patient who's had treatment, whether they're residual or recurrent. And we know that sometimes our patients hear us say, well, we saw some tumor cells, but we really couldn't tell if they were just dormant or they were active. And how do we go about approaching that conundrum?
Great, I'll do my best. So the spectrum of mutations that are listed in this table, first of all, are similar but not identical to what was present in the first case. The gain of 7, loss of 10, the CDKN2A, the EGFR amplification, the P10 mutation, and the TERT promoter mutation were all there before, which would be expected. This is the same tumor, it's just grown and progressed and had therapy. But there are 2 additional mutations, neurofibromatosis 1 and NIPBL as well. So we do these next generation sequencing panels at recurrence at the discretion of the clinical team if they think it would be helpful in treatment. On occasion, we can see a targetable genetic alteration emerge at the time of recurrence that wasn't present in the primary tumor. That's one possibility. It's not that common, but sometimes it does occur. And then there's also the subset of these high-grade gliomas after therapy that become hypermutant. They have a high mutational burden, 10,000 or 10 over 10 per thousand mutations. And that, at least in some types of cancers, is an indicator of response to therapy, response to immunotherapy. It's not my area of expertise. I'd have to ask Nancy or Mark to comment on that, but sometimes we do see a hypermutation pattern at the time of recurrence. The other question that Mark was alluding to was, how do we tell residual from true recurrent tumor cells that we see under the microscope? And that's a very subjective call. Some of the things that add into it are the number or density of tumor cells that are present. Almost every recurrence will have scattered neoplastic cells just by the nature of glioblastoma. There's going to be residual infiltrating cells. The therapies are not 100% effective. So there's going to be residual. Is there recurrence is the main question. And that is subjective, depends on the cell density, the proliferation, if we can see mitotic activity, if we can see a high proliferation index based on MIB1 or proliferation studies, if there's the type of necrosis, the pseudopalisading necrosis that goes along with tumor progression rather than therapy effect, all those things add in, and we do our best to comment on that. We have a lot to learn about the neuropathology of recurrent high-grade gliomas, for sure.
Great, thank you so much. So I'll give Dr. Bush the last, well, actually, Dr. Bush and Dr. Bindra. So Nancy, if you would comment on kind of what your treatment approach would be here, and then Dr. Bindra, what is there a role for radiation again?
Thanks. These are really complicated cases, and this is a lot of what we talk about at Tumor Board. When you see these combinations of treatment effects and tumor, what's your threshold to think that this is really tumor growing back versus just treatment-related changes that the treatments were working? And so this is when we have these kind of spirited discussions with the pathologists, and like, well, really, how many mitosis did you see? Like, what do you see there? And it's really a fun part of Tumor Board. For this particular patient, there was 40% viable tumor. That's kind of past my personal threshold. I use about 25% viable tumor. There's no real data behind that, but that's kind of where I am for thinking, okay, maybe it's time that I put them on treatment again, and potentially the same treatment, a different treatment, because there really is a lot of viable tumor. There's cells growing there, and we have to try to target them. And so the treatment decisions are varied. So if it had been a long time since the patient had been on temozolomide, that certainly could be an option to re-challenge the patient with temozolomide. I wouldn't do it if the patient had hypermutation, like Dr. Bratt said. So that's when the tumor acquires hundreds and hundreds of mutations, and they're really no longer sensitive to temozolomide at that point. So that would be one option. Another option would be another standard of care chemotherapy, something like lomustine or CCNU, which is an oral chemotherapy given once every 6 weeks. It's a bit stronger than Temodar. It causes a little bit more myelosuppression, but in general, really well tolerated. We get MRIs every 6 weeks on that regimen. Sometimes with that regimen, we actually introduce a second agent called bevacizumab, which is a vascular endothelial growth factor receptor antibody to plug up the leaky blood vessels. There's some data that they work together. The CCNU and the bevacizumab can work together. Or we talk about, are there clinical trials or potential off-label targeted therapies that we can add to the mix? So for this particular patient, I would look and see if there were any exciting clinical trials that he might be eligible for that we could put him on. And that would be my first choice if that's possible, because I know that I have the FDA-approved agents kind of in my back pocket. If we didn't have a clinical trial option, I would think about probably lomustine, because it didn't seem like the timing had been, it seemed like it was pretty close to stopping Temodar that he regrew. So I don't know if that would be the best choice for him, and he's unmethylated. So potentially CC-Mu is an option. If he was wearing Optune, I'm not sure I would continue it at this point, is what I would say. And then theoretically, you could think about some targeted agents, either in addition to the CC-Mu, like ablasiclamib to target his CDK mutation, afatinib to target the EGFR amplification. So there are things that potentially you could add as off-label targets as well. And then I'll let Dr. Bindra comment on whether or not it makes sense to do radiation now or hold off for the future.
Yeah, thank you. That was wonderful. I think, our field is often limited because we have sort of one hammer that, some folks in our field want to, bang as much as they can. And re-irradiation at the surface has always kind of made sense. It worked in the beginning, and we can give that radiation dose again. What we've struggled with in our field is that the clinical trials haven't supported a survival benefit when we've tried to add re-irradiation, despite the sort of appealing nature of it. When we do feel that there aren't any other options, we certainly will offer it in a subset of patients. And the way that we think about is the location of the recurrence. If it's in the center of the brain and we're not doing a proton therapy where we can precisely deliver dose without exposing the rest of the brain, we tend to get worried. Or if it's a large field where we think there's going to be a lot of radiation dose to that patient, and the larger dose or higher volume it often correlates with more toxicity. As a physician scientist myself, I spend a lot of time developing novel agents to combine with chemotherapy or with radiation. And what I like to tell patients is that technically we only have a second chance with the radiation. So you get it in the beginning. We typically can give you one more course. Now, in the last 10 years, I've probably treated patients maybe a third time, and some very, very, like that red circle that we saw in the very beginning diagnosis, a little dot that maybe a third radiation treatment could be indicated. But even that hasn't really shown a survival benefit. And so I often tell patients, if we can, as Nancy very astutely mentioned, if we can hold that as the eternal optimist, that there may be a clinical trial with a PARP inhibitor or a DNA repair inhibitor that could be combined with radiation, then that might actually have a better effect. And so maybe we could hold that in our back pocket for later.
Great. Thank you both. That's a fantastic discussion. So we're going to go ahead, and we don't have much time, but I think there are a couple of important points to talk about with the second case. And so the second case, very importantly, this patient aged 90 presented with some weakness in the right leg and foot, which everybody knows is because the abnormalities on the left side of the brain. And an MRI was obtained because she had a fall. And so that, I think everybody was quite surprised to see a large mass like this. And the right lower extremity weakness was noted on examination. So in the interest of time, I think everybody can appreciate there's a very large tumor mass. And rather than go through a lot of detail, we know that this is a long-standing mass, because if this was rapidly growing, the patient would have come to clinical attention much sooner, would have been probably close to paralyzed on the right side, but yet with a slow growing tumor, the brain can accommodate quite well. So we'll go on to the next slide. A previous MRI in 2016 didn't show anything, but you can certainly envision that this growing over 2 to 3 years would be quite possible. Okay, so I wanted to get to this discussion and ask Dr. Q what his differential diagnosis would be and what he would consider. Again, the importance of this is the context, right? So I think just looking at the image, your first impression would be, oh, it's got to come out, but let's discuss this in context.
Absolutely. And that's what would happen. I mean, she's 90, but she's highly functional. You saw there was nothing in 2016. I'm leaning towards this being an extraaxial mass, potentially, hopefully not cancerous in nature, but one that is giving the patient already symptoms. The question is how fast it's growing. So I think that at this point, I would have a discussion with the patient and the family. What are the goals potentially that we want to achieve? Make sure that everything else is normal. And my understanding from the oncologist, a neuro-oncologist had already seen this patient, is that everything was clean, no evidence of any metastatic disease or anything like that. So I think it's reasonable to potentially tell the patient, maybe we should give it one more imaging to see how this is moving or not moving. Right now, I think that I'm afraid that if I do surgery, I may make you worse potentially. So, and here we are. We got an MRI just 3 months later. It had already grown 20% on volume. So at this point, the patient is probably more symptomatic. And I do agree, Dr. Gilbert, I think it's going to have to come out. I mean, especially if it's a highly functional 90-year-old patient. It sounds tough to say brain surgery, but we have patients that are living a lot longer and a very good health.
Dr. Bush, any reason to contradict that?
I think it really just depends on the goals of care of the patient at that point in life. At 90, if they would like to go through a surgery and a rehab, I think with a very experienced surgeon like Dr. Q, probably the surgery is going to go really well. But it is really a discussion to have with the patient, because this will likely continue to grow slowly, but that might be okay in a 90-year-old. So I think you can go either way, but knowing that it's really the patient's has to be part of that conversation.
And Dr. Bindra, any role to avoid surgery and do radiation?
You know, when we think about the indications, this is an example of tumor size. And I think that a larger lesion like this would be a large radiation field, and an elderly woman would give me a little bit of pause. I could imagine being less excited about it.
Okay, great. So I think the patient underwent surgery, and so that's just showing the growth. And we talked about the surgical intervention. We'll move on to the next slide. Underwent surgery, tumor resection, and we'll give Dr. Bratt the last word about talking about what they found, and we'll have a quick discussion about the tumor type and management. Dr. Bratt?
Yes, so on the left hand, we're seeing an H&E slide that shows a very hypercellular neoplasm with a central area of necrosis, which generally indicates rapid growth. In this case, it wasn't clear that it was a meningioma because it was so high grade in appearance. So we used our best immunohistochemical stain, which is somatostatin receptor 2A, SSTR2A, and that was very positive. That's a fairly specific stain for meningothelial differentiation or meningioma. But this to us at low magnification looked high grade, and then at high magnification we see these mitotic figures, which indicates cell proliferation, and there's many of them. We also saw local invasion of the brain by tumor cell nodules. So the combination of these two indicated that it was a high grade meningioma, somewhere between 2 and 3. So we decided to do molecular and cytogenetic studies, which are very useful in borderline cases. And what this showed was that there was actually a TERT promoter mutation, as well as loss of 1P, 22Q, and CDKN2AB. The TERT promoter mutation and the loss of CDKN2AB now fulfill criteria for WHO grade 3 meningioma. So the diagnosis was anaplastic meningioma, WHO grade 3.
Great, thank you. And so this is post-op MRI. It's a beautiful resection. I hope everybody can appreciate what a wonderful operation it was. And again, in the context of a person who's 90 years old, do you just observe or do you do treatment? And we unfortunately don't have time to have an extensive discussion, but I think the point here is that our decision-making is not just based on the type of tumor or the grade of tumor. It's always done in the context of the patient's goals, et cetera, and it's taking into all those into context. Obviously, the risk of treatment chemotherapy or radiation in somebody who's 90 is greater than somebody who's much younger. And I think Dr. Bush alluded to that. And I know that Dr. Bindra would have the same considerations for the treatment. So I think we'll conclude the formal part of the tumor board now and then go to a question and answer. But I want to thank the panelists for just a fantastic discussion and look forward to questions from the folks who've been listening in.
Thank you so much for that wonderful discussion. We do have time for a few questions. As a reminder, if you do have a question that you'd like to ask, please type and submit it using the Q&A button on the bottom of your screen. All right, so one of the first questions we have here is in regards to getting a second opinion. Why sometimes when a patient receives a second opinion, can two different well-regarded brain tumor boards come up with vastly different recommendations? How does that happen? And how can the patient, or what advice can you give to the patient to make an informed decision on what route to go with?
That's a great question. I think, Dr. Bush, you alluded to this earlier, so would you like to expand and expound on that question?
Absolutely. So part of it, it's the art of medicine, and that we don't have all the answers. If we did, we would all recommend the same kinds of treatments. So I always recommend second opinions. I think it's great to get other opinions about what's going on and what treatment makes sense. And sometimes other institutions will have clinical trials that you might be eligible for. So I think that's great. I would say that usually your primary oncologist knows you best. And that really is important when you're talking about treatment options, because I might be across the country and I might say, oh, you could do this and this and this and this, but I may not know your functional status, your goals in life if I just meeting you once from across the country. So I do feel like it's important that you have a really good relationship with your primary oncologist. That being said, some of this is art.
Dr. Bindra, anything to add to that?
Yeah, I think I always the analogy that you can get a really good estimate from a really reputable vendor for something, and often you can get an estimate from someone else that's a little bit different, and it's often affected by the culture, the traditions, the types of expertise even within our subspace, because it's remarkable how even within GBM there's the IDH wild type people and IDH mutant people. It's a very, very much a complex space. So we do see this quite often, and I really like the way Nancy said this. Your primary doctor knows best, and often it's the best to be the quarterback for that.
And Dr. Q, from the surgical standpoint, I know sometimes you guys don't always agree.
That's true, and I would say that we're respectful of each other. I think that is like anything. There's an art, there's a confidence, there's a experience, and also the team that you may have. And sometimes your experience may be influenced by the last case that you did, and it may make you reluctant. And that's why the patients, I always tell them, they have the right to look for several opinions. And we know each other. We send each other, we ask each other questions. I think these tumor boards, many times we interact with each other, and sometimes respectfully we disagree with each other.
And Dr. Bratt, I don't know that a lot of folks realize that the pathologists communicate and help each other out and send complicated situations, cases to each other. Do you want to expand on how that happens?
Yes, we do. And sometimes that's for additional molecular analysis. Sometimes that's for just consultation on a very challenging case. I have to say that during my career, we've gone from having very high levels of irreproducibility, high inter-observer variability, to a place where we are now more protocol driven, and there's more uniformity to the way we diagnose diseases, at least at the large academic medical centers. But certainly we receive as many consultative cases at our department as primary diagnostic cases. So there is a flow of cases for second opinions in pathology for sure. One good thing is that patients who go from center to center to center used to receive 2 and 3 different diagnoses, which was extremely challenging for the neuro-oncology team. Now with the incorporation of molecular studies into the diagnosis, that has gone down dramatically.
Great, thank you.
Thank you all so much for your insight. Another question we have here is asking, is the patient allowed to sit in on the tumor board, or do they ever get access to the discussion notes from the tumor board?
I can answer for the NIH perspective. We don't invite the patients. We don't want the discussion to be at all altered because of the patient's presence. There is often a short description of sort of a summary or consensus. And I think almost all of us, if not all of us, will inform the patient and family, what the tumor board consensus was, and sometimes kind of where the areas of disagreement, just so they have an appreciation that, it's not as cut and dried as sometimes people think, and that there is an art, as my colleagues have indicated. But we'll go around the room, as it were, and this time we'll start with Dr. Bratt.
Was the question about including patients?
Yep, including patients, or do the patients have access to a transcript or some written documentation?
We do not have patients at our tumor board. The tumor board is thoughtful and inclusive as they are. They do move along fairly rapidly, and we'll usually have 15 patients to present within an hour and a half. And I think there's a better time and place for involvement of the patient and their family for decision making. The tumor board is, I think we prolonged the discussion for educational purposes today, but it does have a pace and a tempo to it that might not be benefited by patient involvement.
Great, thank you. Dr. Q?
The way that we normally like to do it is we give a summary to the patient, and at the end, sometimes we'll be honest with the patient. We presented it, there was a mixture of opinions. Some physicians, some nurses, some team members thought that we should do this, some thought that we should do this other thing, and at the end, our consensus was to do this right here. And we want to make sure that they understand a little bit more, and then they are involved in that decision making. I agree with Dr. Bratt. At that moment, with either the neuro-oncologist, the radiation oncologist, or me as a neurosurgeon, then we go ahead and reconvene and making those decisions with the patient, because at the end, they are the ones who should feel empowered to make those decisions.
Great, thank you. Dr. Bindra.
Yeah, and I think what we found is, we often have a note that, as I said earlier, that summarizes the outcome of and what the decision, and often that's even more effective just to give a high-level summary, and someone from our team will call the patient and communicate that.
Great, and Dr. Bush?
Similar, we don't have patients join our tumor board. I always call my patients after tumor board, and if it's more a long-term discussion, we'll have a visit to discuss kind of what the tumor board spoke about, and it's always documented in the chart.
Great, thank you all.
Thank you all. I think we might have time for one more question. This question is asking, how often does a tumor board meet for a specific patient? Is it just during the initial diagnosis? Is this an ongoing thing where the tumor board will meet again halfway through treatment or after?
Well, we'll reverse the order. Dr. Bush, what do you guys do at UCSF?
Great. So all newly diagnosed patients and every surgical resection is always automatically added to tumor board, so we can discuss the pathology with the pathologist. And then we have the ability to add cases anytime we want. So if I have a question, or I disagree with the radiologist, or there's an outside case, we can add it to tumor board. Or if a patient were to request to me, I'd really like my case presented at tumor board this week, then we would do that. And our particular tumor board meets weekly.
Okay, Dr. Bindra.
Yeah, we, almost every new case, and we have all the other folks I'm sure have a molecular tumor board at the very end where we parse out some specific cases of sequencing, and we always, can do last minute add-ons and whatnot.
Great, thank you. Dr. Q?
Exactly the same, nothing else to add right here, Dr. Gilbert.
Okay, Dr. Bratt.
Same here, all primary disease, and then oftentimes at radiographic or symptomatic progression, and we have a separate tumor board for metastatic disease.
Excellent.
Thank you. And we do just have a little bit more time, so I think we can squeeze in one last question. This question is asking, is there a member on the tumor board who is the ultimate decision maker, or is it a majority decision? Does everyone come to a singular consensus? How does that work?
That's a great question. So this time I'm going to mix it up a little bit. Dr. Q.
I got so excited. At the end of the day, the patient will always make the ultimate decision, period.
Yeah.
Okay. Dr. Bush.
I agree, it's the patient, but at the particular tumor board, there's not one person. It's really fun. There's debate. I think maybe the primary neuro-oncologist might be the one who kind of collects everything and makes the consensus, but there are always different opinions.
All right, Dr. Bratt.
I can just add that it's never the neuropathologist that's making the decision, but from what I've seen, it's the person who brings the case to tumor board, at least at the time of recurrence, or asking if there's additional therapies to consider. There's often a lively debate, respectful, that then I think that individual distills down and makes a decision to bring to the patient and have a discussion.
Great, thank you. Dr. Bindra.
Yeah, similar. I think if it's a primary surgical case where there's actually a resectability question, obviously it's the neurosurgeon, and if it's chemo rads, it's often really goes to the quarterback, which is the neuro-oncologist in the post-op setting. So very similar with Nancy, with some debate, some friendly debate as always.
All right, well, that is all the time that we have for questions for this session. Thank you all so much, and I'll turn it back to Emily.
Yes, thank you so much. Thank you so much to our whole brain tumor board panel. This session continues to prove to be one of the most meaningful to all of our attendees to understand how treatment decisions are made. We wish we had more time to answer all of the questions live. However, panelists, if you have the time, please type in a response to some of the unanswered questions in the Q&A box. Thank you for each of you for contributing to this very engaging tumor board, and thank you, Dr. Gilbert, for facilitating. For those of you who need a break, please take this opportunity to do so. We invite the rest of you to stay on to hear a research update. We will return for the rest of our program in just a few minutes to hear from our keynote speaker, Dr. Courtney Burnett. Thank you so much.
My name is Miriam Rahman. I'm one of the neurosurgeons at the University of Florida. I was a recipient of a 2019 Discovery Grant that was supported by the Southeastern Brain Tumor Foundation for a project called A Vaccine Strategy to Overcome Treatment-Induced Immunosuppression in Glioma. And I'd like to share our work with you today. We're going to talk about the life of a T cell. T cells are the immune cells in the body that kill foreign invaders. And they are normally calm, circulating in the blood. But when a foreign invader like a virus comes, there's a series of events that take place that then activate that T cell that allows the T cell to then kill the foreign invader. An important part of this process is the dendritic cell. The dendritic cell, they like to eat. They go around eating foreign material, and when they come across something like a virus or a tumor cell, they run to the T cell, they present that abnormality to the T cell, and the T cell is then activated against the virus or tumor. That usually works very well. The exception to that is tumors, and brain tumors in particular, and the T cells become activated, but when they encounter the tumor cell, they cannot kill the tumor cell. The tumor cell has many mechanisms that it uses to shut T cells off and prevent the T cells from killing them. And that results, as the T cell tries and tries and tries to kill the tumor cell and cannot, in T cell exhaustion. And anyone who has kids knows what that feels like when you try to get something done and you can't get them to listen, that exhaustion feeling. Our project was really focused on looking at T cells that are exhausted due to exposure to temozolomide, which is the most common chemotherapy that we use in gliomas. So we took T cells, we exposed them to temozolomide, and then we looked at the ability of those T cells to expand or proliferate and their function, including exhaustion. We then took temozolomide, took cells that had been exposed to temozolomide, added dendritic cells as a vaccine that we deliver intradermally, and looked to see if we could actually improve the function of those cells or result in better expansion. We found that the T cell expansion was excellent, but there was no significant improvement in function of these T cells, and they were not less exhausted when we gave the vaccine. So we needed to find another way. And we did this by developing a collaboration with the engineers at University of Florida. where we took a hydrogel, which is essentially like Purell, and we used it as a scaffold. We put a protein in it called a chemokine that attracts immune cells. We delivered this subcutaneously, and it attracted T cells and dendritic cells. And then we gave the foreign invader, which is the RNA of the tumor. So the dendritic cells within the hydrogel could take the RNA, present it to the T cells, and activate them, hopefully resulting in an anti-tumor effect. We found this very efficacious even in our most invasive glioma models in the lab. Up here are animals that received no treatment. Here are animals who received that vaccine, and you can see they either have no tumor or much smaller tumors. So we're very encouraged by this, but we have a lot of work to do. So we're now testing the ability of this vaccine to improve T cell function, prevent exhaustion, and result in efficacious migration. And we're also combining with other strategies that enhance T cell function. This is the source of the images that I used today. None of this work would be possible without the collaborators in the laboratory. I'd like to sincerely thank you for supporting the ABTA and thank ABTA for their generous support of our work. Thank you.
Hello, everyone, and welcome to our keynote session. We hope each of you have found today's programming to be informative and emotionally nurturing. We are all here today because we seek a common purpose, a cure for brain tumors. Each of us has our own story that has propelled us to come together to fight this unforgiving disease. For all of the patients, families, and caregivers who are joining us today, the ABTA wants you to know one thing: you are not alone. The ABTA, the medical community, and the research community rise every day to make your tomorrow better. With that said, it is my honor and privilege to introduce you to our keynote speaker, Dr. Courtney Burnett. Dr. Burnett is an internal medicine physician, author, speaker, and brain tumor survivor who will share her unique story as a patient and physician. In this keynote session titled, Embracing the Difficult Gifts of a Brain Tumor, you will hear about Courtney's brain tumor journey that is certain to teach us, motivate us, change us, and inspire us. Please welcome Dr. Courtney Burnett as she shares her story.
Well, good afternoon, everyone. My name is Courtney Burnett, and thank you, Emily, for that very nice introduction. I'm a 30-year-old physician living and working in Minnesota. I also happen to have a diagnosis of anaplastic astrocytoma, or a grade 3 malignant brain cancer. Even as a doctor, a brain tumor diagnosis is confusing, overwhelming, and terrifying. Throughout my journey, I've used writing as a therapeutic way to cope with my diagnosis. I started a blog which gained a very unexpected worldwide following. Incredibly, blog readers told me they found my words to be inspiring, and readers encouraged me to write a book. Now, like many of the doctors here today, I was familiar with the world of scientific writing, but book writing was quite new to me. My first published book, a memoir called Difficult Gifts, A Physician's Journey to Heal Body and Mind, was released this past February. Since its release, I found myself immersed in a very unexpected life of public speaking. Just so you know, I don't have any conflicts of interest to disclose today. First off, I want to thank the ABTA team for asking me to speak today at this incredible annual event. I'm honored, and I hope my story gives you all a small glimpse into the amazing, important work the ABTA does for the brain tumor community. Today, I really hope I can do 3 things for you. First, I want to share some of my own brain cancer journey, starting from around 19 months ago when I diagnosed myself with a brain tumor while studying medicine in Thailand. That was a surprise. Then I want to share some of the lessons I've learned, including how being a patient has taught me to be a better physician, and how much joy can be found in what I've come to call a difficult gift. As I mentioned before, through my own unexpected diagnosis, I turned to writing to cope with these unexpected challenges. The words I write are honest stories of my real life journey to find happiness in this crazy and mysterious life. Even if my story seems unique at first glance, really, it's not. As humans, we're all just trying to find happiness in a life we wish we had more control over. My goal is that my words can help all of us realize that suffering is not unique. When we are suffering, we feel alone. We feel that no one could possibly understand what we're going through. And perhaps no one can know exactly what we're going through, but I've come to think that we actually have much more in common than we realize. Now, I'm a writer, so I love words, but I think pictures often tell the best story. So I'll spend the next 30 minutes or so sharing my story with you through photos, as well as short segments from my blog and book. Now, going into this, I want to be clear that I'm not an advocate for what some people have called toxic positivity in the cancer community. A brain tumor is a heart-shattering diagnosis for anyone to receive. The treatment is not easy, and this emotional roller coaster ride isn't one I would wait in line for. But when life gives us a challenge, I've come to believe that we can do one of two things: retreat or thrive. First, I want to take you way back. From around the age of 2, I've wanted to be a teacher. My mom sent me this photo of me pretending to teach my classroom of stuffed animals sometime around 1994 or so. I grew up in a nice suburb close to Minneapolis, Minnesota. I went to the University of Minnesota for my undergrad, where I majored in genetics, ironically performing my thesis research in the lab of Dr. David Largaspada, who studies the genetics of brain tumors. For years, I planned to pursue a PhD in genetics and become a professor, as teaching has always been my greatest passion. However, I started to find myself more interested in medicine. I volunteered at hospitals and clinics to experience caring for patients, and soon learned that the medical field allows endless teaching opportunities in addition to endless learning opportunities. In medicine, I could learn the stories of my patients, and through these stories, my patients taught me and inspired me. I moved to Chicago for a while, where I studied at and graduated from Northwestern University's Feinberg School of Medicine back in 2017. I then moved back to Minnesota and completed a residency in internal medicine at the University of Minnesota. After spending a year working as a chief medical resident, or a teacher and advocate for students and resident physicians, I very recently started my first attending position as an internal medicine provider in St. Paul, Minnesota. However, in January of 2020, during my third and very last year of residency, I was diagnosed with brain cancer. Let's get to the interesting stuff now. So to understand my journey, I'll take us on a virtual trip. Back in January of 2020, I went to Chiang Mai, a big city in northern Thailand, where I planned to spend 6 weeks studying in a large urban hospital. I've always been interested in Eastern medicine, and I loved the idea of visiting Thailand to work in a complementary medicine clinic. There, I plan to learn Thai massage, acupuncture, and other more traditional Eastern medical methods to supplement my knowledge of Western medicine. I personally believe a good physician is a well-rounded one. Of course, science and evidence-based medicine are key components of my practice. However, I feel there is much more to the field of medicine than science can fully explain. Medicine involves humanity, birth and death, consciousness, relationships, suffering, and relief. Medicine involves cultural awareness, religion, spirituality, and our personal values. Medicine involves science, but so much more than science as well. So I decided to visit Thailand to expand my knowledge of all of these other aspects of medicine in order to become a more well-rounded physician. I'd initially planned to spend my month working in the complementary clinic in Thailand. However, this clinic was closed the first few weeks of my visit, and I was reassigned, quite ironically, to work on the oncology and palliative care services for the first few weeks of my trip. The oncology clinic was unlike anything I had ever seen in the United States. There were at least 200 to 300 people sitting in the waiting room, piles of paper medical records on every desk, and very few computers in sight. The clinic room I sat in was the size of a large walk-in closet, roughly, and held around 8 people at a time. Me, the oncologist I was working with, a second oncologist, the 2 patients both of these oncologists were working with, plus any family members the patients wanted to bring with them. Patient privacy was quite minimal. Trying to keep fear out of my voice, I asked, "How many patients do you see in a day?" The waiting room seemed filled to capacity. "All of the patients that need me," the doctor replied matter-of-factly. On my first day in clinic, I met a 60-year-old woman with newly diagnosed acute myeloid leukemia. She walked into the clinic smiling and brought me and the oncologist bags of cookies. I thanked her quietly in Thai. The oncologist told me it's not uncommon for patients to bring their doctors small gifts in Thailand. Apparently, this patient had noticed me sitting next to her usual doctor before her own appointment, so she rushed to the hospital gift shop and bought a second bag of cookies for me. To this day, I'm still touched by her kindness. This woman, entering what was likely one of the most stressful appointments of her life, took moments of her day to get me a gift. I didn't feel that I deserved that gift. Next, I met a 25-year-old man with a new diagnosis of diffuse large B-cell lymphoma. His young wife accompanied him to clinic. I could see he was trying to hold back tears throughout his visit, as his wife smiled and took notes about our recommendation to start urgent treatment. We brought this young man directly to the hospital to start IV chemotherapy. I ended up seeing him and his wife the next morning on hospital rounds. To my surprise, the couple recognized me immediately and smiled. I didn't feel that I deserved this smile. Later that afternoon, I met a 50-year-old female nurse with a new diagnosis of late-stage chronic myeloid leukemia. She also brought me cookies. She also smiled. She cracked jokes and laughed as we talked about her life expectancy of mere months. I did not feel I deserved her cookies, her smile, or her laughter, but she gifted them to me anyways. After a few weeks of fascinating cases like this in the oncology clinic, I was finally scheduled to start working in the complementary medicine clinic. Unfortunately, I woke up my first day feeling like I was coming down with a cold. Now, this was very shortly pre-pandemic, so I took some ibuprofen, threw on a mask, and decided to walk into clinic anyways, since this type of experience doesn't come along very often. Also, it turns out physicians often make the very worst patients. My symptoms improved with my walk into clinic. It was a beautiful sunny day as usual in Chiang Mai. And arriving at the clinic, I was quickly ushered into the Chinese medicine department, where a doctor asked if I would like to try acupuncture myself to learn what it was like. I happily agreed, having never tried this before. But as I lay on the table face down with needles covering my back and neck, I started to feel much worse. I had to end my session early and head back to my apartment. Now, I didn't have a thermometer with me in Thailand, so this personal case report is sadly lacking in any good quantitative data. I'm sure you're all quite bummed that I don't have any colorful statistical graphs in my presentation to depict my fever curve. Sorry. After I arrived home, my fever intensified. I remember lying in bed when I suddenly felt my left hand muscles contracting rapidly, almost like my hand was squeezing into a claw without my permission. This was quickly followed by a sensation of my throat muscles tightening. All of this lasted around 30 seconds and then resolved. I was awake and alert the whole time. This was odd, and I couldn't explain it. So I drank some water, took another ibuprofen, and napped the rest of the day. I woke up the next day feeling better, so I went to work and then walked to a small local market on my way home to find some souvenirs. As I stood outside of the open-air market, I felt that same awful muscle cramping start up again out of nowhere. My left hand muscle started to contract rapidly, squeezing my fingers together. And a few seconds later, hand now relaxed, my throat closed up on me. This time, the saliva pooled more rapidly than before, and I felt a small drop of drool drip onto my shoulder. Ugh, I was scared. Once again, 30 seconds later, my muscles relaxed and everything was back to normal. I tried to medically rationalize these random temporary muscle cramping episodes. In my mind, I thought, this could be an electrolyte imbalance, an infection, maybe just some anxiety. I guess it could be a small or focal seizure as well, but why? As a 29-year-old, otherwise healthy woman with no other symptoms and no reason for a seizure, I tried to brush this off and decided I'd worry more if it happened again. Well, it happened again, 5 more times. I would soon determine that these were indeed focal seizures, my body's one and only symptom of brain cancer. Literally 1 week before this, I'd had no symptoms of the golf ball-sized tumor growing in my right frontal lobe. No headaches, no nausea, no vomiting, no speech or balance issues, no weight loss. I wasn't even tired. I felt amazing, actually. I was in the best shape of my entire life. I'd been volunteering at an elephant sanctuary without a care in the world. When my largest and longest focal seizure occurred, it was late in the evening in Thailand. Now, the emergency system works quite differently there, and I was not excited about the idea of spending the night in an emergency center without being able to describe my symptoms in the Thai language. Instead, I called my emergency contact at Chiang Mai University, one of the doctors I had worked with in palliative care. Fortunately, she also happened to be a certified neurologist. I described my symptoms to her on the phone late that night. I felt like a complete fool, bothering her at night for what was likely nothing to worry about. But I decided to explain my symptoms as a doctor would, consulting another physician on an unusual case. And this strategy worked. The doctor agreed with me and told me to meet her the following morning to walk with her to the neurology clinic for an appointment with one of her colleagues. The next morning, I found myself sitting alone in the very same clinic I had worked at the previous week. In the busy waiting room, I planned out the story I would tell the neurologist. If this clinic worked in a similar way to the clinic I had been working in last week, I knew I would have somewhere between 2 or maximum of 5 minutes to explain my symptoms and advocate to get a brain MRI. An hour later, with sweaty palms and a voice shaking with nerves, my name was finally called. I walked into the clinic room, shared with a second patient and her physician, and introduced myself to the neurologist. Here I was, both patient and physician for the first time in my life, unsure of my identity or how to act. I told the doctor my story. I told him I was also a physician. I told him I knew these symptoms could just be a figment of my anxious imagination, but I was confident that I had enough signs pointing to seizures that I required an urgent MRI. The neurologist looked somewhat quizzically at me, not quite sure what to make of a young, outspoken physician/patient sitting in front of him diagnosing herself with seizures. The neurologist had me do a brief exam, an exam I was actually quite familiar with myself. As the exam went on, his expression changed. We realized together that my left hand was slightly weaker than my right hand. My shocked eyes looked into his. I agree with an MRI, he said. A nurse took me back to the MRI room, gave me a hospital gown to change into, and asked me to sign a financial agreement. In Thailand, medical payments work much differently than in the United States. Medical quotes are all predetermined. Patients are asked to sign agreements before any procedure, test, or treatment even begins. I signed a bill agreeing to pay about $250 US for my MRI. I laid on the table, eyes closed, and tried to meditate. Now, compared to MRI machines at home, as many of you have been in, this one was large and quite frightening. I didn't receive headphones playing relaxing music, and I wasn't offered any medication to claustrophobia. Instead, the radiology technician played some intense Thai pop dance music and left the room. Lying there in the machine, I decided I must just be losing my mind. These must be panic attacks, I thought, only to feel my left hand tighten and my throat muscles contract as the music blasted around me. I lay in that MRI scanner for about 45 minutes before the tech re-entered the room. Expecting him to say I was finished, I was surprised when he instead asked me to sign a second financial agreement. In very limited English, he asked, "Is it okay to charge you another fee for the surgical planning MRI now?" "Excuse me, a surgical planning MRI? I assume this means you found something," I asked him. "Yes," he responded. Keeping my questions brief due to our language barrier, I asked him, "How many masses did you see? One or multiple?" "One mass," was all he said. I agreed to pay the money, and I spent the next and longest 20 minutes of my entire life creating a list of possible diagnoses in my mind. Possible diagnoses for one brain mass leading to recurrent focal seizures. Brain abscess or an infection was the first thing on my list. I had spent days playing with many beautiful but dirty elephants. At one point, I'd even hopped in the stream to bathe them. No doubt I had caught an infection that had invaded my brain. How could I have been so careless, I thought. Next on my list was a camera smudge. I was lying in an ancient machine with outdated technology around me. This must just be a technical glitch and a big misunderstanding. Now, the inner patient in me didn't want to consider the last possibility on my list, but the inner doctor in me knew that I must. A brain tumor, one mass, one life-changing realization. As my scan ended, I realized the neurologist I'd met earlier that day told me he wouldn't have time to read my scan until the next morning. With a life-threatening diagnosis on my list, I knew that tomorrow might not be soon enough. I got off the MRI table and walked into the technologist's command center. I said, "Sir, I'm a doctor. I need to see my scan and take a few pictures." Now, being the only physician in the imaging center at the time, the technologist reluctantly agreed and scrolled through the images on his computer. On the screen in front of me, I saw a large, irregularly shaped mass in my right frontal lobe causing minor mass effect, or in medical terminology, some smooshing of the brain to a place it really shouldn't be. As I numbly snapped a few photos with my cell phone, I tried to process what I was seeing on the screen in front of me, what you're seeing now. My very first thought was, oh, shoot, I'm going to have to tell this patient some really terrible news today. She has a massive brain tumor. It took me a few minutes to realize that that patient was actually me. The radiology tech said, "Okay, that's enough time," and abruptly kicked me out. "The radiologist will call you tomorrow." Speechless, I left the clinic. There I was, standing on the sidewalk in Thailand, trying to process what I had just seen on my own scan. The physician in me knew this was either a tumor or less likely an infection, both of which could be considered emergencies requiring pretty prompt evaluation and treatment. Outside the imaging center, I realized I should be admitted to the hospital. I shouldn't be standing alone on the sidewalk in Thailand waiting for a bigger seizure to come along. So I texted a picture of my scan to my colleague and told her it looked like a tumor. She agreed, and together we decided I should be admitted to the hospital later that afternoon. I ended up being a patient in Thailand for 3 nights. Some of you might appreciate this picture on the left was actually a balcony, open air balcony from my hospital room there. Now, although the medical care system works differently in Thailand than in America, many of the basic logistical aspects of my hospital stay were quite familiar to me. I knew that the doctor would order some labs and some vital sign checks, so I was prepared when the lab tech came into my room, stuck an IV in me, and took a few vials of blood without saying one word. I was also prepared to be woken up from sleep every few hours for routine monitoring of my heart rate, blood pressure, and oxygen levels. Every time a familiar activity happened, I felt grateful. Okay, I know what this is. I can read this blood pressure measurement. I know this number is fine. When an IV pole was wheeled into my room and fluids were hung next to me, I thought, okay, this is a familiar fluid. I know why the doctor ordered this and I know what effect it will have on me. None of this was explained to me as it was happening, partly because of a language barrier, but more honestly, because none of this is explained well to patients. As a physician, I often make the decision to order labs, place IV lines, start fluids, and do vital sign checks without telling my patient I'm going to do these things. Of course, I always tell my patients of critical changes in their treatment or test results. But routine tasks like these are as commonplace in medicine as sending an email would be in the corporate world. Physicians don't always remember that to patients, an email feels very different than a blood draw. This experience completely changed my perspective. I realized how knowledge is such an incredible tool. I decided then that when I was back at work, I would try to spend a few extra minutes with my own patients and provide them with knowledge to feel safe, respected, and understood, rather than afraid and confused. Being a patient is absolutely terrifying. As a patient, your autonomy is gone. You find yourself with a million unanswered questions and you spend hours waiting for a doctor you just met to tell you the worst news of your life. I hope that I always remember this experience with absolute clarity so that I may act as a more compassionate physician to my own patients. In the end, one of the greatest difficult gifts of my own experience turned out to be my experience as a patient. Through this, I've learned to be a better physician. My colleagues in Thailand and Minnesota arranged for a flight which would take me back home as quickly as possible, straight to the University of Minnesota, where I worked at the time, for urgent brain surgery. I woke up in my hospital bed the next day, suddenly realizing this was my very last day in Thailand. My journey had come to an abrupt end, although when I think back on it, my real journey was really just beginning. Prior to my hospitalization, one of the things I most looked forward to in Thailand was the opportunity to chat with a monk. Many temples in Thailand advertise times when monks are available to have free, informal chats with visitors. In preparation for my eventual monk chat, I had kept an evolving list of questions on my phone. They included things like, what's the key to finding peace with yourself? And how do you find happiness in a world filled with inevitable suffering? Lying in the hospital bed that day in Thailand, I realized I'd never made it to my monk chat. As I looked at these questions, I started to cry. At that moment, more than ever before, I needed guidance. I needed answers. I needed a way to find peace and happiness to cope with this very unexpected suffering. My work in Thailand had fortunately introduced me to an incredible team in palliative care medicine. When my palliative care colleagues stopped by that day, they asked if there was anything they could do for me. One common role of a palliative care team is to provide spiritual guidance for patients when they request it. Now in America, palliative teams often coordinate with chaplains or priests to administer prayers or last rites. In Thailand, there aren't many priests, but there are many monks. I asked my palliative care friends for one favor that day. Can you find a monk to stop by and talk with me before I leave, I asked. An hour later, a monk in a bright saffron robe walked into my hospital room. I sat across from the monk and told him about my brain tumor. He listened to my story with hands resting calmly on his lap, smiling the entire time. After I'd finished, the monk spoke in English, summarizing everything I ever needed to know about life in 2 minutes. In these 2 minutes, the monk, whose name I now can't remember, said to me, "Be happy." I nodded, unsure of really what to say. Find time to calm the mind, he went on. I let this sink in. Be happy? Had this monk forgotten I just told him I had a brain tumor? He went on to say, this happened for a reason. Smile. Do not cry. Hearing this, of course, I burst into tears and smiled foolishly at the same time. The wise monk saw my tears and said, do not worry about your illness. Illness and death are a natural part of life. I had never in my life felt the kind of deep inner peace I felt when the monk spoke those two brief sentences. Do not worry about your illness. Illness and death are a natural part of life. As a physician, I knew this, of course, yet I had never actually considered that this fact of life would also apply to me. I'm young and healthy. Why would I be dying? How foolish, I realized. We're all dying. Death had found me, and I was not prepared for it. The monk was right. How simple, yet how profound. I've had what feels like hundreds of years of schooling, yet nothing in my education was as helpful to me as this monk's brief lesson. As a physician who has seen illness and death more times than I care to count, I can tell you without a doubt that only one thing is medically certain in this life, and that is death. Now, a lot has happened since that day 19 months ago. I returned home to Minnesota and I underwent my first of 2 brain surgeries, a partial craniotomy with a biopsy in February of 2020. That surgery diagnosed me with a grade 2 diffuse astrocytoma. I recovered and returned to work just in time for the COVID pandemic to start, and a few weeks later for my second, more intensive brain surgery to be scheduled in order to resect as much of my tumor as possible. Surgery was fortunately very successful and I was left in good shape apart from some mild facial paralysis, something I've had to work on over the past year to slowly improve. My second surgery led to an upgraded diagnosis of a grade 3 anaplastic astrocytoma. Unlike when I fly Delta and beg for an upgrade, this time I really didn't want it. I started working with a fabulous team of neurologists, neuro-oncologists, radiation oncologists, and pharmacists who helped me get through standard of care treatment during the pandemic. I completed 30 cycles of radiation and 42 days of induction oral chemotherapy with temozolomide, followed by 6 months of maintenance chemotherapy. I finished this treatment last fall, and I've been fortunate to have stable scans ever since then, with no evidence of progression and minimal remaining visible tumor. I owe so much to the forward-thinking, powerful team of brain tumor physicians, researchers, philanthropists, and supporters who gave me quite literally another chance at life. Through this diagnosis, I've been afraid, I've been anxious, but I've also been hopeful and happy and grateful. I was diagnosed at age 29 with an incurable terminal cancer. And let me tell you, when someone told me I was dying, that was when I started living. While undergoing treatment, I graduated from medical residency and spent a year working as a chief resident, teaching and mentoring new resident physicians and students. Despite brain cancer, I wrote a book. I continued to work full-time, and I started to give motivational talks to people all around the world. I owe all of this to my surgeons, physicians, researchers, and the supportive community I've met, many of whom are on this call today. Thank you. We all know that at this time, brain cancer is considered incurable. Mine is stable. I am alive, but more importantly, I'm living. So now you know a bit about my story, a bit about how being a patient taught me to be a better physician, and a little bit about my new perspective on life, a perspective which helps me find joy through even the most difficult of gifts. My difficult gift, my brain cancer, was one of the most unexpected, terrifying, beautiful, and life-changing gifts I've ever received. Brain cancer reminded me that my time, all of our time on this earth, is short. Through dying, I've learned to live. Through suffering, I found joy. Through the loss of the guaranteed future I once envisioned, I've actually found peace and freedom. I'm more alive today than I've ever been before. My difficult gift not only inspired me to write and share my story, it also taught me to be a better, more relatable physician by being a patient as well. Sometimes I reflect on my recent experience and I wonder if all of this happened to allow me to teach others in a way that I never dreamed was ever possible. Time will tell. Thank you all so much for listening. Please reach out to me with any comments or questions on my personal blog or email, both on this slide. I appreciate your time. Thank you.
Thank you so much, Courtney. That was a wonderful presentation. We do have time for questions now. Just as a reminder, if you have questions that you'd like to ask Courtney, please type and submit it using the Q&A box in your control panel on the bottom of your screen, and we'll answer questions as time allows. So Courtney, one of the first questions we have here, I know you mentioned that you came back to the US and started treatment just in time for the pandemic. How has the pandemic impacted your experience as a patient? What are some ways that you've coped with social isolation during this time?
I mean, great question. I think we can all say it's impacted all of us quite a bit. I think probably the most shocking experience was my first brain surgery was just before the pandemic really hit the US. And so I was able to have family, friends, everyone with me for that surgery, supporting me through. My second surgery, I was completely alone. The hospital had a 0 visitor policy. I mean, as many of us remember from early days of the pandemic here. So my parents dropped me off outside of the hospital I worked at, and in I went, walked myself to the ICU bed and went surgery and woke up, to no one there. I think being in the medical field was an absolute blessing in that setting because I had friends and colleagues at the hospital that could visit with me. But it obviously was a big impact that I can't imagine being a patient absolutely alone, not knowing anyone in the hospital, not having anyone with you. It's absolutely makes it even harder than it would be normally.
What advice can you share for patients and families who are trying to find that social connection? They might not have friends at the hospital that can stop by and visit them. How can they still engage with their loved ones and find support?
It's a great question. I think for me, all of the various brain tumor and brain cancer communities online have been incredibly helpful. ABTA is one of many I've found through social media outlets, through various blogs, through Facebook groups, you name it. I found so many great communities that offer virtual get-togethers and networking, and I've met so many incredible people who are going through something very similar to what I'm going through, and that's been inspiring and gives me a lot of hope.
Thank you. Definitely connecting with other patients, survivors, and caregivers is a great way to know that you're not alone and there's others out there who are going through the same thing.
Absolutely.
You mentioned resources. I know you mentioned the ABTA, and thank you for that. What are some other resources that you found helpful through your brain tumor experience?
Yeah, great question. So to give a shout out to the Brain Tumor Network, which I'm a big fan of. They've helped set me up with some nurse managers and care coordinators. They've helped me find some support groups and second opinions. So they've been very helpful. I also have, as I mentioned, I think through some blogs and through some online social media groups, those have been great. Also just finding local brain tumor or other cancer survivor or cancer sort of fighter groups have been helpful. I have, one in my local community that I like to go to. Often we meet virtually or in person, and depending on how things are going with the situation, we'll see what happens. But I think those groups are fantastic.
Thank you for sharing that. I hope that some of our audience members can also find benefit from resources like that. Now, I think one of the biggest clouds that hangs over our heads as patients is that what if, the fear of the unknown. How is that something that you've coped with yourself?
That's a great question. That's really what a lot of my book and my writing is about, because that's a question that we can all relate to, whether you personally have brain tumor or you don't, right? We're all thinking, when will this happen? And I've written a blog and a chapter in my book about this issue and kind of how I switched my personal thinking when I get into those ruts of spiraling and, thinking, when will my cancer come back? When will things get worse? When will this happen? And really, I try to switch this sort of when will question to today, I will. Instead of this passive, when will things get worse? When will my treatment stop working? When will my MRI show something scary? When will my loved one's MRI show something scary? You name it, whatever your worry is. I try, and whenever I have those thoughts, I think, today, I will go for a walk. I'll play with my dogs. I'll go to work. I'll smile because I'm still alive and because none of those things have actually happened yet. And I think the foundation for a lot of that is the practice of mindfulness. And that's something that I practice and that I study and I write about. The practice of that found in a lot of mindfulness and meditation courses, also in a lot of Buddhist philosophy, but I think a lot of those techniques can be applied whether you're spiritual or not. Just this idea of finding peace with living in the moment and trying not to worry about what will happen down the road. None of us know whether we have a brain tumor or not.
Thank you for sharing that. I know living in the moment is not always easy, but I love your advice of mindfulness and making notes of the little wins that we have each and every day. Another question we have here from one of our attendees is about energy levels. You know, as we're going through treatment, we can feel very fatigued, we can feel tired. How are some ways that you've tried to maintain your own energy levels?
Yeah, that's a fantastic question. And I'll be very honest that it's still something I work on, still something I have to really listen to my body every day and decide how much energy do I have today? You know, how much energy can I give to all the things I need to do, my job, to my family, to my other commitments. Absolutely, I think going through treatment at various stages, the fatigue can be unbearable sometimes. I definitely experienced that myself. I think part of it is just listening to your body when as simple as that is, when you need the time to sleep. Don't feel bad about, napping multiple times throughout the day. And I think, it can be actually quite scary. I think I see it as a patient, of course, but to have a loved one with a brain tumor, you may think, oh, my loved one with a brain tumor is napping all day or sleeping all the time. Look, this can't be good. But I think honestly, there are just times during this journey where you have to listen to your body, and it's okay if you need to nap a lot of the day. It's okay if you need to take some time for yourself and get that energy back. Coffee also helped me.
Yeah, I definitely think we have to go easy on ourselves and give ourselves a break sometimes. Another question we have here, I know you mentioned that your tumor is currently stable. Are you in active treatment at the moment? Are you in wait and watch?
Good question. Yep, I'm just waiting and watching. So I ended up doing 6 months of maintenance temozolomide. I know some people with this, the grade that I have of anaplastic astrocytoma do 6 to 12 months, maybe longer, depending on your unique situation. But I elected to do 6 and things look stable. So I'm at this point getting MRI scans about roughly every 2 to 3 months. So lots of MRI time. So, I get used to that sort of anxiety we all feel a little bit before those scans, but learned also to just try and take them as they come.
Thanks for sharing that. I know you mentioned you had colleagues that were supporting you through this, some friends. Who would you say was your main support system? Did you have like a caregiver, or who was in your corner helping you through this?
Good question. So I think, I guess a couple answers to that. First off, my family and friends, I couldn't have done any of this. I couldn't have survived this whole experience without them. They're absolutely I'm very fortunate to be incredibly close with my family, most of whom live in the area. Many good friends who live nearby, and my significant other. So I've had just a lot of help from all of those people. I was really, really fortunate with my own diagnosis in that physically, I actually did quite well after my surgeries. I know that that's not always the case, but I was lucky in that I didn't need a caregiver for helping with physical things. I definitely, a few days after surgery, those days were rough, and I had my family helping me out with a lot of things. But really, it's been more of an emotional support that I've needed, and my family and friends, as well as, again, many of these brain tumor communities have helped me do that.
That's fantastic. I'm really happy to hear that you had such wonderful support from your family and friends through this, because it can be so challenging when you're going through it on your own.
Absolutely.
A question that we have here, we talk about survivor's guilt, but sometimes as patients, we can also feel guilty for our loved ones who are helping us go through this or seeing us go through this.
Mhm.
How have you dealt with that form of guilt?
That is a fantastic question. I'm glad someone asked that. I've never thought of it as guilt in that way, but you're absolutely right. I think of it as almost, I've had a lot of difficulty feeling like a burden on my family, on my loved ones, right? When I've needed, whether it be, emotional support, or those times I have needed more physical support right after my surgery, is definitely. And I think that many of us brain tumor warriors, fighters, thrivers, survivors, whatever you want to call it, right? We deal with this lifelong. And I think it's a feeling that doesn't go away, because we always have this thought, or at least I do, of, oh, it's very hard to think that someday things could get worse and I could leave my family in a lot of pain, right? I think it's a hard thing. Yeah, it is. It's like a a variation on survivor's guilt. I like thinking of it that way. And again, I think for me personally, a lot of how I deal with that is again in mindfulness and in being really open about what I'm feeling with my loved ones. I think prior to my diagnosis, I was very introverted. I never could have dreamed I would become a public speaker and speak so openly about these very intimate details of my life. But I can say that this diagnosis changed that. And in speaking this openly about things, it's given me so much help and so much peace of mind. So I would say that is, the mindfulness and the openness are very helpful in that setting.
Thank you so much. And I'm sure I'm speaking for everyone when I say that I'm really glad that you did decide to start publicly speaking and sharing your story, because it is incredible. And I'm sure that it has been a source of support for everyone who's listening today. It certainly has been for me. Unfortunately, that is all the time that we have for questions for now. Thank you again, Courtney. I'm going to turn it back to Emily.
Great. Thank you all for having me.
Thank you so much. And thank you so much, Courtney, for sharing your story with us today. I echo everything that Brain just shared. It is truly evident that you are not allowing a diagnosis of a brain tumor deter you from living your most fullest life. Your story truly inspired us today and encouraged us and made us, remember to make every moment count. So may we learn from you to find light in every difficult gift. And thank you so much for being with us today. I now would like to welcome Carla Varner, ABTA Board Chair, who will introduce our closing speaker.
Thank you, Emily. Earlier today, you heard me speak a little bit about the history of the ABTA. And ladies and gentlemen, it is now my honor to introduce you to ABTA co-founder and honorary board member, Susan Kramer. Thank you. Susan?
It is an honor to be here today with all of you. As co-founder of the American Brain Tumor Association, it is astounding to witness the advances that have been made in research, treatment, and care, and to see how the ABTA has contributed to those advances. When we started this organization 48 years ago, we could not have imagined what it would become. I'm so proud that a small family foundation that started in my parents' basement in an effort to save my daughter, Stephanie, could grow to become a resource and advocate for people who are going through the same thing we went through. My daughter, Stephanie, was diagnosed at only 5 years old. We were told that Stephanie had 6 months to 2 years to live, with the possibility of the tumor coming back even after treatment. This tragic news ignited a fire in me to find a cure, which led to the birth of the American Brain Tumor Association. When we lost Stephanie at 7 years old, I felt I had to continue in Stephanie's memory to help others find answers and cope with this terrible illness. I am so proud that this organization now serves people all over the country and the world. We hope today's programming offered hope and encouragement, but mainly to show you that you are not alone. The ABTA is here to serve you through information, support services, and research. Thank you.
Thank you so much, Susan. Your determination to develop an organization to serve patients and their families are the building blocks of the ABTA. We thank you for joining us today. We invite those of you who wish to meet fellow attendees from around the world and engage in meaningful conversations, play some trivia, and win some prizes to stay online and participate in the ABTA Social Hour, taking place in just a few minutes. This is the only session where you will get the opportunity to turn on your camera and say hello and meet fellow attendees. Please note this session will not be recorded. Additionally, we invite you to connect with us and share your experience through social media. For those of you who have to go, we encourage you to join us again for another exciting day tomorrow. Our program will begin at 10 a.m. Central Time, and we will include sessions on clinical trials, treatment resistance, and more. You will also get the chance to hear from the patient perspective during our patient and caregiver panel. Please use the same link you used today to join tomorrow's program. This now concludes day one of the 2021 ABTA National Conference. Thank you to our wonderful co-chairs for leading such an incredible program. Thank you to our speakers, sponsors, and researchers for sharing with us today. And thank you to our titanium sponsor, Northwest Biotherapeutics, and our platinum sponsor, SDP Oncology, and Novocure for their support of this year's conference. And last but certainly not least, thank you to all of you who have joined us today. And we look forward to seeing you again tomorrow. Take care, get some rest, and have a good night.